All right, get off that book like that and stop acting out. All right, what's going on, family? Peace, power, and profit to everybody today on this Throwback Thursday or Throw It Forward Thursday, or however we like to bring relevancy to the day. I thank you guys for tuning in. Today is obviously if you want to call it a important day. We're having the FDA go through the review process of trying to ascertain whether or not the Moderna vaccine will have its day of reckoning, so to speak, to get emergency use authorization. And so easily, of course, you guys can go and pull this up and watch it yourself. But I thought it was a great opportunity also to come on, share it live as well, for archive purposes too, to always, where you can go back and study it, because obviously I'm a fan of these kind of different panels and briefings, and I've sat in on many of them, obviously starting out early on in my little trading career, trading a lot of biotech stocks or whatever, and just the process that these things go through, and you learn so much about uh, the government side of the house, the company side of the house. The whole process in general is really good for just edification of knowledge and health and awareness, I should say. So we're always talking about preventive maintenance for a reason, because we want to be well, equipped with the best information to make the best decisions for our health and well-being. So again, thank y'all for tuning in, everybody on the Facebook family. I know you guys are gonna be, probably be popping in and out, whatever the case. Thank y'all for tuning in. All my family over there at Twitter, all the We Trade HQ family as well. Thank y'all for tuning in as well. Again, it was not necessarily advertised. I just kind of went live like impromptu, so it's not necessarily a plan value or speech in lane that I have in mind, but I will have momentary discussions in and out the FDA briefing, and so I'm just gonna let it play. I'm gonna let them talk. Kind of, we listened to the Pfizer one. I don't know if you guys did personally or not, but it was a long one. But they are interested, again, watching these doctors kind of go back and forth and try to explain their position about these certain ideas of vaccines. And this is good because there's a lot of conversation going around the country and the world whether or not people feel comfortable about taking these vaccines. That's always going to be a topic of discussion. It's not a one size fit all reality, right? So we have to be, cognizant of that. So we got to make sure people who feel very strong about vaccines know that there's always two sides to every position, right? Because there's no 100% finite proof value of anything. And those who are anti-vaccine have to be just as open-minded to realize whether or not there's a use case value that can help put them in a better position where they can also uh, you know, be protected in some kind of way, of whether or not they'll feel like it's necessary based on the science and the data. So again, when you weigh both sides, you have to be, equal in that position or that perspective. It's very important that we don't lose sight of the overall agenda, what we're trying to accomplish. And I say we, meaning the people trying to get the best value of information to utilize, um, you know, the data, whatever, whether or not you will take the shot or not take the shot, whatever, and which or what company will you, likely choose, right? Because right now we know that Pfizer obviously got their emergency use authorization last week. This week is all about Moderna, right? We spoke about this a little bit last night on our Urban Economic Empowerment show. We kind of went in about some of the different trades that you particularly could take. And we were spot on, of course, with the Moderna trade. I'll go into that later, but we called it, right? We called it. All right, we called the 140 bump and the 145 even up to the 150s. And so, the stock went up to like 148, right? So again, that's just, like I always say, that's just, our opinion. This is not investment advice. We don't give that over here. That's my disclaimer. We just offer it in an education perspective and allow people to make the type of decision that's good for them. But nonetheless, we got paid, right? Getting paid, young blood, getting paid. And so just want to make sure I reiterate that, because again, that's why a lot of us, or the interest that a lot of us have in this, because we are invested. We're trying to also, long term, but also we're going to take a position just for momentum play for the value that people are excited about the opportunity to trade in such an opportunity, or a historical moment, because this is the first time ever in U.S. history, global history, where such a scenario where they brought a vaccine to trial in less than really about 7-8 months, and it's now being reviewed for emergency use authorization based upon the pandemic, of course. But these reviews typically take 5, sometimes 2-3 years tops. Some people take longer. I've seen some trials go longer, like 7-8 years. They still ain't got approval. They got to go back to the drawing board. They got a CRL letter. They got to go back, figure out what went wrong, come back, and sometimes they'll get strapped for cash. They'll run out of cash. They got to dilute the shares, and it's just a big mess. So in this case, of course, they were the two top ones that came out the gate that kind of showed pretty good, efficacy data. AstraZeneca obviously was one, then they kind of fell to the wayside. Johnson Johnson is still in the race. There's Novavax. There's a few others as well, but some of these marquee names, they really, took the bull by the horn, so to speak, and it's been really, putting themselves in a position where they can get this early authorization, which we're rendering and seeing right now, right? Again, with the panels, how it's going to go again, like I said, they're going to have a all-day discussion breaking down the pros and cons of the vaccine. They're going to be going over the efficacy data, meaning how well did it fare up against the strenuous testing that they was able to provide under such a short-term notice. And then from there, they will obviously prevent, they will submit their votes, right? Whether they nay or yay or whatever, right? Now, it'd be great if they are, go behind that and cut them stimulus checks in Congress, right? Make it a twofold good party day, but you know how that's going to go, right? That thing may get signed late Friday or Saturday, just, as of late, just going over the stimulus talk. And we'll talk about that a little later too, right? Because the stimulus is definitely the biggest thing that the markets are waiting on. Again, the vaccine is great and all, but like most scenarios with Wall Street, they feel like they can't make money off of it. They're not necessarily overly concerned. They feel like that's a more public Main Street reality. They're concerned about how can, is there going to be any money that's going to be made off of it, right? Because again, these people are vultures and they're buzzers. Y'all know how Wall Street is. They can care less about your health and safety, because they're going to make money off your death one way or the other, right? And so that's just that's just the truth of this stuff, right? That's just how this stuff is. So again, so with that being said, guys, we're gonna get this thing popping. Thank y'all for tuning in again. I'm gonna run the FDA live stream as they going live right now. Again, thank y'all for being patient. Thank y'all for coming over to the other channel. Of course, y'all like, well, why are we over here on this channel? Obviously, right? There's some problems and issues going on with the other channel. So obviously got flagged again. And so I got to go through that again and submit some letters and do my own independent review, right, to get that stuff taken care of. So again, nonetheless, that's why we have a backup. We're gonna still keep grinding. Y'all know I am pretty much definitely a rebel when it comes to these social medias. I can care less, but I understand they do provide a window where we can come together and exchange information. So I'm not necessarily trying to test the limits, but I really don't really care at the end of the day, just to be honest. So, but that's just that. But nonetheless, we'll make do with what we have, and we'll go forward with this information one way or the other, and we'll see what will come again from this. The information, false vaccine, everything else, and we'll weigh the options and see whether or not it's a good opportunity for people to, feel like if they have to or want to take a vaccine, or who would they would go, who would they would choose or whatever. We can discuss that as well. All right. So, like I said, it is a snow day. It was snowed in, obviously, in my neck of the woods. So I'm entertaining as well. But I also want to be on live with you guys too in the same breath. So bear with me if you have or hear any background noise within my area, on my microphone, whatever. But nonetheless, it's all about the vaccine news. That's what we're here for. And we'll take this as a great opportunity, so that we can utilize this as a way to, again, to build up our knowledge bank about about how these things work, right? The processes and all that, right? So again, thank y'all for tuning in. Let's get it, right? And let me see here if there's anything else missing. Yeah, I think we got everything pretty much on point here. And so I'll just go ahead, like I said, cue the FDA meeting that's in progress right now, and we'll get at it again, guys. Got any things you want to pop and say in the back chat? Go ahead, punch it in. I'll be able to, being able to type in the back chat, so that way I can keep up with any kind of questions, or if we, whatever that we can share with amongst each other. We want to kind of go through the process of listening in and seeing, what they're talking about. All right. So again, as you see, I always got some kind of crazy little meme or thumbnail up. So of course, a miracle vaccine, right? Rushed into production after only what, a brief trials, right? The bottom half of this is not able to be shown, but it said what could actually go wrong, right? What could go wrong, right? Rushed into production, right? What could go wrong, right? Like, and see, that's the part again where a lot of people got to realize, just because, yeah, it seemed to be an end all be all because it's going to really put a damper on the idea of the virus becoming just even more virulent than it is. At least there's something out there. But like in most cases, like even when they have a first model of anything, it's going to come with its problems too. So we do understand we're not this type of people who are romanticizing the idea just because it's a vaccine is going to save the world. That's not how this stuff works. There are going to be side effects. And anybody telling that they're not, they're lying. There are people who have died during the trials. If they're trying to tell you that it wasn't because of the vaccine, they're lying. Don't listen to people who romanticize the truth, right? You're not, uninformed. You're not ignorant. You do have common sense. These things happen a lot. And it's a reason why, these companies that are for profit, not ethics, they are for profit. They're not for ethics. That's why they pay a lot of money to governments to keep from being sued, which that's the next publication that we're going to talk about earlier too, or later today, about now that, it also came out that Moderna or Pfizer cannot be sued in the event that anybody have an allergic reaction or a death from the vaccines, right? And so that's just how this stuff works. So again, but we're gonna delve into that as well at some point. Once again, I kind of get through some of the information that we're going to discuss or hear, I should say, excuse me, hear from the live stream in itself with the FDA panel that's going live right now, and we'll come back and double through, y'all that stuff go. Y'all know how we get, right? We'll have our after party discussion, so to speak, right? Well, we can go in, and hey, just put everything on the table, right? Lay it all out, right? Lay it all out and see, whether or not if it's, feasible, or pertinent information for us to be adherent to going forward, dealing with this vaccine, all of this stuff basically. All right. So thank y'all again. Peace, power, and profit. Let's make it happen. Let's get it. Let me go over here again so I can light up. I'm sharing it a little bit too, again, trying to get it out to a little social media. Again, I know I did this impromptu on y'all, so it be like that sometimes. It's a busy day as it is myself, like I say, because of the snow day, and I got my hands full a little bit here, but y'all know how we work, right? We adapt very well under pressure, right? We are really good at adapting under pressure. And so that's just the gist of this stuff, right? All right, so all right, I think I got most of this stuff. In some of the most places I know I want to share, I just want to make sure I get it out to a few people in the event they want to stop through and hang out with the family. We will be happily uh, and excited to accommodate them. Peace, power, profit, the family. Got some family coming in. What's going on, family? Eddie C's in the building. Obo Divine Full in the house. Brother Matt is in the house. Right. We have a family legendary eights in the legendary eights in the house. Legendary eights in the house. Peace, power, and profit to you. What's going on? Over Chris Woods in the house. Go subscribe to his YouTube channel. Don't think twice. Get that good content. Right. Go subscribe. Go subscribe. Go subscribe. What's going on? Over Marquise in the house as well. Of course, like I said, we was up burning the midnight oil like we always do. Yeah, we take training to the next level, right? We stay up, what I'm saying, with the night owls and wake up with the roosters, right? That need to be a t-shirt moment, right? For investors and traders, right? We have a unique, a unique energy that no other, people have or share with us because we are, so, ensconced in the ideal of wanting to be the best we could be. So we take our training very serious. So we sacrifice a lot of our time and all that to become better at what we do. So, it's hey, y'all know how this stuff is, right? We love it, right? We love what we do. So staying up late ain't nothing for us, right? So thank y'all again for tuning in. Peace, power, and profit. And let's get this thing popping, right? Let's get this thing popping. And, so we can, see what's going on with that. So I'm going to do a brief little, you know, let me see what I got here really quick here. I kind of went through the Weeble scenario real quick. All right, yeah, so that's it. So yeah, let's get it. So of course, it's all about the vaccine, right? Either we're gonna drink bleach, right? Y'all know how this stuff go. Either we're gonna have to drink some bleach, right? If the vaccine ain't working, we're gonna drink a little bleach, or we have to keep wearing these masks, one way or the other, right? One way or the other, we got to make sure we protect ourselves. So just make sure we are, we're aware of that. There's no getting around that. So nonetheless, we're gonna be prepared one way or the other. So with that being said, let's get at it, right? Let's get at it. All right. So let me crack this up over here. All right. And we are ready to rock. All right. Again, thank y'all for tuning in. Like, share, subscribe to the channel. If you're new, this is the other backup, for the backup, right? And so we'll always have a place where we can go get it in. So, all right, let's get this popping. All right, 1 second here. All right. All right. So share screen. Let's see here. Let's go. All right. All right. All right. Hold up. Let's see. Bring up the other one here. Oh, did I close that out? Oh, I didn't. Okay. So make sure I didn't. I hope I didn't close this out. Oh, no wonder I got to bring it over here on this channel. All right. All right. Yeah. So there we go. I think we're good to go. Yeah. There we go. All right. Let's get it, guys. All right. Hold up. Make sure my volume is good here. All right. Speaker.
No relevant financial disclosures to share. I speak on behalf of our National Council that was formed 10 years ago to provide an advocacy voice for physicians who are actively committed to the healthcare needs and public health needs of vulnerable Asian, Pacific Islander, and Native Hawaiian communities. Technical assistance, could you please go to the next slide, which shows the logo of the National Council of Asian Pacific Islander Physicians. In this context, the impact of COVID-19 on the AA-NHPI community has been underreported. Its impact on our communities mirrors that of other communities of color. And can you go to the next slide, which talks about the underreported story of COVID-19 burden on Asian Americans. For example, according to a recent report from the Kaiser Family Foundation, derived from Epic electronic health records from 52 million patients across 32 states, Asian Americans were less likely to get tested for COVID, more likely to have a positive test result, and require a higher level of care at diagnosis. Moreover, they were more likely to be hospitalized and die compared to all other racial and ethnic groups, according to the Epic data that I referenced. Against this sobering backdrop, Incapit greets the news of the Moderna vaccine with cautious optimism. Our communities need the protection offered by the promise of this vaccine. Its provision to the AA-NHPI community must incorporate critical components that are both relevant and unique to our population. I'd like to go to the next slide, which starts with the title, Critical Issues in Deployment, Vaccine Deployment for our community. Therefore, our organization recommends, number one, disaggregating data for the broad category of the AA-NHPI and efficacy and potential adverse vaccine effects in recognition that this category is comprised of dozens of subgroups and important differences can be lost when data is not broken down. And I have the next slide, which has the enumeration of Asian Americans. As the vaccine is deployed, the immigration status of Asian American individuals should not be a barrier for access. Although the vaccine itself may be free of charge to U.S. residents, the special status of individuals from the specific jurisdictions, such as Micronesia, should be accounted for. Can I go to the next slide? As the Moderna vaccine is deployed, every effort should be made to provide information about its background in a culturally competent and linguistically accessible manner. Since many AA-NHPI individuals travel to and from Asian countries and also obtain information about COVID-19 from sources other than those that originate in the mainstream and/or American press, efforts should be made so that there is no confusion or misinformation about an individual's vaccine status. Number four, physicians and other clinicians from Asian American, Native Hawaiian, Pacific Islander communities, like those at community health centers, should be supported as critical ambassadors that advocate for the Moderna and other COVID-19 vaccines. Thank you for allowing me this opportunity to comment on the important issues relative to the Asian American, Native Hawaiian community as we look forward to the approval of the Moderna vaccine.
Time. Goodness.
Thank you. The next speaker is Ms. Lisa Butler. The Butler Executive Director. Hello, my name is Lisa Butler, Executive Director of the GBS CIDP Foundation. At this time, I have no financial interests or conflicts of interest to disclose. Thank you to the FDA for this opportunity. Guillain-Barré syndrome is an acute inflammatory disorder of the peripheral nerves. GBS is characterized by the rapid onset of numbness, weakness, and often paralysis of the legs, arms, breathing muscles, and feet. This paralysis is ascending. The cause is unknown. We do know that about 50% of cases occur shortly after a microbial infection, viral or bacterial, some as simple and common as the flu or food poisoning. Many theories suggest an autoimmune trigger. The COVID-19 pandemic sparked a flurry of anxiety for healthcare professionals and former GBS patients. Our community waited eagerly for the news of an increase of GBS cases being triggered by COVID-19 infection. Fortunately, despite a handful of GBS cases happening around the time of COVID-19 infection, there has not been any indication of an increased risk of GBS from a COVID-19 infection. A recent study out of the UK published in the Brain Journal of Neurology this week confirmed that there is no epidemiological association between the COVID-19 and GBS in the UK. The resulting commentary from that published article highlighted the opinion of leading peripheral nerve experts that there should not be any increased risk of GBS from the COVID vaccine. Please see the chart on the slide. In 1976, there was an apparent association between the influenza vaccine and GBS. However, since then, several studies have researched the risk of GBS after influenza vaccinations and have found no or a very small increase in the risk of someone contracting GBS after influenza vaccine. And this finding was recently highlighted by an article from CBER, CMS, and Immunization Safety Office of the CDC. Additionally, leading peripheral nerve experts remain confident that any GBS cases resulting from mass COVID-19 vaccination of the global community are coincidental and likely in line with the expected rates of GBS. Regardless of the science, though, the GBS community expresses understandable skepticism towards vaccination. A safe and effective vaccine against COVID-19 serves as a beacon of hope for many Americans. But the Guillain-Barré syndrome community feels a renewed sense of worry and panic at the news of this expedited scientific miracle. Though the data is still quite limited, the Foundation's Global Medical Advisory Board and the Peripheral Nerve Society are hopeful that the relative risk of GBS after a COVID-19 infection is not significant and that there is no reason to suspect that the vaccine would cause it. The Foundation urgently hopes for a partnership with the FDA to collaborate and truthfully instill necessary and earned trust in the GBS community regarding vaccinations, especially the COVID-19 vaccination. We will continue to rely on experts who serve the Global Medical Advisory Board of the Foundation for their assessment of science and safety. So in conclusion, we are a very nervous patient community, yet we are very optimistic for the future. Thank you for your interest.
Thank you, Ms. Butler. Next speaker is Dr. Diana Zuckerman. Hi, I'm Dr. Diana Zuckerman, President of the National Center for Health Research. Next slide, please. We scrutinize the safety and effectiveness of medical products, and we don't accept funding from companies that make those products. My expertise is based on postdoctoral training in epidemiology as a former faculty member and researcher at Vassar, Yale, and Harvard, and a former fellow in bioethics at Penn. I've also worked at HHS and Congress. Next slide, please. I'll focus on three concerns. Number one, the 2-month median follow-up is too short, so Moderna's proposal to immediately unblind and offer to vaccinate the entire placebo group should be rejected. Number two, Moderna recruited a diverse group of participants, but only 4 COVID cases were Black, and even fewer were in other racial groups. We can't assume that the vaccine was highly effective in demographic groups with so few cases. And there were 25 cases among participants with comorbidities, which is slightly more substantial. Number three, I'm glad to see that unlike Pfizer, Moderna provided a total number of participants who reported 1 or more adverse events. That's important. Unfortunately, the total of severe systemic adverse events after the second dose was over 17% for the vaccine group compared to 2% for placebo. Next slide. There were 30 severe COVID cases after the second dose, none in the vaccine group. This is a strong finding. 9 required hospitalization, but 12 were based on the questionable criteria of at least slightly low blood oxygen saturation. Next slide. Long-term care patients were not included in the study. 1,300 people over 75 were in the study, but only 3 were cases. We want to save their lives, but with no data, it's not possible to provide useful, informed consent to nursing home patients. That puts a tremendous burden on those patients and their family members. Next slide. We need longer-term data on benefits and risks. The vaccine is clearly effective, but does it last 2 months or 4 months or a year? To learn that, the FDA needs to ensure the blinded RCT is continued. Last slide, please. In conclusion, FDA should initially target authorization to priority populations. If the EUA is given for all adults, celebrities and others who are well connected will cut in line. We've already
Oh my
seen
gosh,
that.
I'm just so like shaking.
Other people could apply for the vaccine under FDA's expanded access program. We need at least 1 year of blinded, randomized, controlled data. I agree with Dr. Goodman's proposal that FDA should delay access to vaccines by placebo group members unless they are in priority populations. Blinded crossover is better than not continuing a blinded controlled study if that's the only alternative. Thanks so much for the opportunity to speak today.
Thank you. Next speaker is Dr. Charles Lee.
Good morning. I am Dr. Charles Lee. Next slide, please. I represent the American College of Correctional Physicians, and I'm speaking on behalf of correctional workers and those incarcerated. There are no conflicts. Let's look at the numbers. There are 2 million people incarcerated in the United States and 500,000 workers working within correctional facilities. The infection rate amongst those incarcerated is 6 times that of the general population. 1,700 folks have died. Why so many? There's an inability of inmates to follow the CDC guidelines. Why? They cannot socially distance. They're unable to get proper hand sanitizers because of the alcohol content. They live in close dorm quarters or cells. There's an inability to get frequently tested. There's poor ventilation. Many of these facilities are 18th and 19th century, and they may not get masks. I realize that this varies from facility to facility. There are increased inmate vulnerability. An inmate has a physiologic and medical age of 20 years younger than that of the general population. Someone 50 incarcerated, his body equates to that of someone 65 on the outside. There are increased percent of minorities within correctional facilities. There's a significant increase of patients who have comorbidities, diabetes, asthma, cardiovascular disease. There are also increased vulnerabilities of workers. Out of necessity, they have close contact with inmates. They have extremely demanding working conditions. Unfortunately, too many officers may get sick, thereby unable to properly manage the facility, increasing the danger within a correctional facility. What are the consequences of this? Increased deaths, suicidality. There have been fears of patients that they may die from coronavirus committing suicide. As a result of this, there's increased community infections. 90% of inmates are released at some point in time. Workers go home daily. There's increased use of community resources, clinics, emergency rooms, hospitals. When patients who are incarcerated become sick, they are referred to the community resources. The Moderna vaccine has certain advantages that may be extremely applicable to correctional populations. As a result, the American College of Correctional Physicians recommends approval of the EUA for Moderna vaccine. Thank you very much.
Okay, thank you, Dr. Lee. The next speaker is Dr. Bisola Ozikutu.
And I have no. Thank you for this opportunity to speak. My name is Dr. Bisola Ozikutu, and I have no financial disclosures. I am an infectious disease specialist and a frontline provider based in Massachusetts, which has one of the highest death rates from COVID-19 in this country. I work at Brigham and Women's Hospital and Massachusetts General Hospitals, and I've been working directly with Black community members for the last few months to promote acceptance of the COVID-19 vaccine, as many of us have. Many of the community members that I've worked with have suffered personal losses secondary to COVID-19, so this is a particularly important issue to them. Next slide. In this process of working with the Black community, I have attended numerous town halls and had many meetings and discussions. And I think it's really important to emphasize that mistrust of government and of the pharmaceutical industry runs deep. And though the recent polls show that willingness and acceptance may be increasing, we still believe that it will delay, the mistrust will delay and even completely inhibit uptake of these vaccines. While it is highly unlikely that we will make our institutions more trustworthy over the course of the next 2 weeks as vaccines are rolled out, I and others believe that the same amount of effort and funding that was placed into the development of this and other successful vaccine candidates needs to be directed toward ensuring uptake and promoting vaccine confidence, specifically within Black, Latinx, and Indigenous communities who are most disproportionately affected. What do we need to do? First, we need better messaging, articulated by trusted messengers that will resonate with racially and ethnically diverse individuals. Second, we need more intensive community engagement. So I'm well aware of several initiatives that, quite frankly, recently just got started. What has been done thus far is nowhere near enough. Next slide. In terms of messages, first, we need complete transparency in lay language regarding potential side effects, and we need to be honest and emphasize that there are many unknowns and much work remains to be done. Secondly, our government institutions and industry need to consistently acknowledge that systemic inequity and structural racism have led to this deeply rooted mistrust. Thirdly, we need to reframe vaccination as a form of empowering our communities and fighting back against COVID-19 related inequity. And lastly, we need to explain this process that we're part of today, to our communities. People want to know who is looking out for them and their best interests and the interests of people who look like them, and who is really at the table. Next slide. And in regards to that table, quite frankly, communities of color have not been at the table throughout the entire vaccine development process. They were not engaged early enough, and that is a problem. Going forward, we must change that dynamic. People of color will begin to trust this process and the process of other vaccine development if they feel that they're truly part of it. Therefore, community engagement and community investment must be enhanced, amplified, and fully supported. I believe that this is necessary. We will continue to see racial and ethnic disparities, and we will not end this epidemic. I'll stop there. Thank you.
Thank you. The next speaker is Dr. David Berger.
David Berger, thank you. Hi, my name is David Berger. Thank you for the opportunity to address this committee again. I have no conflicts of interest. Slide 2. I'm a board-certified pediatrician and senior medical advisor for the Vaccine Considerations Project. Slide 3. From the available data, it appears the Moderna and Pfizer vaccines are quite effective in minimizing the incidence of serious COVID disease. This is an amazing scientific accomplishment that will hopefully aid in our defeat of the virus. Slide 4. Vaccine hesitancy is prevalent in the healthcare community and public at large. Full transparency can reduce this hesitancy. As more manufacturers apply for authorizations, I urge the FDA to provide timely information for review. Meaningful input is not possible when we are given only 2 days to review manufacturers' data before addressing the committee, or when data is released after deadlines pass for submission. Slide 5. Seniors are one of the first targeted populations to receive COVID vaccine. Yet only 860 subjects over 75 years old were included in the reported Pfizer data. Moderna's data mentioned subjects over 55 years old, but made no distinction of participants over 75 years old. Our team found minimal data on pregnant women or those with pre-existing allergic, hyperinflammatory, and autoimmune conditions. If this data is not available, it will be very difficult for individuals to weigh the risks and benefits, which is fundamental to making an informed decision. As with the Pfizer vaccine, Moderna's report revealed incidents of Bell's palsy. While the number of cases was a small fraction of participants, we should closely monitor this to see if the trend develops for this and other inflammatory conditions. Slide 6. Please provide long-term data and outcome for patients with or who may develop autoimmune and hyperinflammatory conditions. Significant symptoms may take longer than 2 months to become evident. Please provide quantitative standards for COVID IgG antibodies so people can determine if they have immunity and if their immunity is persistent. Slide 7. Our team has not discovered significant differences in efficacy or adverse events between the Pfizer and Moderna vaccine. We will continue analyzing and commenting on other manufacturers as they apply for emergency authorization. It will be helpful to have comparative data to guide the decision-making process between brands. Slide 8. The Vaccine Considerations Project is building a central repository of COVID vaccine health and safety concerns. Our national network of medical and graduate students are compiling and analyzing science, data, and evidence-based information to help address these concerns. We are inviting all interested students, professionals, and others to join this important effort by connecting with us at vaccineconsiderations.com. Slide 9. It is critical that rigorous safety mechanisms are maintained and we are given complete transparency with data. We should closely monitor and report on unique subpopulations, such as different minority and racial communities, the elderly, and those with allergies, autoimmune, and hyperinflammatory conditions. With such actions, the FDA and vaccine manufacturers have the opportunity to provide Americans the information they need to make the most informed decision possible for themselves and their loved ones. Thank you.
Thank you, Dr. Berger. The next speaker is Dr. Renu Dhanasekaran.
Thank you very much. Thank you very much for the opportunity to speak at this public hearing. My name is Renu Dhanasekaran. I'm a board-certified gastroenterologist and hepatologist at Stanford University, California. I'm here as a physician to advocate for vaccine access for my patients and also as a scientist conducting COVID-19 research. I have no conflicts of interest to disclose. Next slide. COVID-19 is a global public health crisis. It has led to more than 1.5 million deaths in the world, with more than 290,000 unfortunately occurring in the United States alone. Next slide. Patients with chronic medical conditions like cancer, heart disease, and obesity experience worse outcomes with COVID-19. As a physician taking care of some of the sickest patients with chronic liver diseases and immunocompromised patients with liver transplantation, I have personally seen the devastation COVID-19 has caused for our patients, both directly and indirectly. Hence, clearly, the vaccine is a welcome relief for our elderly patients and those with chronic medical conditions. Next slide. As discussed by the earlier speakers, the Moderna vaccine has been shown to be effective in preventing COVID-19. When I looked at the data, I was happy to see that among the 30,000 participants in the phase 3 core study, around 7,000 were older than 65 years, around 5,000 patients who were younger than 65 years had underlying medical disorders like diabetes, obesity, and cardiac disease. Overall, around 42% of the cohort consisted of medically high-risk groups. This is reassuring to me. These are the very patients who are in dire need for this vaccine. Next slide. Moving on, I would like to acknowledge a sad reality that communities of color have been disproportionately affected during COVID-19. The CDC reports that American Indians, Blacks, and Hispanics are at more than 2.5 times the risk for death with COVID-19 than white Americans. Several investigators, including us, have shown that socioeconomic factors and medical comorbidities play a huge role in this. Next slide. I'm happy to see that the core study cohort overall included 11,000 people from communities of color, with more than 6,000 Hispanic and more than 3,000 Black. I believe these vulnerable communities will benefit greatly with the Moderna vaccine approval. Next slide. I have reviewed the safety profile of the Moderna vaccine. The vaccine was generally well tolerated, as can be seen from the grade 3 events listed here. In my opinion, the benefits far outweigh the risks with the vaccine, especially in patients with comorbidities. Next slide. I would like to end with these two take-home points. Number one, a safe and effective vaccine is the need of the hour. Number two, vulnerable populations will be especially well served with vaccine approval. Next slide. Thank you very much for the opportunity to speak.
Okay, thank you, Dr. Dhanasekaran. The next speaker is Dr. Marie Gerlach.
Warm greetings. I'm Dr. Marie Garlock. I'm a board member of the USA Patient Network. We're a grassroots patient advocacy group, and we're not funded by or beholden to industry in any way. We're completely independent of hundreds of members across the nation. Like me, we're patients, caregivers, and leading health justice advocates for driving device safety, efficacy, and affordability. Our letter submitted to the federal docket today has references for all four of our main concerns and action items. And I'd like to say, before we move to the next slide, titled EUA is a stopgap, not a stand-in, Given recent project on government oversight reporting, I want to start with a note. Unlike at last week's EUA hearing, today's AdCom deliberation must take time to transparently include all expert members' questions, voting amendments, and vote explanations. Today is not about PR. It's to take public health seriously, a commitment on which the FDA leadership must make good. So the next slide, titled EUA is a stopgap, not a stand-in. Clinical trials must continue. Here's the basic part of it. Do we want to control COVID-19? Then we have to keep the control groups going. Anything less skirts accountability for industry and FDA. We need public trust in COVID-19 vaccines that will only come from transparent public knowledge about how they work long-term, when, and for who. What does that mean? Placebo groups must continue alongside phase 4 trials. We need metrics that matter. Does the vaccine prevent transmission? Does it mitigate severity of disease that results in hospitalizations and deaths? Next, we need to incorporate the National Vaccine Injury Compensation Program. Folks can go to hrsa.gov/vaccine compensation. And then we need for health-focused media, elected officials, FDA, and Moderna and its peer industries to know that EUAs are not standard FDA approvals and authorizations. We need transparency on that. And an EUA should not ever be precedent for future similar or unrelated drugs and devices to be rushed through on loopholes. And next slide. We need transparency on diversity. So this means for age and comorbidities, because this population is so vulnerable, how many are at or near 75 years old? How many are frail elderly, i.e. older and with comorbidities? On sex and reproductive health status, we need to understand that females should know they should not get pregnant for a specified time after getting the vaccine, given lack of data on both developing fetuses and pregnant parents. And most of all, we need to understand for racial and ethnic difference. Given systemic racism as the root of COVID-19 health disparities, we need precise numbers for Black, Indigenous, Pacific Islander, Latino, and Hispanic folks. And in order, those folks, in comparison to their white counterparts, Indigenous, Black, Pacific Islander, Latino, and Hispanic people are 3 times as likely to die from COVID-19 and 4 times as likely to be hospitalized with severe COVID-19. In a framework called structural competency, we know systemic racism influences these upstream inequities in employment, housing, transportation, parallel health challenges, and healthcare insurance coverage, and that is directly reflected in COVID-19 severity, hospitalizations, and deaths. So we need nuance on the numbers, and we need retainment of these specific groups in placebo groups for phase 4. Most of all, FDA needs clinical trial diversity standards that have a systemic fix. We commend Moderna for slowing its trial recruitment, but it should not be only optional for companies. And our next slide. Okay, thank you so much. And I would like to ask the FDA to focus on nuance on the numbers, keeping the control groups going, knowing that integrity requires adverse event reporting infrastructure and that action means action. The FDA must ensure safe manufacturing practices. Thank you.
Okay, thank you. The next speaker is Mr. Gwen Shell.
Ms. Gwen, do you have your personal phone muted?
Sorry about that. My name is Gwen Shell. I represent a community of rural population. I'm a nurse and I work for a public health district. I want to describe the impact that COVID-19 has had on the rural population and touch on the value of a vaccine. We have very limited nursing staff in this part of the United States, and in a rural population, that nursing staff is covering an area of about 500 miles. We have noticed an uptick in people being sent home from the hospital who are not meant to be home. All of the local assisted living and skilled nursing facilities are very particular about who they take. A vaccine would not only benefit those who are at risk for contracting COVID-19, but would also benefit the health population at large. I wish to express our excitement and gratitude for treatments that are coming. And I forgot to mention I don't have any financial ties. But I just wanted to bring to light the impacts that a vaccine will have on rural populations. Thank you.
Thank you, Michelle. Next speaker is Dr. Douglas Dietrich.
Thank you. I'm Dr. Douglas Dietrich. I'm the director of the Institute for Liver Medicine at Mount Sinai Health System and a professor of medicine at the Icahn School of Medicine at Mount Sinai. I'm here as a patient, actually, not as a professor. Even though COVID-19 causes significant liver disease and significant mortality in patients with pre-existing liver disease. I think it's important to recognize that there is a space between life and death. You see the deaths, which are extraordinary, 3,600 yesterday, and the number of people infected. I was infected in mid-March, as was about 2/3 of my clinical team. I was hospitalized for about a month, sent home on 6 liters of oxygen. Subsequently, I discovered that I had severe peripheral neuropathy in my feet and severe fibrosis, pulmonary fibrosis, which I'm still getting treated for, actually both of them. And of course, my sense of smell is completely gone. So I think it's important to recognize that as good as our treatment is now, prevention is clearly much better. There's a lot of long-term effects of COVID. After I was at home for a few months, I developed some severe atrial arrhythmias. When they subsided, I developed severe hypertension, which I'm still battling. And of course, I'm still taking medicine so that I could feel my feet and hopefully recover some of my sense of smell. So I think the important thing is that that there's a real price to be paid for getting COVID, whether it's severe or not. There are long-term side effects, and I think that the vaccine is the answer to prevent COVID-19 and not to get it and get treated as good as treatment is nowadays. In addition, actually, even though my antibody levels remain extremely high, I will get vaccinated when my time comes. I think that's an important thing to recognize as well. So I wanted to thank the Moderna people and the other vaccine makers for helping us prevent this disease so other people don't suffer like I have. Thank you for the opportunity to speak.
Thank you. The next speaker is Dr. Jasmine Marcelin.
Yes, thank you very much. My name is Dr. Jasmine Marcelin, and I'm an infectious diseases physician in Nebraska. I'm employed by the University of Nebraska Medical Center, but my comments do not represent my employer, and I have no conflicts or disclosures to report. After reviewing the available information about the mRNA vaccine developed by Moderna, I'm encouraged by the 94% effectiveness demonstrated and review of expected adverse effects. I would advocate for, continued long-term monitoring of clinical trial participants to evaluate for the long-term effectiveness and safety. However, I am encouraged for this vaccine to receive EUA status with prioritization of those at highest risk. We still do need data regarding pregnant people and children. I'm encouraged by the sharing of outcomes with people who become pregnant during the trial period. I noted that there were 36% of participants in the trial from communities of color and few reported cases from these participants. Considering how and what we know about the disproportionate rates of COVID-19 in Black and Brown communities, I urge vaccine discussions to avoid centering mistrust of the Black and Brown communities as originating within those communities and instead
acknowledge the fact that the healthcare profession has previously betrayed these communities through centuries of structural racism, including grievances that are happening today. So therefore, we need to have open listening and understanding of the concerns of these communities, and trusted healthcare professionals from communities of color need to be engaged to ensure that the approach continues through a lens of equity and cultural congruence. I will also comment on the importance of funding campaigns with appropriate messaging and community engagement in the rollout to emphasize safety and efficacy for lay people to encourage vaccine confidence, and appropriate messaging about expected side effects so as to not alarm people when they occur. And then finally, hoping for an equitable distribution plan that ensures that people in rural, low-income, and communities of color have adequate access to the vaccine, including follow-up for second injections. Thank you for the opportunity to participate in this open comment, and I'm looking forward to seeing what the vaccine has to do for the community in the future. Thank you.
Thank you. The next speaker is Dr. Robert Wong.
Hi, good afternoon. I have no conflicts or disclosures. Dear committee members, thank you for giving me an opportunity to speak today and share my thoughts on the importance of timely and equitable implementation of this COVID-19 vaccine. My name is Robert Wong. I'm a clinical associate professor of medicine at Stanford and a practicing gastroenterologist and hepatologist serving our U.S. veterans at the VA Palo Alto healthcare system in Northern California. In addition to my clinical practice, which focuses on management of patients with complex liver diseases, my clinical research is focused on healthcare disparities, particularly among ethnic minorities, vulnerable populations, and underserved safety net health systems. Even prior to the COVID pandemic, ethnic minorities and vulnerable populations suffer significant healthcare disparities, from receiving timely screening and surveillance exams to delays in access to life-saving treatments. Specifically for patients that I serve, my research has demonstrated disparities in timely receipt of high-quality liver disease care, including access to viral hepatitis treatment for patients with chronic hepatitis B and hepatitis C, as well as timely screening for liver cancer among cirrhosis patients. In the past 9 months since the pandemic began in the U.S., we have seen these disparities exacerbated as our attempt to deliver high-quality care has been disrupted by this pandemic. Patients avoiding care due to fear of venturing out to medical visits for lab or imaging for cancer screening. Also, healthcare systems transitioning to telehealth, delaying non-urgent procedures, and trying to balance the risk of delaying diagnostic and treatment procedures with the risk of our vulnerable patients being exposed and infected with SARS-CoV-2. These vaccines that are now before us present some hope at the end of this deadly year, where many of us have lost not only patients, but close friends. While these vaccines will not be the magic bullet that miraculously reverses all the damage this pandemic has caused, it gives us hope that one day in the not too distant future, some semblance of normalcy will be within our reach. While I have no doubt in the eventual approval and the dissemination of these vaccines, I would like to encourage all of us to be particularly cognizant of ensuring equitable access, particularly among those underserved and vulnerable populations whose existing healthcare disparities have been disproportionately exacerbated by this pandemic. Thank you all very much for taking time to hear my comments.
Thank you, Dr. Wong. The next speaker is Dr. Joseph Fick.
Good morning. My name is Joseph Fick, and I'm an infectious diseases specialist serving as statewide director of healthcare services for the California Department of Corrections and Rehabilitation. I have no financial disclosures to report. I appreciate the opportunity to speak to the committee regarding the importance of including those who work and reside in our jails, prisons, and detention centers in the first phase of COVID vaccination. Over 2 million people are incarcerated in this country. Over 500,000 individuals interact with them on a daily basis as correctional officers, nurses, cooks, respiratory therapists, physicians, teachers, and others. More than 260,000 inmates and 58,000 correctional employees have been diagnosed with COVID, resulting in at least 85 employee and 1,700 inmate COVID-related deaths. The age-adjusted death rate due to COVID among the incarcerated is several-fold higher than what is seen in the outside community, and case rates among both inmates and employees are significantly greater than those seen outside of incarcerated settings. Many of the largest COVID outbreaks in this country have occurred in correctional facilities. Many facilities do not routinely test for COVID, and therefore these numbers underestimate the true burden of COVID in these settings. Most inmates are housed in large, overcrowded, congregate living environments in which consistent physical distancing is not possible. Many of these settings suffer from insufficient ventilation and hygiene, contributing to the likelihood of widespread COVID outbreaks. Inmates are disproportionately people of color and often have multiple comorbidities that increase their risk for serious illness, hospitalization, and death if they become infected with COVID. Delaying vaccine distribution to inmates will exacerbate the disparate racial impact of COVID-19. Advanced age is one of the greatest predictors of poor outcome with COVID, and age-associated risk for prisoners begins to rise in their 50s. The average age of inmates in this country has risen significantly over the years. Currently, over 10% of prisoners are 55 years of age or older. Many of our prisons are essentially nursing homes, long-term care facilities, and skilled nursing facilities with bars. Jails, prisons, and detention centers are often a major employer in some rural settings. When employees unknowingly introduce COVID, the disease can be rapidly amplified and subsequently fuel large outbreaks in the outside community. Inmates who require hospitalization can quickly overwhelm bed capacity in surrounding community healthcare facilities. Cases among staff and inmates are currently surging to unprecedented numbers, threatening to overwhelm local resources. Not including correctional staff and high-risk inmates in vaccination phase 1 will result in preventable illness and deaths, burdens upon local economies, unsafe jails and prisons, and increased pressure upon overstressed community hospitals. In closing, I urge you to include high-risk inmates and frontline correctional workers in phase 1A for this and all future COVID vaccines. Thank you.
Thank you, Dr. Dick. The next speaker is Dr. Donald Middleton.
Hi, I'm Don Middleton, a professor of family medicine at the University of Pittsburgh School of Medicine. I am unofficially speaking to support the EUA approval of the Moderna mRNA vaccine, which has shown its worth in rigorous blinded clinical trials. I do serve on a Moderna mRNA vaccine advisory board. My background is in vaccine education, and I am one of the developers of a free vaccine app for iPhones and Androids called Shots by AAFP/STFM. COVID-19 is ubiquitous. It's in the air, on doorknobs, on computers, in the trash. Even when social separation policies are followed to the fullest, infection still occurs. The number of infected persons is staggering. The number of deaths more so. In the U.S., 300,000, a number that is difficult to grasp. Basically, the city of Pittsburgh wiped out. As we have already heard, recovery from COVID often takes months or is incomplete. Most days when I walk into UPMC St. Margaret, my true home, a community hospital with about 200 beds, I wonder, how many COVID patients do we have? Is this the day, is this the one when I will become infected? Others who work here share that fear, but it does not stop thousands of our hospital employees from doing their jobs. Our hospital staff always keeps in the forefront that the patient is a person, something the statistics fail to convey. Before November, we used to have a few, maybe 5 or 7 COVID inpatients daily. Now we have 60 out of 190 inpatients. One day this week, 9 out of the 10 patients in the ICU had COVID and 7 were on respirators. A 70-year-old woman on a respirator had to communicate with handwritten messages. Just before being sedated to improve her oxygenation, she scribbled a note to the outstanding resident doctor taking care of her. Quote, I love y'all. My life is in y'all's hands, end quote. A heart with an arrow through it was attached to the bottom of this note. Endless lights, noise, strangers in the rooms, not loved ones. Everyone is gowned and masked. You cannot really sit to talk with patients or hear their fears. Even though the staff does their duty daily, they are working in hell. Control of COVID requires vaccine, billions of doses. The Moderna vaccine offers real hope that this pandemic can be truncated, and with published evidence of lasting immunity, help to keep it permanently at bay. Please advise the FDA to give this outstanding vaccine full EUA status. Thank you very much.
Thank you. The next speaker is Mr. Sidney Wolf.
Good morning. I'm Dr. Sidney Wolf, Public Citizens Health Research Group. I have no conflicts of interest. During the October 22nd meeting of this committee, before seeing data from either Pfizer or Moderna vaccines, FDA's Dr. Doran Fink pointed out that, quote, deployment of a weekly effective COVID-19 vaccine could result in more harm than good. It could do so by providing a false sense of security that interferes with measures to reduce SARS-CoV transmission, such as wearing of masks, other PPE, and social distancing. I would argue that even with current evidence that both vaccines are highly efficacious, there is still understandable concern about the danger of a false sense of security if those getting vaccinated no longer adhere to proven preventive public health measures such as wearing masks and appropriate social distancing. The FDA's 2017 EUA guidance included a requirement for an FDA-approved patient fact sheet to accompany the use of all EUA products, quote, to ensure that recipients are informed about the product they receive and to inform them of any available alternatives to the product and of the risks and benefits of available alternatives. Since 2017, no EUA for a vaccine had been granted prior to the Pfizer vaccine, but providing written information about proven health measures such as wearing masks and appropriate social distancing is clearly necessary and appropriate for COVID vaccine recipients. Flashing back to last week, less than 24 hours after the EUA for the Pfizer vaccine was granted, the FDA posted a Pfizer fact sheet for recipients and caregivers intended for recipients of their vaccine. The fact sheet accurately states the Pfizer BioNTech vaccine may not protect everyone. Unfortunately, it contains no mention of the need for wearing masks and appropriate social distancing. For further information, the fact sheet suggested asking the vaccination provider or your local or state government health department, and then list websites that do not state such preventive measures should accompany vaccination. Though necessary as a part of companies' EUA submissions, such fact sheets were not included in briefing packages provided to the public or possibly the advisory committee for either today's or last week's advisory committee meeting. But this morning, Dr. Doran Fink mentioned that FDA's review FDA mentioned that the review and revision of fact sheets for vaccine recipients were part of what happened when FDA looked at the EUA submission. So this is at least mentioned in today's meeting, which it hadn't been before. I hope your advisory committee urges that important public information such as that must immediately be added to vaccine fact sheets before millions more people are vaccinated. Thank you very much, and I hope you will ask the FDA questions about this. It does not seem to be in their presentation for this afternoon. Thanks again.
Thank you, Dr. Wolf. Next speaker is Dr. Rebecca Luskin-Hawk.
Thank you. I'd like to thank you for the opportunity to comment on today's deliberation. My name is Dr. Roberta Luskin-Hawk, and while I'm employed by Providence St. Joseph Health, I'm speaking as a private citizen today, and I have no relevant financial disclosures. I'm an infectious disease physician with extensive experience in conducting and analyzing clinical trials, in addition to experience in overseeing healthcare delivery across both urban and rural settings. My current role as hospital chief executive serving remote area in Northern California provides a unique perspective on the potential impact of emergency use authorization of mRNA 1273 COVID vaccine on rural communities. The current surge in COVID-19 is having a devastating impact in communities across the country, and the demand for care is starting to exceed capacity in parts of the U.S. healthcare system, with further increase in cases forecast in coming weeks. While the numbers of patients with COVID-19 in rural communities may seem limited, even small numbers of cases or illnesses among healthcare workforce can threaten the fragile healthcare infrastructure and limit the ability to provide critical care to people in these communities. Swift intervention is needed, and we are fortunate to have had a robust response from the scientific community. It is therefore essential that we rapidly deploy vaccines that are found to be safe and effective against SARS-CoV-2 to both rural and urban communities across our country. The data provided on the Moderna mRNA 1273 COVID vaccine demonstrates exceptional vaccine effectiveness in the reduction of symptomatic COVID-19 across all ages, in addition to beneficial impact on the severity of disease. The vaccine also seems to have a favorable side effect profile in early evaluation. Use of the vaccine with this efficacy will not only save lives that could be lost to COVID, but will help leave ICU capacity available for the care of patients with other acute medical conditions. The fact that storage requirements can be met by healthcare organizations without access to ultra-low temperature freezers will have an added benefit to many small rural hospitals and clinics. Vaccination of 21 million U.S. healthcare workers and vulnerable populations is urgently needed to protect our healthcare workers, our healthcare infrastructure, and to change the tide of the pandemic. Rapid and broad distribution of vaccine will require EUA and eventual approval of more than one safe and effective SARS-CoV-2 vaccine. I urge you to provide emergency use authorization for mRNA 1273, which has met the necessary safety and efficacy benchmarks in the analysis of the clinical trial data. I personally believe that this approval is needed to support our healthcare workers and to save lives. Thank you.
Okay, the next speaker is Ms. Veronica Holloway.
Good afternoon, and thank you for the opportunity to speak today. My name is Veronica Holloway, Chief of the Center for Minority Health Services at the Illinois Department of Public Health. I have no conflict of interest. I want to recognize Dr. Damon Arnold, who has been leading community conversations in education about COVID-19 vaccine on behalf of Illinois' COVID-19 Equity Task Force. To ensure that disparately impacted urban and rural communities of color are informed and engaged in the process of building trust, raising awareness, promoting the importance of vaccination, and creating equitable access and distribution, we launched several initiatives. We engaged with a diverse group of community partners, including faith-based, people with disabilities, the homeless, refugee and immigrants, returning citizens, seniors, and the LGBT communities, to discern a need for special assistance. We launched a community ambassadors program to ensure confidence with directed messaging surrounding COVID-19 vaccinations. These conversations made clear that education and targeted communications regarding misinformation and myths, rebuilding trust, vaccine science, and active collaboration with communities are key. Accurate and timely information concerning the safety and efficacy of the vaccines from the manufacturers and scientific community is vital. National and state data shows that COVID-19 kills more males than females, and Black males already have a life expectancy 8 to 11 years shorter than their white counterparts. Special outreach efforts should be made to engage Black males in order to improve participation in both outreach and vaccine uptake. Messaging must be consistent with community beliefs and perceptions about the vaccine. We convened Zoom meetings to collect perspectives from communities mentioned. We noted that both cultural and linguistically appropriate language is essential for effective communication and delivery of quality healthcare. Providers appear to require additional training with respect to cultural norms and implicit bias. Providers must be intentional about fully engaging with local gatekeepers and community members about the vaccine. The current COVID-19 pandemic also underscores the need for a more diverse healthcare workforce reflective of the communities they serve. In closing, There is concern that the access and distribution of vaccines will encounter hurdles within already negatively impacted rural and urban communities of color. Federal, state, and local support is needed, such as additional funding to support the use of tools like COVID-19 Community Vulnerability Index, which combines the CDC's Social Vulnerability Index with epidemiological and health system factors to target areas most likely to be impacted. Thank you for your time and attention to this important matter.
Thank you. The next speaker and the last speaker of the session is Dr. James Woody.
Hello, I'm Dr. James Woody, and I'd like to thank the FDA for the opportunity to speak. I have no financial disclosures. I'm a pediatric immunologist and a biotech executive who, in a prior life, discovered and developed a drug called Remicade. I'd be interested in how patients on anti-TNF inhibitors do with your vaccine, but that's not why I'm here. I'll talk about what I see as the optimal format for deploying a COVID vaccine for the Navy and the Marine Corps. My comments are my own, do not reflect in any way the opinion of the Navy or Marine Corps. So I'm a retired U.S. Navy captain who spent 20 years in the U.S. Navy as a medical officer. I ran worldwide Navy medical R&D. One of our jobs was to be aware of any infectious disease risk anywhere in the world where a Navy ship might port or personnel go into conflict. By way of experience as a former commanding officer of the Navy's medical unit, Namru 3, a BL-3 force facility in Cairo, Egypt for 4 years, my team of about 50 Navy people did surveys for infectious disease over the entire Eastern Africa and Mideast region. And they included HIV, hepatitis, Ebola, Congo Crimean, Rift Valley fever, Lassa, and serious stuff. So as you know well, space on Navy ships is very confined and berthing space is always limited. So transmission of infectious diseases is a concern. We have actually shut down ships in the past due to chickenpox outbreaks. As you have seen in the press, over 190 Navy ships have had COVID cases, representing about 65% of all Navy ships at sea. Likewise, the Marine Corps recruits who live in congested facilities have also had significant numbers of COVID cases. So should the Marines be required to deploy on ships, which is the usual sequence, the overcrowding will be even worse and they'll even be at higher risk. So assuming a 2-dose schedule will work to provide protective immunity, so what's the best format for use by the Navy and Marine Corps? Common sense needs to prevail here. Simple is better. Available storage, no diluting. So in situations where multi-doses are required, the smaller shore-based clinic facilities and the shipboard facilities must have similar kinds of storage equipment and capacity so that once a seaman or Marine is deployed with a first dose, they can actually get a second dose that can be administered anywhere on shore or in the fleet. So most shore-based facilities have the usual minus 20-degree home-type refrigerator freezer. So vaccines could be stored in any of these locations and the second dose be administered quite easily. Use of the much lower temperature specialized freezing at minus 70 or 100 is not a reasonable option, as such kinds of equipment is only available on very few very large ships or in shore-based hospitals. So in summary, from someone who's actually been in the trenches, common sense needs to prevail here. Simpler is better. Thank you very much for the opportunity and listening to my talk.
Thank you, Dr. Woody. I would like to thank all the OPH speakers at this point for making their comments. This concludes the open public hearing session. And then now I would like to introduce Dr. Peter Marks. He wanted to state his thanks as well. So Dr. Marks, are you ready?
Thanks very much. So thank you very much to our public speakers.
I just want to take a moment before we move on to the further questions and then the FDA presentation and then deliberations later on. There wasn't an exact perfect time to thank everyone today, but this may be a reasonable one just to thank everyone for their participation. This is somewhat of an historic event to have these two advisory committee meetings so close together, and we really thank all of the advisors for taking the time to go through a very large amount of material. Also need to thank our FDA staff who have worked tirelessly going through an amazingly large amount of material over the past week. And that was only made possible because they had worked for several months with the companies internally and with stakeholders to prepare things so that this relatively rapid EUA review would be possible. So incredible thanks to our FDA colleagues and thanks for all who are tuning into this process. I also need to call out the advisory committee staff, which has done a remarkably great job in putting together this meeting. So I won't hold this up anymore, and I'll turn this back to Dr. Monto.
Thank you, Dr. Monto. Dr. Monto, the floor is yours.
Thank you very much. We're going back to questions directed to the sponsor. And I see Dr. Miller's ready, and I'll readdress the question I interrupted when we broke, and that was about escape mutants and what you're going to do about them, sequencing and the rest.
Jacqueline, you have your own phone muted.
Thank you for the reminder that I was still muted. Apologies for that. So thank you, Dr. Monto. Yes, indeed, the question actually was about whether we were intending to sequence the samples we receive from breakthrough cases, and the answer is yes. We are in the process of deep sequencing virus from those cases, and I was going to invite Dr. Darren Edwards, who is the head of our preclinical group, to address the work that we have been doing to assess the effectiveness and immunogenicity of the vaccine against emergent mutants. Dr. Edwards?
Thank you for that. Thank you, Dr. Miller. In addition to deep sequencing of cases in our phase 3 trial, we're also performing additional assessments, research assessments. These include the evaluation of vaccinated either animal or human sera, the ability of that sera to neutralize these breakthrough or these variant strains. We're also additionally monitoring for additional strain variants, both through our own internal efforts as well as through collaborations with external research partners. We have thus far identified 5 strain variants that are of key concern, and we have at this point assessed both mouse and non-human primate sera that were vaccinated with mRNA-1273 to protect against these strain variants, and we see they equally protect. In the future, we are also performing assessments on human sera. Thank you, and I hope that addresses your question.
Thank you. Let's go on to Dr. Sawyer. I believe you have a question.
Thank you, and thanks for the great presentations. Given our new and unexpected focus on anaphylaxis, I just wanted to ask if you've seen anaphylaxis in any of the other, I believe you said 8 vaccines that you had previously developed and given to a quite small number of people. Whether you've seen allergic hypersensitivity reactions in any of your animal models, and whether you have done or are planning to do any in vitro studies to see if this mRNA lipid platform creates interactions that would predict allergic type reactions.
Yes, Dr. Sawyer, thanks for that question. And indeed, we have been doing a very rapid review of our overall clinical database in light of the information that has come forward about the other mRNA vaccines. So as you mentioned, we do have a clinical database across 8 other vaccines. It includes approximately 1,700 recipients of a similar lipid nanoparticle with specific mRNA sequences. In those cases, we have had one other report of anaphylaxis. It was a woman with soy allergy and more than 2 months outside of her vaccination. And I should clarify that although participants have been excluded on the basis of a known allergy to one of the components of the vaccine, we have not routinely excluded participants who have a history of allergies or anything. And then your second question was about potential in vitro studies. In fact, Dr. Zaks has been in discussion actually with thought leaders at the NIH, BARDA, and so forth, talk about what additional activities we might collaborate to better understand what this potential.
Thank you. Dr. Lee.
Yes, I had a question about the unblinding. A number of people indicated that there is a clinical trial supply that could be used for that purpose, and that would not interfere with any supplies that would be given, say, to the general public if the EUA were to be granted. So my question is, what the indication was that it had a limited shelf life, and I think my first question related to that is, how long do you think that supply will last? And related to that is, would you have enough doses to vaccinate in two doses for all 15,000 placebo participants? Were they all asked to do that?
Yes, so thanks for your question about the vaccine supply. And yes, it is true that we have sufficient supplies to be able to vaccinate our placebo participants. The supply actually will be expiring relatively soon. So by the end of the next month, the supplies will be expired, so they cannot be used for emergency use.
Okay, thank you.
Dr. Cohen.
Hi, Dr. Miller, thank you. I was wondering if you could give us a little bit more information about in the 3 or 4 cases of bug palsy, including how many days after vaccination symptoms started to occur and how long symptoms recurred, and if those persons recovered. And if there's a history of bug palsy.
No, thanks for that question, Dr. Cohn. So the cases occurred between 17 and 32 days after vaccination. They were either resolved or resolving at the time of this presentation. And they were, 3 were non-serious, 1 was a serious adverse event.
Dr. Carrillo.
Thank you. Dr. Miller, in terms of your efficacy evaluation, you began counting 2 weeks after the second vaccine dose, but your Kaplan-Meier curves between vaccine and placebo begin to diverge after about 2 weeks after the first dose. But your immunogenicity results in your phase 1 say that even by 2 weeks after the first dose, there's no neutralizing titers, and there doesn't seem to be any bump in T cells, which suggests that there's some kind of non-specific antigen vaccination-mediated protective effect potentially going on. And the question becomes, how long does that actually manifest? And do you know what that is? With reactogenicity, I would presume it's inflammation and interferon and NK cells and that sort of thing. I'm just wondering how much that may be bleeding into the primary efficacy endpoint analysis.
Yeah, thanks for that question. So we did show a difference in the reported cases in the Kaplan-Meier curve after randomization, as you mentioned. We do know that our vaccine induces innate immunity with the first dose, and the adaptive immunity clearly increases with the second dose. Understanding this phenomenon a bit further is why we looked into that one-dose efficacy in several different ways. So looking at it in terms of the time period when the mRNA 1273 cases might be reported, as well as looking at the PCR swabs and looking at the ability of the differences between the vaccine and placebo group in terms of that positivity. So I am going to also ask Dr. Melissa Moore if there's anything else, our Chief Scientific Officer, if there's anything else she'd like to add about patterns of immunity we observe with the platform after the first dose.
Thank you, Dr. Miller. I actually would like to send that question over to Dr. Tal-Zaks, who has more experience with the clinical trials.
Thank you both. Yeah, I think the salient part here is that we see binding antibodies come up very quickly. And while everybody focuses on neutralizing antibodies, and appropriately so, I think their sensitivity is lower than looking at the binding assays. And if you look at binding antibodies, they actually come up within a couple of weeks. And so I suspect what happens here is that as you get the first dose, you're primed, binding antibodies start to come up, and now you've got a race between, is your infection going to, in a sense, be a boost? Because we know this virus takes some time, and you're still protected against symptomatic disease. So I suspect that's the reason for the discrepancy we see between the neutralizing antibodies that are clearly measurable better after a boost, but the sense that protection may start as early as the first dose. And I think in that regard, our results are very concordant with that were reported here last week. So while there is some potentially innate activation, I think the story here really is the SARS-CoV-2 and the quick antibody binding and total response that you see after the first dose. Sure, with the boost you get maturation and further increase in that, and now you start to measure consistent neutralizing titers. I will say though that at the end of the day, for me, that first dose efficacy is really supportive evidence overall, but coming back to the fact that what we really studied was a prime boost, and what we see is clear boosting and a high level of protection across all age groups, and hopefully that will be durable. And so I would take the first dose efficacy as supportive evidence, but remind us all that we actually need both doses as far as we know to achieve this high level of protection. Thank you.
Dr. Sylvester.
Thank you, Dr. Monto. I wanted to briefly revisit that blinding versus unblinding issue. As the industry rep, you don't need to convince me that a randomized double-blinded clinical trial is our gold standard. However, I don't believe this would be the first study that would be the first RCT that would meet their primary endpoint and vaccinate the placebo group before the protocol-described timeframe ends. I believe that HPV-4 Gardasil and the original pneumococcal conjugate vaccine, Prevnar 7, vaccinated their placebo group after the primary endpoint was met, and the data showed overwhelming evidence of benefit similar to what we're seeing here today. I don't know Dr. Baden at Brigham and Women's, but I share his concern about losing a significant portion of his study population without offering the COVID vaccine, and I think his open label continuation seems like a practical solution. Thank you.
Dr. Meisner.
Thank you, Dr. Monto, and thank you, Dr. Miller and others for a fascinating presentation. I have a few questions related to the vaccine that are all related. First of all, why do you think you were successful with this particular messenger RNA vaccine, whereas the previous 8 are still development? Number one. Number two, when we see adverse reactions in the first 48 to 72 hours following administration of the vaccine, do you think that's a reaction to the messenger RNA or more likely to the lipid nanoparticle? And along that line, is there understanding that these are proprietary issues, but can you say anything about differences in the lipid nanoparticle between Moderna's vaccine and the one that we spoke about last week? And then finally, why did you select a 28-day time interval between the first and the second dose? Was there a reason for that? Thank you.
Jacqueline, did you mute your phone again?
I did, to not interfere with my colleagues. I apologize. So thank you, Dr. Meisner, for those questions. And what I was saying was I'm going to start, and then I'm going to pass the mic along to our Chief Medical Officer, Dr. Zach. So with respect to our development program for mRNA 1273 and the other development programs we have ongoing. So our company has been in the clinic now for about 5 years. Most of our programs actually have been in the clinic now for about 2 years. And the difference between the 1273 program and others, of course, is the unique circumstances in which we find ourselves and the strong medical need which the vaccine requires. So we have expedited many elements of the development program, including conducting the 3 phases of our study staggered, but also much of the conduct has been done in parallel. And that has required an absolute focus and collaboration across multiple groups with their focus as well on the scientific questions that have been raised throughout the course of development. So for example, what safety data did we need to have available in order to move from one step to the next step? With respect to your question about the component of the vaccine that is responsible for the reactogenicity, I'll ask Dr. Zach to join the call now.
Don, sir, you are still muted, sir.
I apologize for that. Look, for the vaccine, I think it's also important to note that we're in the midst of a pandemic, and it's the paradox of vaccine development. For a case-driven trial, cases are occurring, unfortunately, and that's why these trials delivered information so quickly. As it relates to the components, and this was a point of discussion yesterday with an expert panel convened by the NIH, where FDA also attended. I think if you look at the lipid nanoparticle and you ask yourself about the anaphylaxis, people look at 3 elements here. There's the PEG component, which is actually not just the PEG. PEG is connected to a lipid, and in that regard, not all PEGs are the same. And indeed, the PEG and the covalently attached lipid that's in our vaccine is different than the one in the Pfizer vaccine. The second potential culprit is the aminolipid, and that's where we and Pfizer use very different each proprietary aminolipid. So these are different components. The other components are probably innocuous. Cholesterol is enough in our body. The mRNA itself is unlikely to be the culprit here because it's all naturally in cells. The final element here is the physicochemical particle properties, right? Because we know that these particles can actually induce responses in of themselves due to their physical properties. And in that regard, I would expect that the physicochemical nature of our particle is actually going to be very different than Pfizer's. So while we all say, oh, there's an LFP here with some lipids and mRNA, therefore they must be the same, I actually think that as far as the components likely to be the culprits here, I would not necessarily assume that. Now, that being said, of course, we're going to be looking very carefully, as has been noted, and continue to collaborate with colleagues to try to understand the mechanism here and make sure that we understand this as the picture evolves. I think the last question you asked was about the 20-day interval.
I think this is basic immunology. I don't think there's a big difference between 3 weeks and 4 weeks. In the history of our vaccines that Dr. Miller alluded to, we've always done a 4-week interval between prime boost. That's sort of based on, immunological first principles of vaccination as we understand it, and when it starts to be optimal for priming. But I would note here that the window for the second vaccine actually in the protocol was reasonably wide. It was -3 + 7. So, we say 4 weeks, but there's some spiel there. And I think when we did our analysis, we made sure to include all that. So I doubt that is materially different when the dust settles. Thank you.
Thank you. And can I ask one follow-up question? So,
I'm in the unenviable position of having about 8 hands raised and 5 minutes to go. So we're going to have to put that off until later.
Understood.
Mr. Taubman.
Yes, thank you for the presentation. The data is impressive, but I'm still nervous about only 9 weeks median data. So to try to put myself at ease, there's a couple questions. One is with regard to the severe disease endpoint. The supposition is that it's advanced disease, severe disease, but we really need data. Pfizer did not really have data on that. They had very few cases, and they were given the opportunity to provide recent data. They declined. You have 30 cases in the placebo group and none in the vaccine group, which is great, but is there more recent data? I mean, I assume you know how many severe cases there have been since they closed on 11/21. So how many cases has that been, and has the split reflected the 30 in the zero support? My other question is related to the unblinding. This is really important because we don't have enough data, and maintaining the placebo-controlled studies is the way to get more data. And you're proposing, your plan is specifically to end that. And the arguments, we've heard a bunch of arguments for that, one of which is you don't want to disadvantage trial participants relative to others. And also there are supplies that have been set aside that you could use for all the trial participants. You just answered the question good enough. But I think that's kind of beside the point. The real question is, what is the expectation? And I'd like to ask, as I understand what Dr. Goodman was explaining, he indicated that the participants in your trial were not told that they would jump the line, that they'd be entitled to get the vaccine before others in their same demographic group, in their same risk group. And if that's true, they have no expectation of getting it different from anybody else in their group. Is there any other ethical reason why Moderna thinks that its trial participants who got placebo should be getting the vaccine compared to Pfizer, which Pfizer appears has rejected the blinded crossover study, but they have, according to this letter they just sent out to one of the trial participants in my state, it's only healthcare workers, 20% more healthcare workers who are being offered this, and the rest are being told will be at a later date. I'd like to know if there's an ethical reason, if you haven't told people they're going to all get it, why you're any different than Pfizer, and you couldn't do the same with the 25% who are healthcare workers in your trial, and then the rest will be later, and that way you maintain placebo-controlled studies for the remaining 75%.
Thank you for your questions, Mr. Taubman. Maybe I'll start with the first question about additional data. As Dr. Fink reviewed in his presentation, we've actually made 2 submissions to the FDA. So the first was on what was intended to be a first planned interim analysis. There were so many cases reported in November that actually we achieved our final primary analysis approximately 5 months earlier than we anticipated. We have continued to collect cases since that submission on December 7th. And we currently have over 450 cases that are actually making their way through the adjudication process. And you can imagine that our adjudication physicians also have been working extremely hard to keep up with this real tsunami of data that are becoming available. So I don't have that information available for you today. Do intend, though, to continue to make data cuts and update those efficacy analyses. So that should be in the weeks to come. Your second question was really about the ethical basis for the proposal
to unblind placebo recipients. And I think some of your questions really speak to the interface that Dr. Baden has with the trial participants. So I'm going to turn the floor over to him in a moment. I guess the one thing I would say is we do have one death that has been reported in our trial in a case of severe COVID that occurred in a placebo recipient. And that death weighs very heavily on me. But I do understand that that death occurred at a time when we did not understand if this vaccine was going to have the efficacy that it does, and we didn't have a clear understanding of what the benefit-risk profile looked like. I do think that with the 450 cases that I just mentioned, additional severe cases and deaths are a question more of when than a question of if. And I think the knowledge that that may be waiting in some of our trial participant future weighs heavily on me. But Dr. Baden, do you please also take the floor and discuss the question?
Yes, no, thank you, Dr. Tubman, for raising those issues. I think the question is not that they were promised. You know, we should not disadvantage the volunteers, but we have to be practical of where we are. Unblinding is going on, vaccine is available. The vaccine availability is going to rapidly extend to multiple groups. So it's not as if this will take place over 6 months to a year. This is going to take place over days to weeks in terms of the extending the vaccine supply to additional groups such as 1B and 1C. And I think what we need to do is to keep the volunteers in the study. And that keeping it in the study, there's not only one flavor of study. It's not just a double-blind study. There are other formats of the study that can enable us to learn, particularly to learn about asymptomatic transmission through the serology at the transition point, the nasal swab to look at contagiousness and infectivity, and that if we don't come up with a plan that is easily understood and practical for all of our volunteers, some of whom are very health savvy and some of whom are not, then it will become very confusing and disruptive and corrosive in my view. And so I don't think it's an issue of a double-blind study or nothing. There are different formats of an ongoing clinical research trial that leverages or accepts the reality that we are facing over the next 2 to 6 weeks in terms of the transition as vaccine becomes more available. Thank you.
Okay, we're going to have to go on to Dr. Fuller. And let me say in advance that we're going to eat into our lunch. I'm going to try to break for at least a short period of time because we have no breaks scheduled from now to the end of the meeting. So we will take a break for a short period of time, maybe for 15 minutes. But since I've got a lot of hands raised, I'm going to continue to go. Dr. Fuller, please.
Thank you, Dr. Monto, and thank you, Dr. Miller and Moderna, for your study and what seems to be a very carefully crafted and executed study. I have two hopefully quick questions. One, you mentioned that you will be doing surveillance in the follow-up in phase 4, not only through CDC and FDA, but your own system of real-time global monitoring of events. The first question is, will that be done in conjunction also with other vaccines that may be approved? For example, the one that's already been approved through Pfizer or EUA. And then the second question is probably a little bit more theoretical. You noted that you have greater pain or third-degree pain for the second injection than the first injection. And I've been wondering about these vaccines that, especially to the S protein, where they boost specific immunity. What happens when people are exposed over and over again to the virus in a circulating pandemic when they've been highly boosted to something that binds to, say, in this case, the ACE2 receptor? Do you have any idea why there's more pain in the second injection? And do you have any thoughts about this idea of having highly boosted immune systems in the middle of a pandemic where they're continuously challenged?
Yeah, so thanks for both of those questions. And I'm going to go to the second question first, so that afterwards I can turn the floor over to Dr. David Martin, who can then speak a bit to the pharmacovigilance plans we have both in conjunction with the safety surveillance systems at FDA and CDC, and then also the study we intend to undertake ourselves. But your question about the reactogenicity observed with the vaccine, and could that potentially have to do with vaccinating during a pandemic? So what we've observed in terms of the vaccine reactogenicity actually really parallels what we see in terms of the vaccine immunogenicity. So the increase after the second dose really goes along with the increase in neutralizing antibody that we see in all participants and the induction of our T-cell responses. We did actually have 2.2% of the population in the study who did not have a history of COVID-19 disease, but when we tested their baseline swab for RT-PCR, and we tested their serology for existing anti-nucleocapsid antibodies, were found to be baseline seropositive for SARS-CoV-2. And in fact, the observed reactogenicity in that group was lower for both local and general solicited symptoms. So we think the vaccine can be safely given to people that have previously been exposed to SARS-CoV-2, and think it's more likely that the increases in immunogenicity are really what are related to the pattern of reactogenicity. And so for the second question, Dr. Martin, would you like to talk about the post-authorization safety study that we are proposing?
Before you go to the second question, just a quick follow-up. So does that mean that when people who are immunized get re-exposed, say during, over the next 3 months to virus that's circulating, that the boosted immune system should not have any systemic effects because of just exposure to virus? I just don't know the answer to that, and I don't know if anyone does.
Yes, I think you're right that you bring up a good point. There's only 2.2% of the population were baseline seropositive in this study. So certainly that is another important reason both to keep the clinical trial ongoing and also and to follow the patients who might get vaccinated in a crossover design for their safety events, but also to conduct the post-marketing safety surveillance that we're proposing to do. And so perhaps Dr. Martin, do you want to talk to the study that we're going to conduct?
Briefly, please.
Excuse me?
Brief.
Oh, briefly. Okay, absolutely. Understood. Thank you, Dr. Fuller, for the question. As you were mentioning, visibility for the Pfizer vaccine as well as the Moderna vaccine. So there are vaccine-specific administration codes which are brand-specific, and so the U.S. FDA and CDC surveillance systems, which have described their activities publicly in ACIP meetings in the last few weeks, they will be able to observe both vaccines in a brand-specific manner and certainly aggregate if they choose to do so. Moderna, as is customary, will primarily focus its monitoring on its own product and will obviously work bilaterally if contacted by the FDA. We've been notified by the U.S. government that we should expect communications regarding safety signals from the U.S. FDA. And so that is customarily how these things are done.
Thank you. Okay, Dr. Hildreth, please.
Dr. Hildreth, we're not hearing you.
That's the problem.
Dr. Hildreth, just make sure you're not on mute. There we go. Now we can hear you.
Can you hear me now? Oh, thanks.
Yes.
Good. First, I'm apologizing that we're still not able to get my camera to work. I apologize for that. My question relates to the minorities you enrolled in the study. My understanding is that many of them, or large numbers of them, were enrolled late in the process. And I wonder if you have the same median follow-up for those individuals as you have for the
specific data about minorities and the follow-up in each of those groups. It is true that they were enrolled a bit later in the process, and that was really because we invested in working with community leaders to understand what we needed to do in order to make participation in clinical trials something that those communities would consider. Again, to Dr. Bateman's previous point, building trust with those communities and ensuring that they benefit from the clinical trial in which they have so generously donated their time and their willingness to be examined is really critical, I think, to encouraging in minority future clinical trials. And we will continue to follow, as we would propose to transition to an open label study, we will continue to follow those individuals for further breakthrough cases and for their safety outcomes to generate these very important data.
Thank you.
Dr. Perlman, please.
Yes, I just have a relatively quick question. So this vaccine can be kept at room temperature for some number of hours and at 4 degrees for a long time. And since it's an RNA vaccine, how much degradation of the RNA occurs during that time? I worry when it goes out to more distant places that conditions won't be so perfect. So how long is it really stable?
Yeah, so to speak to the stability studies, I am going to ask our CMC expert, Dr. Nadim Altaris, to take that question.
Hello, hi. Can you hear me? Yes, we have performed, we have started performing our stability study with the development of vaccine very early on in January when we started developing this vaccine. And we have collected significant amount of stability since that time, which we have shared with FDA, including our stability at 228 and room temperature, which basically we provided to FDA to be able to make the shelf life claim that we are making. And FDA, as you have noted in their briefing document, have agreed with our CMC package that's suitable for emergency use authorization.
But is there any degradation?
mRNAs have degradation over time at different temperature, and that we characterize that, and we assure that at the end of the shelf life, our product remains potent and maintains the quality attributes across all quality attributes to maintain effectiveness. And also during the phase 3 study, we actually put lots in the study representing the quality attributes across the shelf life of the product.
Okay,
thank you.
Final question before we break from Dr. Rubin, please.
Do you think? And how long does the mRNA stick around for? Inside the cells.
I apologize, Dr. Rubin. I missed the first part of your question. The audio took a moment to come up. Would you mind repeating it, please?
Okay. Sorry. So which cells do you think are important for antigen presentation? And how long does the mRNA last intracellularly?
So to answer your question, I'm going to ask Dr. Melissa Moore, our Chief Scientific Officer, to come up in a minute. But the cells that we believe are important for the antigen presentation are the dendritic cells and the subcapsular macrophages. But to give you more detail, Dr. Moore?
Thank you for the question. Yes, the main cells, and as illustrated on the slide I'm showing here, that take up the lipid nanoparticles and express them in the draining lymph nodes are the monocytes and dendritic cells, also known as antigen-presenting cells. In terms of how long the RNA sticks around, the peak antigen expression is about 48 hours, and it's gone by 72 hours. The mRNA is generally gone by around 24 hours. So the protein sticks around longer than the mRNA.
Thank you.
Okay, I am going to to call a mercy rule here for everybody and apologize to the 6 people with their hands raised right now. Your turn will come later on. We're about 15 minutes late, and to allow everybody a little bit of time off, let's start at 2:05. So a 20-minute break right now.
All right, my headset on here. All right, jumping back in the saddle here. All right, so let's see what we got here. All right, so let's change this. Come back to the microphone here. All right, guys, peace, power, and profit. Again, thank you guys for tuning in. This is going to be a long day. These panels take a long time to go through all the information that they want to bring up, and you have last minute questions that come in out the blue. It's a strenuous process. You know, whether or not, again, of course, we say that our medical review boards and things of that nature take such a a fine tooth comb care value to make sure they put out the best information. Of course, this is some of what it entails. And but like I said, a lot of this stuff is not outside you guys' own cognitive value of understanding biology, at a level where you can comprehend it. Some of these medical terms, this, that, and the third, it's within you guys' ability to kind of absorb it and also make functioning, data analysis behind what they're actually saying, right? It's not outside our scope, and then we need to learn more about these things for that reason. And it's one of the reasons why there's so much, skepticism, again, like they're trying to say within it, definitely the Black community, but not just the Black community, because of course the Black community is the scapegoat, right? But they're not accounting for all the other groups out there who are very, very, adamantly anti-vaccine. And even more so, the fact that a lot of people who are following Trump, who really took the stance that they don't trust a lot of the data coming out, obviously, with anything that Dr. Fauci has to deal with, anything that Bill Gates has his name attached to, they don't want no parts of it. There's like, no, just strictly off the strength that the person is involved, we not trusting the data. And so, of course, y'all, like I said earlier, there's a big movement out there where there's two sides to this conversation or this argument, or whatever you want to call it that, where they're not necessarily trusting, this whole vaccine stuff, right? This ideology of, you guys, brought it to trial, but we may not be feeling so good about that right now, right? That kind of scenario, right? But that's why, and this is just one of many scenarios where you will have to kind of weigh the evidence in your own way, right? taking what they've been able to say, cross-reference against different ideologies. You never want to go to all the stuff that fits your narrative. That's the worst thing to do in any kind of scientific analysis. Just try to go to all the things that support your argument or your mental thesis, right? The mind or perception can obviously always lead you in the wrong direction when you have a strong feeling or appeal to the information that suits your narrative, right? When confronted with opposing views, how do you respond then? Right? Do you get flustered? You get upset? You feel like people challenge you as if you the one that wrote the dissertation on it? It's not even your information, right? So you're going to war with somebody else's information. So what does that really say about the researcher, right? We always going to come at it from a neutral perspective and try to again, obviously weigh both sides, the pros and the cons, so we can make the more detail-orientated analysis that suits our best interest based upon the most neutral information, right? And so we kind of fail at that a lot because we let our feelings get in the way, again, because of our strong, positions that we like to take sometime in some of these arguments. Again, I'm like, do you got beacons and microscopes and all that stuff hanging around the house? Or are you just reading what somebody else wrote and then you say, oh, that's facts? That kind of mentality, right? So, there's always going to be a handicap at some point in the aspect of information. But truly, when we're talking about peer-reviewed information, you're going to have to take it the distance, right? You're going to have to take it the distance, again, and cross-reference every and anything you can get your hands on so you can get the best, viable value of information that will help you make the best decision, right? And that's just that, right? So, of course, up for debate today is the Moderna, obviously, vaccine, the COVID-19, mRNA, serum, right? Because that's what I gotta call it at this point. It's a serum that they're going to somewhat try to get emergency use authorization so they can obviously administer it along with that what we got with Pfizer at this point, Pfizer and BioNTech, right? Got to make sure I add BioNTech in there, right? And they're again, it seemed to me earlier they was trying to have some questions where some of the doctors were trying to do that kind of slick value of trying to do a compare and contrast, right? They want to know what's the obvious differences. Well, one difference we know out the gate is that Moderna does not have to go through all the strenuous freezer, isolation in order for the vial or the strength of the vaccine or the serum to remain effective, right? And of course, like I said, just as a disclaimer, again, going way back in February, March, I was very adamant that, of course, if I had to have any dog in the fight, my best choice would be Moderna, based upon my value of research, especially at that time. And some of the players that were being named, like in Novio, Sorrento, Novavax, obviously AstraZeneca, Oxford study, the Johnson & Johnson kind of slightly came on the scene a little bit later. Then Pfizer jumped into the race, and then some of these more smaller subsidiary companies, they jumped in. I say subsidiary because they really don't have a really big backing, but they still have just as much opportunity to produce a vaccine like anyone else. But again, with that said, I was very adamant about the Moderna one because I like the science approach, the way they was going at sequencing the virus. Because when they got the sequence from the WHO, the the World Health Organization, and then they went to work because their technology is basically, they're kind of using that futuristic stuff to a degree, all right, this nanotechnology, biotechnology, so to speak, right, where they produce, or they work strictly from the mRNA aspect of DNA building, often in terms of being able to produce the antibodies, and you don't have to be affected with the actual virus. Right, unlike all the other major players out there, like GlaxoSmithKline, Pfizer, Merck, right, all the Sanofi, all the usual suspects, right. There are a lot of people that's still up for question today whether or not they've done horrible things to people, through their for-profit mentality and not for ethics mentality. How the medical industry, as we know it today, obviously is not necessarily the most ethical institutions out there, regardless of how much people say they've taken the Hippocratic oaths and all this, that, and the third. These people are for profit. If they were non-profit, of course, it'd seem more relevant. But they got to pay for this stuff, right? And we're not a government state sanctioned scenario or a socialist capital, I should say just a socialist environment alone. But either way, there's always going to be that disconnect. I guess that's my point I'm trying to say, right? And so it's going to revert back to us being able to do whatever we can to also help bring this information forward in a way where we can all understand it too, right? And so again, you don't have to go to school to have a PhD to be able to discern whether or not people talking the kind of language that you can comprehend too. That should be basic, right? If they're trying to sell something to you, if it's complicated, it's a catch, right? There's something missing, right? It shouldn't be so overly flamboyant of a conversation, right? All right, this extra verbose aspect, the dialect, to try to get you all hyped up about. No, it should be very streamlined and direct, right? Speak to the data, so to speak, right? And if you have to, use layman term words for people to understand it, right? There are lay people out there that obviously don't have, maybe a high school education or college or whatever the case, but they understand basic common sense, right? That's innately in them from birth. Every human carries that in most cases. And so again, in these moments, of course, you had a rush trial, which is obviously never have happened ever in the history of medicine. In this case, of course, in our time, where we are under these governmental regulations and organizations, where you had such a expedited scenario. So of course, within that, people want to know exactly what y'all did, because if you rushing it, that means you might have missed some steps. It may be some problems, right? And we don't want no problems, right? And so that's why people are hesitant. And not, again, they keep emphasizing the black community or brown people or Latino community or whatever, or minorities, whatever. But ultimately, let's be honest here. Let's be honest. Majority of people that push back against this stuff at that level are definitely, I would say, people of European descent. That's a lot of what the data says. They're using that per capita kind of analysis, right? Per capita, the black people as that the white people. It seems to live as if black people still more head. And I can show you tons of people and websites that are dedicated. Black people ain't putting up websites to be real big detractors of the vaccine. We just like, in our own way, we're like, look, man, I ain't taking that mess, right? We know what happened with Tuskegee. We know what happened under the medical apartheid, Carolyn Washington. We know about, all the stuff that happened back in the gynecology timeline when that first came about, how they were using, no anesthetic values with black women experimenting on them, using clamps and spatulas just prying open our black women. Let's be honest, right? So you can't escape that. So our people are skeptical because we know the people who we're dealing with. And you just got to take that, right? Well, no matter who you are, that's just the reality. You cannot, downplay history because it doesn't make you feel right, right? All right. That's just how life is, right? This is self-preservation. It's the animal kingdom out there. Even in our Homo sapien aspect, you still have to understand that, when people violate you, of course, there's always going to be distress there. There's always going to be some issue of trust, right? And so, in knowing that, that's why you have such a divide in the black community when it comes to us being able to be ready to live it. And again, it's not like we don't trust science, right? And I'm tired of hearing people say that, right? Just because you have an opposing view. Well, guess what? There's a thing called shades of gray. You don't have to be a linear thinker. If you're a linear thinker, you're missing out on life anyway, right? You're so missing out on what life is all about, right? Because everything don't come with a horizontal, grid value of how you should take it. I mean, a yes or no, right? There are shades of gray. There's stipulation. There's context, right? There's a lot of things that comes into factor when we start breaking down the analyses or the data. So again, the vaccine issue is one of, you have to be transparent as ultimately possible, and you have to use the kind of verbiage that reaches everybody's comprehension level, right? The government is already paying for it, so at this point, it's not like you will have some kind of secret serum juice, right? Go ahead and reveal that stuff and break it down. Give people these, these chemistry and biological lessons, case studies, so people can be more confident, and then let them make their decision. But again, and at the rate that we got the information, because the COVID was such a politicized situation coming out of the administration, of course, and Trump, they caused it more than anything. They made it worse. They compounded the issue even more because they kept trying to politicize it. Wear masks, don't wear masks. The Surgeon General, remember we posted that article with the Surgeon General way back in March? He was like, end of February, March, he was like, don't wear masks, right? They're not effective anyway. But in the same sentence, he said, we need more masks for healthcare workers. Like, what? Don't wear masks, but they're not effective. But we need to make sure you guys don't go out and buy healthcare or masks, N95 masks or surgical masks, because we need more for what? Frontline workers and this, that, and the third. Like what kind of, like, dude, you the Surgeon General. Who's briefing you? Where'd you get your education from? Like, seriously? So again, this is why there's skepticism. And then we already know everything that happened since after the fact. It was just like a snowball effect of just misinformation and craziness and just politicization, right? Of all this. Again, I like playing it down. I've always liked playing it down, right? Your brother, send your truck, right? You know, maybe you can inject Lysol or inject Lysol into your body and drink some bleach, right? Y'all remember that mess? Again, you know, maybe we could turn up, you know, a fifth of Clorox, right? Clorox couldn't even stay on the shelf, right? People went from cleaning it to thinking about actually drinking it, and they actually people that went out there and drunk that trash and died. There people went out there and drunk Clorox when he said it, and then he came back and said, "I was just playing." We just, you know how I did. We just kind of talk. You got to be careful because again, by doing so, a lot of people did those things. They try to inject Lysol in their body and they try to drink bleach, and they end up getting out of here. So I'm just saying. So words matter. So again, the politicization of this made it worse for everybody, right? And so that's why you have that difference or that somewhat of a sect of society. And I say sect because it's not a white or black. It's about we just don't trust. You know, we don't trust, and even more so, of course, black people. But we just don't trust, right? That's y'all problem, not ours. You spend the money that taxpayers paying anyway to educate them better. Then maybe you'll have more people more comfortable with the idea. But until that happens, y'all kick rocks, right? And so, of course, it's interesting when we listen. While we're listening to this panel discussion, you heard a lot of those doctors come on, explain the very thing that most of us are already already knowing, right? That's why there's a divide in the black community, and they were going out their way to try to pay people for the vaccine trials. I saw the articles pop in my feeds. You know, they'll pay you $1,200 to $1,500 to be a trial recipient for a vaccine, and I was like, okay, you know. But again, this is why it's important, right? This is why this stuff is important because we are up against a somewhat of a time clock. People already then kind of got what they call COVID fatigue. They already, you know, we saw what happened with Thanksgiving. How many people were in Thanksgiving? What happened? It was one of the most, it was unbelievable the amount of people that got on airplanes to go build their family for Thanksgiving. And the majority of those people that went, we already did the timeline. Those are the people who are now going in the caskets right now for the most part, for the majority of them that had, again, these underlying medical conditions that were, their body couldn't, sustain, and they end up dying. Or they infected a loved one. They flew home, infected grandma, granddad, somebody, even young people got messed up too. And then them people up out of here. All for a turkey leg or some mac and cheese or some candy yams. Right? America's a selfish nation. Let's stop playing. It's the land of decadence, right? It's the land of quote unquote stolen milk and honey. It's the land of stolen milk and honey. So people carry that entitlement mindset around here. They don't have the discipline that it would take. So because of that, that's why these vaccines are rushed. Because everything in this country has to be about so-called freedom of speech. But freedom of speech doesn't override common sense. I'm sorry. I don't care who you is, where you are, where you from, what you believe in, what's your ideology, what's your political affiliation, what's your whatever. Common sense still rules the roost at the end of the day. That's like idiots doing the prehistoric era, of course, when you had dinosaurs, right? If there was any kind of semblance of whatever so-called quote unquote upright walking being was like, he wasn't out there trying to compete with dinosaurs in terms of saying, I'm gonna hang out with these T-Rex today. Right? I think I'm one of the boys today. I just know they ain't gonna eat me today. Like, yeah, seriously, go out there and try it. Then go right on over there and hang out the left shoulder on T-Rex and get swallowed right on up. Right? Again, you got to think about that. So again, that's why we have this type of scenarios or issues that we're having today. People they ain't built for this. They talk a good game, but they don't live a good game. That's simple. They talk a good game, but they don't live a good game. Right? And that's the problem. And so until we understand that, right? Until a lot of people get come to terms with that, we're forever going to be subjected, right? We're forever going to be subjected to having to deal with, again, this kind of situation. So, of course, these vaccines are important for that reason, right? They're important for that reason. So, go. So, again, we're going to talk about it a little bit more in depth. Like I said, they get ready to start back up here. We'll see what's going on here. As far as what they're talking about. And then, of course, we'll kind of come back in and do some more commentary. And thank y'all again for tuning in. I know it's going to be a long day with this stuff because this stuff takes a lot of time. So I know you're going to be in and out the building. So we appreciate you guys for your support. Like, share, subscribe to this channel over here as well, right? The other channel's over there under quarantine right now, all right? But that's why we have a backup, right? So like, share, subscribe to African Emporium 2. Stay tuned for any kind of information dealing with anything that has to do with what we consider a benefit to us monetarily or even health in this case. But we are investors and traders. That's what that's our niche or niche. And so this information is important to us because Moderna, the stock itself is under trial today, right? It's just, it's going through the Rio Road, just like what we witnessed with Pfizer, right? And actually Pfizer are actually breaking out today. Ain't that something after being selling off? After it sold off on this news, but again, there's a difference there between both of the companies, and we're going to see exactly how Moderna respond if they get their emergency use authorization, right? The difference of how the stocks may react, and we're going to be watching it, all right? So stay tuned, all right? Thank y'all once again for tuning in. Let's get back at it. Appreciate you guys. All right, let me cut this back on.
Controlled efficacy studies in individuals 18 years of age and older. So looking at the phase 1 study, they enrolled a total of 120 participants in 3 age cohorts. There were 60 between the ages of 18 and 55, 30 between the ages of 56 and 70, and 30 participants 71 years of age and older. There were 4 dose levels tested, ranging from 25 micrograms to 250 micrograms. The immunogenicity assessments from the study showed that 2 doses induced SARS-CoV-2 binding and neutralizing antibodies, and Th1 biased CD4 T cell response was elicited. The safety profile supported further clinical development, and there were no concerning safety findings. As of the time of the EUA request, there has been no serious adverse events reported from the phase 1 study. The study was staggered in design, where the younger cohorts were enrolled earlier than the older cohorts, and some doses were later added onto the study. So there's a range in follow-up duration. At this time, all participants from this study have had at least 3 months of follow-up after dose 2, and a very small number has had up to a 6-month follow-up. So now looking at the phase 2 study. So in this study, there were 600 participants, half between the ages of 18 and 54, and half 55 years of age and older. Subjects were randomized 1 to 1 to 1 to either the 50 microgram dose, 100 microgram dose, or a placebo. Similar to the phase 1 study, 2 doses induced comparable SARS-CoV-2 binding and neutralizing antibodies in both age cohorts. No concerning safety signals were found. As of the time of the EUA request, there has been 3 SAEs that were reported in the vaccine group, but none were assessed as related. The immunogenicity and safety data are from the day 57 data cut, which comes to about 1 month after dose 2. But SAEs are reported more in real time. So the 3 SAEs are current as of the beginning of December. So moving on to the phase 3 study. So in this study, 30,351 adults 18 years of age and older were randomized 1 to 1 and vaccinated with 2 doses of the vaccine or placebo 28 days apart. Randomization was stratified by age and risk factor for severe COVID-19 into one of these 3 categories. Those 18 to 64 years of age without risk factors, 18 to 64 years of age with risk factors, and individuals 65 years of age or older regardless of risk factors. The protocol specified that the latter 2 categories should make up 25 to 50% of the total study population. And the risk factors for severe COVID-19 specified in the protocol are chronic lung disease, significant cardiac disease, severe obesity, which is BMI 40 or greater, diabetes, liver disease, and HIV. All subjects are followed for solicited adverse events, adverse reactions for 7 days after each dose, unsolicited adverse events for 28 days after each dose, and SAEs and medically attended adverse events for the entire study duration. The planned study duration is 2 years. So this is a graphical depiction of the study timeline in terms of scheduled visits and also when are the 2 efficacy analysis time points occurred. So starting on the left-hand side, subjects were administered 2 doses of the vaccine or placebo 1 month apart. A nasopharyngeal swab for SARS-CoV-2 RT-PCR was collected prior to each dose, as well as blood for immunogenicity. There are further scheduled study visits for safety and immunogenicity assessments during the follow-up phase of the study. Throughout the study, subjects are given weekly e-diary prompts as well as monthly safety phone calls, and active surveillance for COVID-19 symptoms begins after dose 1. Looking at the top of this graph, you'll see the dates of the 2 analyses that contributed to the data that we will look at today. So this slide just shows the case definitions used for the efficacy endpoints. So starting from the left-hand side, the primary efficacy endpoint for COVID-19 disease, that case definition is positive SARS-CoV-2 PCR plus at least 2 of the following systemic symptoms: fever, chills, myalgia, headache, sore throat, new olfactory, sorry, my screen disappeared. New olfactory and taste disorder. Or I'll just keep going since I have backup slides. At least 1 of the following respiratory signs or symptoms: cough, shortness of breath, or difficulty breathing, or clinical or radiological evidence of pneumonia. Let me know if I need to click something too.
Yes, just give us a second. Somebody accidentally hit stop sharing, so let me pull it back up. All right. It'll just take a moment here. Let's see. I just got to check the names, so I'll just take a moment. I think somebody accidentally. Did you try to hit the arrow accidentally? And was that it there?
I didn't touch.
What's the title of yours? Oh, okay. Share document. Hold on. This is going to be a small. We're just going to take a quick little break. Chad, pull us up on a break. Just while we pull this up, I want to make sure we get it. This is.
illness based on one of the vital signs, respiratory failure or ARDS, shock, significant acute renal, hepatic, or neurologic dysfunction, ICU admission, or death. So this slide shows the primary efficacy endpoint and how it was analyzed. The primary endpoint is confirmed COVID-19 occurring at least 14 days after dose 2 in participants without evidence of SARS-CoV-2 infection prior to dose 1. And baseline SARS-CoV-2 status is based on RT-PCR for SARS-CoV-2 and serology against the nucleocapsid prior to dose 1. For the primary endpoint, an independent blinded clinical adjudication committee confirmed whether each case met this case definition and should be counted. Vaccine efficacy was defined as the percent reduction, vaccine versus placebo, in the hazard of the primary endpoint, so V equals 1 minus hazard ratio from the Cox model. The primary objective would be met if the null hypothesis of H0 vaccine efficacy less than or equal to 30% is rejected at any of the interim or primary analyses at the pre-specified O'Brien-Fleming boundary. There were 2 protocol-specified interim analysis time points, the first after 53 cases have accrued and the second after 106 cases have accrued. Because of the rapid rise in cases around the time that the first interim analysis was triggered, there were actually 95 cases included in the interim analysis data cut. Similarly, for the primary analysis, which is specified in the protocol to occur at 151 cases, there were actually 195 cases by the time of the data cut. These are just 2 of the key secondary efficacy endpoints included in the study. The first is efficacy against severe disease, using the definition we just looked at a few slides ago, starting 14 days or later after dose 2 in participants without evidence of SARS-CoV-2 infection prior to dose 1. And the second is a less restrictive definition of COVID-19 based on the list of symptoms for COVID-19 by the CDC. And similarly, these are cases confirmed 14 days or later after dose 2 in participants without evidence of SARS-CoV-2 infection prior to dose 1. Cases of severe COVID-19 are reviewed in real time by the DSMB to monitor for possible signal for vaccine-enhanced respiratory disease, and a protocol-specified study stopping rule will be triggered if the one-sided probability of observing the same or more extreme case split was less than or equal to 5% when the true incidence of severe disease was the same for the vaccine and placebo participants. This was not triggered for this study. Okay. Next slide. These are the key analysis populations defined in the study. So the full analysis set are all randomized participants who received at least 1 dose of vaccine or placebo. Participants are analyzed according to the group to which they were randomized. The modified intent to treat set are all participants in the full analysis set who had no evidence of prior SARS-CoV-2 infection at day 1 before the first dose. The per protocol set are all participants in the modified intent to treat set who received the planned doses per schedule and have no major protocol deviations. The safety set are all randomized participants who received at least 1 dose. And sorry for the typo here, as opposed to the full analysis set, in the safety set, they are analyzed according to the treatment they actually received. So this slide will hopefully make it easier to see the difference in median follow-up duration for the 2 different analyses that we looked at, that we're going to look at today. So on November 30th, Moderna submitted data from their interim analysis to support the NUA. And as you can see in the orange bars, the median follow-up for safety and efficacy in these subjects at the time of the interim analysis was 7 weeks after dose 2. To align with the expectation for a minimum of 2 months of follow-up, as outlined in FDA's guidance, Moderna later submitted on December 7th additional data from the scheduled final analysis as an amendment to the EUA. As you can see in the blue bars, the median follow-up for safety and efficacy at the time of the final analysis was around 9 weeks after dose 2. So the majority of the slides that I will present today will show data from the interim analysis unless it's otherwise specified as the final analysis data. However, I just want to note that we have independently verified the vast majority of the analysis from the final analysis, and this includes the primary endpoint, the associated subgroup analyses with the primary endpoint, the key secondary endpoints, and the solicited and unsolicited safety data, including serious adverse events. And we have not identified any notable differences in terms of efficacy or safety profile with these additional 2 weeks of data. So these data did not alter the conclusions that we had already arrived at after a thorough review of the interim analysis data. So moving on into the efficacy data. So this table shows the demographic characteristics of the study population, and you can see that it was very similar among the vaccine and placebo participants. The median age was 53 with a range of 18 to 95. Around 25% of participants were 65 years of age and older. Looking at race and ethnicity, we have 9.7% of subjects self-identified as African American, 4.7% Asian, 0.8% American Indian or Alaska Native, 0.2% Native Hawaiian or Pacific Islander, 2.1% other, and 20% of subjects self-identified as Hispanic or Latino. Around 25% of study participants were healthcare workers. And based on protocol-defined risk factors for severe COVID-19, around 22% of study participants had at least one high-risk condition present. So this is a subject disposition table. And looking at this, you can see around 8% of subjects were excluded from the per protocol set, which is the set used for the primary efficacy analysis. And the primary reason was the subject being positive or having an unknown baseline SARS-CoV-2 status prior to dose 1. Around 95% of the subjects completed two doses in the per protocol set. And discontinuation from the study was rare, with only 0.2% from either group. So now here is the primary efficacy endpoint at the scheduled final analysis. So we can look at the top line. In all subjects, there were 11 cases of COVID-19 in the vaccine group compared to 185 in the placebo group, with a vaccine efficacy of 94.1 and a 95% confidence interval of 89.3 to 96.8. Dividing that up into age subgroups, in the 18 to less than 65 years age group, the vaccine efficacy point estimate was 95.6, so very similar to the efficacy in the overall population. In 65 years and older age group, the vaccine efficacy point estimate was slightly lower at 86.4%. However, the number of cases are small and the confidence intervals overlap with those in the younger age cohort and the overall study population. This is a subgroup analysis of the primary efficacy endpoint broken down into further age categories, stratification categories, and sex. And you can see that vaccine efficacy in each subgroup was comparable to the overall study population. And again, going through these next few slides, shown here is the interim analysis, but we have verified the final analysis for these subgroups, and there's no notable difference. This is the subgroup analysis of the primary efficacy endpoint by race and ethnicity. As you can see, efficacy was uniformly high across the groups. However, I do want to point out that for many of the subgroups, the sample size and the case numbers are small, and that limits the interpretability of the individual efficacy results. This is the subgroup analysis of the primary efficacy endpoint by the protocol-defined risk factor for severe COVID-19, and also includes at the bottom a post-hoc analysis of obesity defined as BMI greater than 30. Again, as you can see, efficacy across the board is consistent with what was seen in the primary endpoint. But for some of these groups, it is again limited by the small number of cases and the population. So this is a subgroup analysis of the primary efficacy endpoint by baseline SARS-CoV-2 status. And just as a reminder, that is based on RT-PCR and serology against the nucleocapsid protein prior to dose 1. Just over 2% of the study subjects were positive at baseline. So you can see that there is just one single case in the seropositive. So there's not really any sufficient data to make any conclusions on vaccine efficacy in participants with a prior history of SARS-CoV-2 infection. This is the secondary efficacy analysis of severe COVID-19 at the scheduled final analysis. So looking at all subjects, there were 30 cases in the placebo group, and 9 of these cases resulted in hospitalization and 1 resulted in death. In the vaccine group, we do note that there was 1 severe case in a vaccine recipient which occurred 2 months after dose 2, requiring hospitalization, but had not been adjudicated by the time of the data cutoff. This is the cumulative incidence curve of COVID-19 starting after randomization in the modified intent to treat set. And the arrows showing where the vaccine doses were given. And as you can see, the curve starts to diverge a little bit after the 2 weeks mark, and the divergence becomes more prominent as time goes on and more cases start accumulating in the placebo group. This is a post hoc analysis of COVID-19 cases from time of randomization in the full analysis set. So that means this includes all participants who have received at least 1 dose of either placebo or vaccine, and it's regardless of baseline SARS-CoV-2 status. So just looking at the second line, efficacy anytime after dose 1 to before dose 2 was around 69%. So this could suggest some protection after the first dose, but data is limited by the very short follow-up, so around 28 days, as the majority of the study subjects received the second dose. Okay, now moving on into the safety data. This again is a graphical depiction of the scheduled safety visits and safety calls throughout the study. Just as a reminder, all solicited adverse events are collected from all study subjects via an e-diary for 7 days after each dose. Unsolicited adverse events are collected for 28 days after each dose, and serious adverse events and medically attended adverse events are captured throughout the entire study. This is a subject disposition table, and you can see a vast majority of subjects completed 2 doses, and very small percentage discontinued the study, and it was similar between the vaccine and placebo groups. Okay, the next few tables are going to show the solicited local and systemic reactions. But again, before we dive into it, I just want to reiterate that although the data shown are from the interim analysis data, we have verified the data from the final analysis, and there was no notable difference compared to the interim analysis data shown here. So looking at the solicited local reactions after dose 1, you can see the most commonly reported local reaction was pain. Grade 3 events were rare after the first dose, and something that you see through the next few tables is that there is a lower rate of solicited reactions in the elderly cohort compared to the younger cohort. This is looking at solicited local reactions after dose 2. It is slightly higher compared to after dose 1. Grade 3 events are still pretty low. Now switching to systemic reactions after dose 1, similar to the local reactions, there's a lower rate in the elderly compared to the younger adults. And after dose 1, grade 3 or 4 events were rare. And finally, looking at solicited systemic reactions after dose 2, you can see there is a higher rate after dose 2 compared to dose 1, including a higher rate of grade 3 events. So, for example, fatigue myalgia is around 10% grade 3. Overall, based on the review of these last 4 sites, there were no serious safety concerns based on the data. Shown here is an overview of solicited safety by baseline SARS-CoV-2 status. The rates of solicited adverse reactions were comparable or sometimes slightly lower in participants with baseline positive SARS-CoV-2 status. But again, this group is much smaller in size compared to participants with negative SARS-CoV-2 status at baseline. This table shows unsolicited adverse events rates overall, and then further broken down into which are those which are related, which are considered serious, and medically related adverse events, and then also broken down by baseline serostatus. So again, the rates of these events are comparable or a little bit lower in those who are baseline SARS-CoV-2 positive compared to those who are negative at baseline, but again, that subgroup population is small. Unsolicited adverse events in general was comparable between the vaccine group and the placebo group. So FDA conducted standard measure queries, SMQs, using FDA-developed software to evaluate for constellations of unsolicited adverse events with onset following dose 1 through the data cutoff. The SMQs were conducted on adverse event preferred terms that could represent various conditions, including but not limited to allergic, neurologic, inflammatory, and autoimmune disorders. Here we just highlight the unsolicited adverse events which had a higher frequency in the vaccine group versus placebo. So starting with hypersensitivity related events, there was 1.5% in the vaccine group versus 1.1% in the placebo group. And the most frequently reported AEs in the hypersensitivity SMQs were injection site rash, injection site urticaria, and maculopapular rash. This we thought had a plausible relationship to this vaccination. And then also of note, no anaphylactic or severe hypersensitivity reactions with close temporal relation to the vaccine were noted. Lymphadenopathy-related events that's outside of the solicited period was noted in 1.1% of vaccine recipients and 0.6% of placebo recipients. The most frequently reported lymphadenopathy SMQs were injection site lymphadenopathy, lymph node pain, and lymphadenitis. Again, we thought this had a plausible relationship to vaccination. We also noted a delayed localized injection site reactions with onset after 7 days, mostly after dose 1, and this was noted in 1.4% in the vaccine group versus 0.4% in the placebo group. There was a numerical imbalance in Bell's palsy cases, with 3 cases in the vaccine group and 1 case in the placebo group. The case in the placebo group occurred 17 days after dose 1. The 3 cases in the vaccine group occurred 22, 28, and 32 days after dose 2. The observed rate was consistent with the background rate in the general population. There's no clear basis upon which to conclude a causal relationship at this time. Moving on to serious adverse events and deaths. As of December 3rd, there were 13 total deaths reported in the study, with 6 in the vaccine group and 7 in the placebo group. None of these deaths were assessed as related. Really quickly, in the vaccine group, the first 3 participants listed all had underlying cardiac disease. The first subject died of cardiac arrest 21 days after dose 1. The 77-year-old participant died of myocardial infarction 45 days after dose 2. The 70-year-old subject was found deceased at home 57 days after dose 2. The next participant was a 56-year-old subject with hypertension and chronic back pain being treated with opiate pain medication, who was found deceased at home 37 days after dose 1. And the official cause of death was head trauma. Then we have a 72-year-old participant with Crohn's disease and short bowel, who was hospitalized 40 days post-dose 2 due to thrombocytopenia and acute kidney failure, and then later developed complications during the hospital stay, including a perforated ulcer that resulted in multi-organ failure and death 59 days after dose 2. And lastly, we have a 62-year-old participant who died to suicide 21 days after dose 1. There were 3 SAEs thought related by the FDA. One is a 65-year-old participant with a history of severe headache and nausea requiring hospitalization, who developed intractable nausea, vomiting requiring hospitalization 1 day post-dose 2. And there were 2 subjects who reported facial swelling 1 day and 2 days post-dose 2. Both of these subjects had a prior history of dermal filler cosmetic injections in the cheeks. For one subject, it was about 2 weeks before vaccination, and for the other subject, it was about 6 months before vaccination. Also related, but there was one subject who had lip angioedema about 2 days after vaccination, and that subject also had a prior dermal filler injection in the lip. Interestingly, that subject reported a similar reaction after a previous influenza vaccine. I do want to point out that for these 3 subjects that I just mentioned, so the 2 with the facial swelling and the 1 with the lip swelling, the swelling was only localized. There were no systemic symptoms observed. Women were screened for pregnancy prior to each vaccination, and a positive test resulted in exclusion or discontinuation from vaccination. As of December 2nd, there were 13 pregnancies in the study, 6 in the vaccine group and 7 in the placebo group. Vaccination occurred prior to last menstrual period in 2 vaccine recipients and 3 placebo recipients. Vaccination occurred within 30 days after LMP in 2 vaccine recipients and 3 placebo recipients. And vaccination occurred greater than 30 days after LMP in 1 vaccine recipient and 1 placebo recipient. The LMP is not known in 1 vaccine recipient. In terms of outcomes, there's 1 case of spontaneous abortion and 1 elective abortion in the placebo group. Otherwise, all the other pregnancies are ongoing and the outcomes are not known at this time. So in summary, for the efficacy, the totality of the clinical data submitted with the EUA request meets the expectations for duration of follow-up. In the scheduled final analysis, vaccine efficacy 14 days or later post-dose 2 was 94.1%, with a confidence interval of 89.3 to 96.8 in participants without prior evidence of SARS-CoV-2 infection. Efficacy outcomes were consistent, greater than 93% across demographic subgroups. In the scheduled final analysis, there were 30 severe cases of COVID-19 in the placebo group and 1 still unadjudicated case in the vaccine group. The data suggested potential efficacy following a single dose, but interpretation is limited because almost all participants received a second dose. As far as for safety, the totality of the clinical data submitted with the EUA request meets the expectations for duration of follow-up in greater than 30,000 participants. Reactogenicity was generally more frequent after dose 2 in all age groups, mostly mild to moderate, and less frequent and severe in adults 65 years of age or older. There were no safety concerns identified in subgroup analyses by age, sex, race, ethnicity, health risk for severe COVID-19 or prior SARS-CoV-2 infection. Lymphadenopathy reported as solicited and unsolicited adverse events were more frequent in the vaccine group compared to placebo. A delayed localized injection site reaction with onset after 7 days was more frequent in the vaccine group compared to the placebo and mostly seen after dose 1. Hypersensitivity-related events were more frequent in the vaccine group compared with placebo, but no anaphylactic or severe hypersensitivity reactions with temporal relation to vaccination was noted. As of the scheduled final analysis, 3 cases of Bell's palsy were reported in vaccine recipients and 1 in placebo recipients. Although there's no clear basis upon which to conclude a causal relationship at this time, FDA recommends further surveillance if vaccine is authorized for widespread use. Now, moving on to the pharmacovigilance plan. Moderna submitted a pharmacovigilance plan to monitor safety concerns that could be associated with Moderna COVID-19 vaccine. The sponsor identified vaccine-associated enhanced disease, including vaccine-associated enhanced respiratory disease, and anaphylactic reactions, including anaphylaxis, as important potential risks. Use in pregnant and breastfeeding women, use in pediatric populations, long-term safety and long-term effectiveness, immunogenicity in subjects with immunosuppression, and concomitant administration with non-COVID vaccines are areas the sponsor identified as missing information. Pharmacovigilance activities, including adverse events reporting. Adverse events reporting under EUA may come from vaccine recipients, vaccination providers, or the sponsor. First, the vaccine recipients will be notified that an adverse event can be reported to VAERS, the fact sheets for recipients and caregivers. Another source of adverse event reports from recipients is the VSafe program, which is a smartphone-based program that uses text messaging from web surveys from the CDC to check in with vaccine recipients for health problems after vaccination. Reports from vaccine recipients are voluntary. Adverse events reporting by vaccination providers and the sponsor is mandatory. Both the sponsor and vaccine providers administering the Moderna COVID-19 vaccine must report to VAERS the following information associated with the vaccine: vaccine administration errors, whether or not associated with an adverse event, serious adverse events, irrespective of attribution to vaccination, cases of multi-system inflammatory syndrome in those, cases of COVID-19 that result in hospitalization or death. In addition, the applicant will also conduct periodic aggregate review of safety data and submit periodic safety reports at monthly intervals for FDA review. Each periodic safety report is required to contain a narrative summary and analysis of adverse events submitted during the reporting interval, including interval and cumulative counts by age group, special populations, such as pregnant women, and adverse events of special interest. Newly identified safety concerns in this interval and actions taken since the last report because of adverse experiences. Both FDA and CDC will take a collaborative and complementary approach to reviewing adverse events. FDA will individually review all serious adverse events on a daily basis. FDA will also examine other sources for adverse events, such as literature, and will perform data mining to determine if adverse events are disproportionately reporting for the candidate vaccine compared to all other vaccines in VAERS. Any potential safety signals identified will be investigated. The sponsor provided a description of studies they are currently planning on conducting. The studies will include completion of long-term follow-up from ongoing clinical trials, as well as the following two planned safety surveillance studies. The pregnancy cohort. The sponsor plans to establish a passive pregnancy registry to monitor vaccination during pregnancy with populations expected to receive the vaccine under EUA, and to submit a protocol for FDA review and approval. Active follow-up for safety. This study is an active safety surveillance activity conducting retrospective analyses of medical and pharmacy claims data to address three objectives: estimation of background rates of 23 pre-specified adverse events of special interest, descriptive analyses of observed versus expected rates, and self-controlled risk interval analyses that will be conducted if certain criteria are met from the descriptive analyses. FDA will provide feedback on these studies after further review of protocols once submitted by the sponsor. Proposed revisions to the ongoing phase 3 study, if an EUA is issued, is still in discussion. We have not yet received a revised protocol for review. In general, Moderna is proposing that there will be no changes for participants who choose to remain blinded, but for participants who choose to be unblinded, they will proactively reconsent and offer vaccine for those in the placebo group. Regardless of whether the participant remains blinded or unblinded or which treatment they receive, all participants will be continued to be followed for 2 years. Finally, we will now go into the benefit risk assessment. So the known benefits of the vaccine: reduced risk of confirmed COVID-19 at least 14 days after completing a 2-dose vaccination regimen in individuals without prior history of SARS-CoV-2 infection. Reduced risk of confirmed severe COVID-19 at least 14 days after completing a 2-dose vaccination regimen in individuals without prior history of SARS-CoV-2 infection. And in the subgroups, efficacy findings are consistent across subgroups by age, race, ethnicity, and comorbidity. The known risks. So local and systemic adverse reactions are reported at a higher rate after a second dose and a higher rate in younger adults compared to older participants. There were 3 SAEs related to vaccination, and they were all temporally associated and biologically plausible. And this includes the one subject with a history of severe nausea that had the intractable nausea and vomiting, and then the 2 cases of facial swelling in subjects that had a prior dermal filler injection. Serious hypersensitivity reactions have not been reported in this study, but have been reported in clinical experience with Pfizer mRNA vaccine. No specific safety concerns were identified in analyses of subgroups, including prior SARS-CoV-2 infection. The limitations of our risk assessment include the short follow-up duration and the fact that pregnant women were excluded. Here, just to remind everyone, the question that we would like the committee to discuss. In considering Moderna's plans for unblinding and crossover of placebo recipients, please discuss the most critical data to further inform vaccine safety and effectiveness to support licensure that should be accrued in ongoing clinical trials with Moderna COVID-19 vaccine, other studies, such as additional clinical trials or observational studies with the Moderna COVID-19 vaccine. And here is the question for vote. Based on the totality of scientific evidence available, do the benefits of the Moderna COVID-19 vaccine outweigh its risks for use in individuals 18 years of age and older? And this is the end of my presentation. I welcome any questions.
Thank you, Dr. Zhang, for not only being succinct and comprehensive, but also keeping us to time. What I propose is that first we entertain questions for Dr. Zhang on her presentation, and then go into a broader questioning of both Dr. Zhang and the sponsor about issues related specifically to the vaccines and the vaccine trials as has been reported. We should reserve the discussion about the unblinding issues to the later comprehensive discussion that the committee has, which will go on for a couple of hours, including the voting discussion. So first, let's ask questions, if you have them, for Dr. Zhang, and then we can have broader discussion, and I've alerted the sponsor to be ready for these questions. And when we get into the committee discussion about unblinding, we really need to focus on that issue. We got a hybrid discussion last week for those who are on with the committee, and I think we want to avoid that and focus on the FDA discussion point. So Dr. Offit, please.
Thank you, Dr. Zhang, for a clear presentation. I just want to follow up on something that both Dr. Cohen and Dr. Gans brought up earlier, which is just briefly this issue of Bell's palsy. And I understand that we're looking through there's the tyranny of small numbers that, derived from a large database, and you can't determine causality from such small numbers. And I'm really glad that you're doing follow-up. But I don't quite see how we're comfortable that what we're seeing with both the Pfizer trial and Moderna trial are background rates. If you look at that, the Pfizer trial was 4 cases of Bell's palsy in a group of 22,000 vaccinated per 3 months, which works out to about 8 cases per 10,000 per year. If you look at the Moderna trial, it's 3 cases per 15,000 per few months, which also works out to about 8 cases per 10,000 per year. If you look at the one placebo case if you add the 2 placebo groups, roughly 37,000, that over a few months, that works out to about 1.2 cases per 10,000 per year, which at least what I had read was roughly the background rate. That in combination with the fact that SARS-CoV-2 has been reported to be a cause of Bell's palsy in a handful of people, presenting actually first presentation being Bell's palsy and then found to have SARS-CoV-2, offers at least some biological plausibility. And in fact, it may be true that SARS-CoV-2 is a more common cause of Bell's palsy than this vaccination. And we'll find this all out in follow-up. But I'm just not quite sure how we are so comfortable that this was a background rate. I guess that's my question. Thank you.
Sorry, I just had to find the unmute button. Yes, this is something we're also looking into and thinking a lot about. Based on each of these individual studies, we're looking at the cases there is still no clear basis upon which to conclude a causal relationship, but we definitely see your point with the two studies combined, the numbers, and it's something that we are, looking into and thinking much about.
Thank you.
Thank you. Dr. Wharton, please.
Thanks. I'm interested in these 3 cases of facial swelling associated with the prior injection of the dermal fillers. How long did those swelling reactions take, and should this product be authorized, will this information be included in the information for healthcare providers?
Okay, I can just give you a little bit more information on those cases. Again, all 3 of those cases that I mentioned were just localized, like swelling in the cheeks or swelling in the lips, and they resolved with either antihistamines or a steroid course. And again, no systemic reactions were noted. And it was really interesting that one participant who reported a similar reaction after a previous vaccine. And we did a literature search, and it seems like this is something that has been reported, that with these dermal filler injections, it's localized some interaction in the cheek or swelling after a natural infection, such as like an influenza like illness, with these dermal fillers that create this temporary swelling that usually resolves pretty quickly with the steroids or by itself. So we are planning to note this in the prescribing information.
Thank you. Dr. Gantz.
Thank you. Thank you for that. I just had one question about a clarification. You had noticed that, noted some of the regulatory events that will happen in terms of adverse events, and you listed it only under EUA, which obviously is what we're considering now. And I just wanted to clarify that those functions will continue as we move out of an EUA into maybe a BLA or other forms in which we should still be looking at adverse events as this vaccine is rolled out. So I just wanted to make sure that it wasn't so specific to just under an EUA.
Yes, so if you remember that really busy slide with a lot of boxes and arrows, so the surveillance and follow-up for EUA is not any less demanding or more demanding than a regular BLA. So all of those will continue.
Thank you.
Dr. Sawyer.
Thank you. My question relates to the anaphylaxis story, and you described an imbalance in hypersensitivity reactions between the vaccine and placebo groups, but that there was no cases of anaphylaxis. I wonder if you can characterize for us what those hypersensitivity events are, because I wonder if some of the media reports are reflecting hypersensitivity reactions that aren't truly anaphylaxis, things like simple hives, at least until those cases get fully adjudicated.
Sure, thank you for that question. So, like I mentioned, when we searched by the SMQs, the most common preferred term events that we found under the hypersensitivity related events were injection site urticaria, injection site rash, or rash in general, or or hives, or itching. So nothing that really are close even to anaphylaxis.
Thank you.
Dr. Neaton.
Thank you. Thanks for the presentation. I wondered a couple of things on the safety. I noticed for the safety kind of cohort that was looked at, there's an excess of withdrawals and consent from the placebo group. Did you notice that, and was there anything, reasons for those withdrawals that kind of could make you question the blinding, or was it due to adverse events? But there was quite an excess.
Yeah, it was a little bit imbalanced in terms of withdrawal by subject, but it wasn't due to adverse events or physician decision due to any medical conditions or anything. Overall, the numbers are still very small. That's the difference of maybe like 60 subjects between the vaccine and placebo group. So looking at the overall safety set, it doesn't really make any impact.
Or the difference I was thinking about is like 3 or 4 standard error difference, which seems potentially not due to chance. So, okay. So my other question was is that in the FDA book, you provided more information about the duration of some of the solicited symptoms, and I noticed that, for example, for a lot of the symptoms, you just take myalgia as an example. There's a pretty striking difference if you look at solicited symptoms, but the difference is very, very small with unsolicited symptoms. And is that, do you think, primarily attributable to the timing of when those measurements were made?
Yes, correct. The solicited symptoms were collected within the first 7 days after vaccination. That's when we expect most of these symptoms like myalgia, fatigue, and things like that to occur.
Any medication provided to patients or recommendations for medications to prevent kind of some of the symptoms that were recorded?
I don't have that data offhand, but there is a use of antipyretics was also collected in the e-diary.
Thank you.
Thank you, Dr. Carrillo.
Thank you. Yes, this is about the potential for correlates of protection out of this trial. There was no immunogenicity data that was presented as part of the phase 3, but looking at the phase 1 immunogenicity, particularly in the elderly population in the 2.5 month period, there was rather substantial drop-offs in both the total ELISA as well as the neutralization titers that were measured. And I'm wondering, from your presentation, it looked like there was a blood draw at day 57, but not another one till 209. And I'm wondering if there's just an adequate a measurement of immunogenicity in that phase 3 to try to derive potential correlates of protection.
Maybe I will ask Moderna to address how they're planning to assess correlative protection in their studies.
Sure, happy to do so. So the correlative protection, as you noted, are the immunogenicity assays that were not yet available at the time of submission of the EUA. We're anticipating that the immunogenicity analyses should be available in the coming months. And so trial actually routinely got blood samples at various time points, so pre-dose 1, pre-dose 2 at day 57, which is 1 month post-dose 2, and then at 3 months, 6 months, and 12 months afterwards. And the idea would be that we would first present the immunogenicity analyses, and then once we have sufficient breakthrough cases to be able to perform the serocorrelate analysis, that will be done as well. We're actually working in collaboration with the NIH, so Dr. Fullman and Dr. Peter Gilbert to pull together this analysis, and it actually will be done with NIH assays in order to be able to look at consistency across other products.
Dr. Fuller.
Yes, thank you. So Dr. Zhang, there are some side effects which are expected with most vaccines, and they're just part of what happens. Especially in this time when COVID is such a major issue, what is FDA or CDC or Moderna, perhaps Moderna has the plan for informing people of what to expect? In other words, we can handle things if we know that this is part of what's expected and it's only going to be a few days, and we have somewhere to report it to if we think otherwise. So I don't remember the plan for how people will be informed of what the side effects may be as they go to take this vaccine. Can you help remind me, please?
Well, I do know that the side effects are going to be described in detail in the fact sheets for providers and as well as for the recipients. I'll open up for other people to chime in other things.
So I guess I'm asking if there's going to be some sort of campaign to make sure that people, and we all get fact sheets with our medicines or our vaccines or whatever, and we read them sometimes and sometimes we don't. And you can say that's on the person who's taking it. But for something like this, it would be really helpful and build trust if there's a major effort to say, this is what you should expect. These have been seen often, or and these have not been seen. That would, I think, give people a lot more confidence.
Hi. So as you've heard, we have an intensive safety surveillance system stood up for distribution of vaccine under EUA. We'll be monitoring the system closely. If we detect any signals, we will investigate those rapidly. And if we conclude that there is a need to inform vaccine providers or recipients or the general public about a risk that has not been previously appreciated, we will do so in revisions to labeling or sooner through safety alerts if we determine that that's warranted.
Thank you, Dr. Hildreth. Dr. Hildreth.
Did you call my name? I'm sorry.
I did. You had your hand raised.
Yes, sir, I did. My question relates to the fact that for every diagnosed case of COVID-19, there are probably several others that go undiagnosed. And I'm wondering if by giving the persons who've already been infected a single injection of the vaccine, could that serve as a boost and achieve the same goal as given 2 injections of the vaccine? In other words, infection serves as the prime and one vaccination will serve as the boost. Is that something that Moderna or FDA or has considered as a possibility, just out of curiosity?
I'll defer this to Moderna. This was not in the scope of the data or the study design.
Okay.
So just to make sure, Dr. Hildreth, I understood your question, are you asking about the interchangeability of our vaccine with the other mRNA vaccine, whether you could get a mixed schedule of both?
No, I'm referring to the fact that we now know that there are probably tens of millions of Americans who've already been infected by the SARS-CoV-2, and we know they can get reinfected. We also know that all of them make an antibody response to the virus, but it appears not to be protected because they can get reinfected. What I'm asking is, if you took the ones who've already been infected and gave them an injection of your vaccine, could that possibly serve as a boost, whereas the infection itself served as the prime?
Okay, thank you for that clarification. So I think it's something that we may be able to tease out a bit in our booster study. I mean, again, we had only 2.2% of the study population that indicated that they were previously SARS-CoV-2 positive. We are intending to evaluate booster doses, and as we review the immunogenicity, that is certainly something we can look at. And once the immunogenicity data are available, we'll be able to see what the initial vaccination looks like in the mRNA 1273 group. So don't have data, unfortunately, to share with you today, but we are anticipating those data in the coming weeks.
Thank you.
Thank you. And Dr. Miller, don't go away because we're expanding the discussion right now. Dr. Meisner has been waiting to ask some questions of you.
Thank you. One question I'd like to ask is about the CARS plot that are on figure 7 and 8, and I realize you probably don't have this right in front of you, but my question is, there were approximately 9,000 white subjects in the placebo arm and 5,000 from communities of color. But the rates of infection were 16 per thousand versus 8 per thousand in the community of colors. That is, they were lower. And usually we think of COVID-19 causing more disease in the community of colors. And is there a ready explanation for that? Perhaps it's the small numbers. Do you think that was truly representative of minority groups?
So thank you for that question. I'm attempting to pull up that fourth slide now so that just to reorient everyone to the discussion we're having. And so to your question about the small numbers, it is true that enrollment of minorities in the trial was a priority for us. We received lots of help and advice from our collaborators and from thought leaders in those communities. Nonetheless, the study was not designed to look at individual efficacy estimates in various demographic groups. And so, indeed, the numbers in each specific group are quite small. The study was actually powered only for the symptomatic COVID-19 disease. Hopefully, we'll have some refinement of those numbers. We intend to, regardless of what happens with the evolution of the clinical trial, we will continue to follow the participants who have been vaccinated with vaccine, placebo, or have been crossed. Our overall attack rate was 56 approximately per 1,000 person years, which is close to reported rates in the literature. So we'll be able to further add to those numbers and get some more refinement on them.
Thank you. I'd like to ask, secondly, a question in regard to sterilizing immunity. I think your preliminary figures are very promising that the vaccine may reduce infectious virus and the risk of transmission of fully replication competent virus. Has there been any effort to look at antibodies in respiratory secretions from the upper respiratory tract or the lower respiratory tract? Because I suppose if this is in fact true, I guess it means the intramuscular injection stimulated efficient circulating IgG so that it gravitated out into the mucous membranes of the respiratory tract. Is that reasonable?
So I think you're correct that it's certainly reasonable to expect that IgG is playing an important role in what we're seeing from an efficacy perspective. Don't have data on the IgA, but what we will have to hopefully be able to help us better understand viral shedding and burden of infection are the viral shedding samples that were taken from the cases confirmed by our SARS-CoV-2. So those subjects days over the month of their convalescence, and ultimately we will take those results and compare in the breakthrough cases, placebo cases, with viral shedding.
Right. And Dr. Meisner, that's something you may want to bring up in our discussion about other studies the sponsor might be asked to do. Dr. Pergam.
Thanks for great presentations, guys. My question is specifically around additional data transparency. A lot of what you're talking about here is things that are coming down the line, and I'm just trying to figure out additional shedding data, additional follow-ups that you're talking about. Moderna has been really transparent with data so far. I'm curious about what the FDA's approach is going to be in presenting this additional data to the public and to other committee members as this moves forward.
Apologies, Dr. Foregone. I think that was a question for Dr. Zhang, but I just want to be sure I understood that correctly.
Dr. Fink.
Yes. So we will continue to update the prescribing information and fact sheets as appropriate as we get new information. If we determine that that information is necessary to inform vaccine providers and recipients about the benefits and risks of the vaccine, of course, we will include as part of our review process for any licensure application a transparent review of the data to support that application as well.
Thank you. Dr. Perlman.
So I just had a question about the vaccine adverse events, the respiratory disease, and the general systemic problems that could occur after vaccination. It's mentioned in the protocol, but there aren't really very many details of what's going to actually be looked at. And the fact that it's so efficacious makes it less of an issue, but still, what's the exact plan for measuring adverse events after vaccination, the respiratory disease and the other?
Sure, I can take that one and speak to the various ways in which we're measuring safety in the protocol. So after vaccination, subjects had an electronic diary on which they recorded solicited local symptoms, so the injection site reactions, and then solicited systemic symptoms like fatigue, headache, myalgia. After 7 days, the e-diary as well as safety phone calls from the site prompted subjects to respond back about unsolicited adverse events. So these were any adverse events that may have happened to them, and we followed those 28 days after each vaccination. Then for some specific categories of adverse events, including medically attended AEs as well as serious adverse events, we are going to continue with the safety phone calls throughout the duration of the study for the subjects and capture that information. And so that's really the framework in which the respiratory illnesses you're speaking about will be captured. Then as part of the efficacy surveillance, there's also the surveillance for COVID-19 disease. And for those subjects who are not COVID-19 positive or SARS-CoV-2 virus positive, we'll also be looking at a respiratory panel of viruses to try to understand that respiratory disease. And again, some of these endpoints are not yet available to be reported out, but we intend to continue that surveillance throughout the study.
Yes, I guess I really meant vaccine enhanced.
Okay, thank you. We're going to have 2 more questions, and then we're going to be shifting and going to a discussion among the committee of FDA questions. So, Dr. Chatterjee next.
Yes, thank you, Dr. Monto. I have 2 questions, Dr. Miller. The first one is with regard to the nanoparticles. I think we heard today about the rate of decay of the mRNA and the protein that it codes for. But what about the nanoparticles? How long do they hang around?
Yes, so the nanoparticles have been evaluated in biodistribution studies, and they hang around for approximately 48 hours.
I see. And then the follow-up question to that is there a theoretical possibility that the body will mount an immune response to the nanoparticles, the lipid itself? And if that happens, would it then preclude the use of these nanoparticles for any future vaccines that are developed in the same manner?
So to answer that question, I'm going to ask our Chief Medical Officer, Dr. Zach, to take that one.
Thank you, Dr. Miller. Not as far as we know. So let me make a few comments here. The particles, traces are gone by 48 hours, just to be clear. They hang around for just a few hours. The components of those particles, as far as we know, are non-immunogenic in the sense that, as I described to you, you've got the PEG with a lipid. Most of us are walking around with antibodies against PEG, but they're not really meaningful in the sense of preventing further utility of drugs. And in fact, lipid nanoparticles, both by us and other companies, are being used for routine administration of other drugs and other experimental medicine so far without any evidence of that kind of reactogenicity. So I don't think we have any basis to expect that, neither based on our totality of preclinical data from our experience, nor based on the history with these kinds of LNP medicines used in other applications. And those applications are even using much, much larger amounts of quantities. So in short, I don't believe that's the case.
Thank you.
And finally, Dr. Kim.
So this is a question for Dr. Miller. I'd like to ask how you, Moderna, came about selecting 100 microgram as the vaccine dose for phase 3. In your briefing material for phase 1, you outlined your considerations for comparing 100 micrograms to 25 and 250 micrograms. And in study 201, you concluded that the data support of two-dose schedule of either 50 microgram or 100 microgram for rapid induction of functional antibodies against SARS-CoV-2, and then selected the 100 microgram dose for phase 3. What other considerations did you weigh in selecting 100 microgram over 50 microgram? And I ask this question because reports of any local reaction to the 100 microgram vaccine were around 70 to 80% in phase 3, and wonder what the safety profile might have looked like otherwise.
Thanks for that question. And I would like to emphasize that at the time the phase 1 study was ongoing, and when we had to select the dose to be able to start phase 3, the 50 microgram data were not yet available in the phase 1 study. Nonetheless, it's hard to look backwards and say what you would have done, but I'm not sure we would have taken a different decision. At the moment, we're quite comfortable with the consistent and high efficacy that we've observed, but at that point in time, all we knew was that we were in the midst of the pandemic, and we wanted to be sure that if we were going to undertake this large-scale safety and efficacy trial, and we were going to expose people to a novel vaccine, that we had the best possible chance of demonstrating efficacy. Another important point, as the 50 microgram data became available later, and particularly in the subjects over 71 years of age, there was an indication that the 100 microgram dose was more immunogenic. And so knowing that the older age group is a group that is at significant risk for severe complications of COVID-19, that was another reason really to choose the dose. And then the final reason is duration of efficacy is going to be important as we hopefully ultimately exit this pandemic. And we believe that the highest possible antibodies might lead to the longest possible duration of protection. So for all of those reasons, the 100 microgram dose was selected. Nonetheless, again, recognizing that we are in a pandemic, and now that we have data from the phase 2 available in 50 microgram doses, that's why we put such emphasis on that correlative protection work we're doing in the phase 3 study to see if there may be possibilities for immunobridging based on that correlate in the future.
Thank you, and thank you to Moderna and FDA for your presentations. Now we're going to be moving on to the item for committee discussion without a vote. And is it possible to put up the questions? I'll read them off while I've got them in front of me. In considering Moderna's plans for unblinding and crossover of placebo recipients, please discuss the most critical data to further inform vaccine safety and effectiveness to support licensure that should be accrued in, and let's do this one at a time, ongoing clinical trials, so it's ongoing clinical trials with the Moderna COVID-19 vaccine. And then we'll talk afterwards about additional studies. So this is the ongoing studies. Let's see. Dr. Gantz.
Thank you for this. So ongoing critical data, we still have multiple time points at which the Moderna is going to be collecting blood. And I think it's a really missed opportunity, particularly if they are actually collaborating with the NIH, who has quite sophisticated ability to look at T-cell immunity, which we know is very important to maintain our humoral immunity and will be very, very important. So there's two elements to this. Moving forward, which I think are very critical. A, to get them any time points at which you're collecting other blood samples, who is mentioned 6 months and further. But specifically when there's breakthrough, it's going to be very important to be able to do the parallel T-cell study to our B cells, because if the B cells aren't present, it's going to be very important to understand what T-cell immunity was there that could be potentially boosted. So in both of those scenarios, that's critical data in which to move forward and be able to understand this better. The other critical piece of ongoing information that I think is going to be very important is to look at this idea of whether people who are vaccinated can continue to be spreaders of the disease. And so looking in household contacts to see if there's any disease in those individuals who are not prioritized to receive vaccine is going to be very important, so following those forward. And then lastly, doing the viral studies that are needed to be done within the vaccinated population. So continue to do those surveillance of the PCRs for RNA, but it will be really important not only to look for the positive strand, but the negative strand, so that we can understand that this is a viable virus. So outside of the populations that they've already talked about in terms of ongoing, these are the critical things that I think are important. Thank you.
Thank you, Dr. Rubin.
Thank you. I echo what Dr. Gant said, and a couple more things. Antibody studies and T-cell studies, so we can look at correlates of immunity, because that will be a very, very helpful in the further development of the vaccines and for following waning immunity. So I think that those immunologic studies continue to be important. Of course, monitoring asymptomatic infection, as Dr. Gant said, is critical. And as has already been brought up, and it sounds like it's already a plan, looking at escape mutants for loss of neutralization by the antibody or loss of T-cell reactivity. I do want to go back, though, to Dr. Goodman's talk, because that is all part of this. And the current, it seems to me, at least, that the trial should have been designed as a blinded crossover study from the start. And my guess is that it's relatively impractical at this point to do it, disappointingly, because it's so late in the game. But I would encourage FDA. I know it's not quite a level playing field, but as new sponsors come in, I would encourage FDA to really consider that going forward. For now, I think they're going to be stuck with an open label study of the kind that Dr. Dayton outlined. I'll stop there.
Thank you for bringing us back to the nub of the question. Dr. Wharton.
So I am particularly interested in continued safety follow-up, as well as follow-up on the duration of protection. I think those are really critical factors that need to be taken into account as the study continues.
Any suggestions?
Well, there will be opportunities to learn more based on other studies being done, that in terms of the ongoing clinical trials, it's just important that the safety follow-up be continued, and that there be attention to the duration of protection question.
Hey, Dr. Nathan.
Okay, thank you. Yeah, thank you. Well, I want to go back to the presentation that Dr. Goodman gave this morning and also Dr. Baden. I understand, and I guess it's all in reference to just speaking to one factor, and that's the durability of this vaccine. So it seems no matter whether you're going to do a blinded crossover, as was suggested by Dr. Goodman, or are you going to do an open label kind of approach that Dr. Baden thought was appropriate given the situation, and practically that's what can be done right now. There's an opportunity to at least do immediate versus deferred kind of vaccination of the vaccine subgroups which were identified as at different risks. So the healthcare workers, the high-risk older people, and so that I would take advantage of that. Because right now there's only 17% of the participants that have 90 days of follow-up. And I think additional follow-up, which I guess is accruing right now, another couple of weeks, I think we need more follow-up with this vaccine versus placebo to understand more the kind of durability of protection. That's what I would suggest doing.
Thank you, Dr. Schooley.
Thanks very much, Dr. Monto. I also want to emphasize that I think this planned crossover study is a great opportunity to get some of the data about durability of immunity in a very structured way. And I'd encourage these sponsors to consider carefully constructed cohorts representative of the populations that are of most interest, ranging from age to gender, ethnicity, and so forth, that would let us look at decay of both humoral and cellular immunity. The crossover would be a chance to reset the clock and get cellular immunity from the outset, and to incorporate mathematical models to look at decay kinetics based on the induced immunity in individual people, and decay across different groups based on their demographics and hypotheses about immunogenicity in different patient populations. And to correlate that with viral shedding, that is in breakthrough cases, not just dichotomous data, but quantitative data, to get a better idea about durability of immunity, and to start thinking about how this might play into studies later about when to boost and when to revaccinate, because we know about the durability of coronavirus immunity in general, and there's no reason for this virus to be any different.
Dr. Chatterjee.
Yes, thank you, Dr. Monto. With regard to the ongoing clinical trials, my understanding is that there are pediatric trials ongoing, so this is not in reference to the trials that we were discussing today, but certainly would encourage those trials to continue and for us to track those data. As far as pregnant women, my understanding again is that according to the criteria for inclusion, efforts were made to not include women of childbearing potential, but I think it's critical given the workforce and the role that those women have in our workforce and the high risk that they incur caring for patients with COVID, that the studies be also conducted in that population.
Dr. Sawyer?
Thank you. I think in the ongoing trials, we have an opportunity to learn more about asymptomatic infection in that a significant percentage of the study participants are healthcare workers, and many healthcare systems are starting now to do routine testing of all healthcare workers. In my system, it's every week. And I would encourage the sponsor to try to collect that data and make some comparisons between vaccine and placebo group.
Dr. Pergan?
Hey, Arnold. So I think two things that make sense to me is when they're, I really hope that if there is this crossover design, that they continue to do additional viral testing within those individuals, because that's a critical piece to know about potential transmission in that sub cohort, and particularly to look at viral load. I know that's sort of sometimes can be a difficult process with nasal samples, but when we're thinking about transmissibility and the levels of virus that are there, that might be one of the advantages, potential advantages of the vaccine. I'm also curious within this study, if Moderna can speak to us at some point about how many of the 25% that are in the study that are healthcare workers have already opted out because they know they might be eligible to get the Pfizer vaccine. That would be an interesting piece of data. Maybe too early to know that. That would be an interesting piece of data for us to know sort of what expectations might look like for other groups who may be deciding to go and get the actual available vaccine.
What I'm hearing from our members is two streams of discussion. Additional studies that can be done, whatever the specific design, unblinding with open label, a crossover design, and additional studies that might be done. I'm not sure how to bring the two together. What I think we might want to do in our discussions is to focus on what happens with the issue that I think is troubling to some of us, and that is the inevitable loss of the placebo group, which occurs whatever you do, whether it's unblinding and open label, or unblind, or a crossover design without unblinding. Can we focus on that with some of our questions? Then we'll get back to some of the additional immunologic shedding, viral shedding. This is a very difficult thing for us to do in a virtual setting. If we were around the table talking to each other, we could address these issues much more efficiently. But let's try to talk about the placebo group first. And Dr. Gans, are you going to be talking about the placebo group?
Yes, thank you. I did want to just raise an important component that I think maybe the twist hasn't quite been raised yet. We're all concerned about losing that placebo group and really the integrity of the data moving forward. But I think we do realize that that is something that is going to be offered to individuals who got the placebo. So the only way that I see that we can really hold on to the integrity and continue learning something is that is to continue the blinding of the study. So it doesn't impact, everyone gets what they want. It doesn't impact the participants. They're all going to be vaccinated, and within 6 weeks, everyone will actually know that they've fully been vaccinated. I mean, we need 2 doses, and we know that that's what you need to be sufficiently immune, for this to be efficacious. And so, so everyone gets what they want. You could use the vaccine that actually is now coming to expiration, but you do it in a blinded manner, and in that way you uphold the integrity and the ability to really look forward. It's going to change everyone's behavior otherwise, and that will actually impact the results. So that is what I would plead to Moderna and to say that it seems that everyone gets actually what they want at that point. Thank you.
Thank you, Dr. Gans. Mr. Taubman?
Can you hear me?
And, yes.
So, I'm taking a broader view of this, that this study was funded in part by the taxpayers through Operation Warp Speed, and therefore the government does have some ability to impose some rules. And it seems to me there's an assumption that Moderna is going to unblind the entire group, the entire placebo group. And it doesn't have to be that way. We haven't been asked to vote on it, though we could vote on it as well. But we could say that we don't think it's acceptable for the Moderna plan to go forward if it's granted EUA. And as just an example, it could be either you do exactly what Pfizer is doing, and Pfizer ignored the advice from Dr. Goodman basically last time, do the blinded crossover. So they're not doing that. And instead, they've just offered unblinding to all the healthcare workers, and the other 80% stay in the placebo. So that's one option. Another option would be the blinded crossover. But if we aren't very clear that we think strongly that that's what should happen, one or both of those, or one or the other, then what's going to happen is that Moderna is just going to do what it's going to do, as happened with Pfizer. So I would strongly urge that we discuss the possibility of having a vote or directing what we think for only Pfizer. I totally understand that. But if the committee felt strongly that this is the way it should be handled in the existing study because of the worry about losing placebo folks, I think that would have significant impact with FDA. And FDA, federal government dollars here, would say these are the conditions upon the EUA being granted as we want the study to be maintained in a certain way.
You've raised some specific questions. I would urge the members to try to address some of these questions so we get a sense of the committee. Dr. Meisner.
Mr. Tubman, I think that a very reasonable suggestion, take a vote, maybe give some support to Moderna. I would also like to go back to the point that Dr. Melinda Wharton raised and the importance of having a blinded cohort in this study, because eventually this will go for a BLA, and it will then be added to the vaccine injury compensation table. And it's going to be so difficult to add this to the table without some evidence of well-established adverse reactions. If they occur, and without a blinded trial, it's going to be, or a blinded group, it's going to be very difficult to answer that question. So and what I would like to do is ask Moderna if they have a sense of how soon they might submit a BLA to the FDA, because once that happens, it'll be the end of any randomized trial. And how quickly might the FDA turn around a BLA that they receive from either of these two companies?
Thank you, Dr. Meisner. You've raised some points that we may need some guidance from FDA and perhaps from Moderna as well. Dr. Ruber?
Can you hear me? I'm sorry.
You can.
Good. I just want to make a comment regarding Dr. Taubman's suggestion to turn the discussion point into a voting question. I believe, we had discussed that if we should do this, but we decided because of the complexity of the situation, and as you said, we have not only one, we have the two companies, to not turn this into a vote at this time. We didn't really ask this discussion point to be a voting question a week ago, but I think what we would like to hear from the committee, and I have heard some committee members here opining very clearly that some said they support the open label design or crossover that Moderna is suggesting, and others are pleading with really entertaining a blinded crossover. So if we hear the committee members to speak out on these very specific issues, what they would suggest and what they think should be done, then I think we have reasonable guidance from the committee on how to proceed in our discussions with the respective companies over the next couple of weeks. In terms of BLA and biologics license application and how fast we could move to that and what data we need, I mean, we all realize that the placebo-controlled blinded follow-up is the gold standard of every clinical study that is conducted. At the same time, we do realize that it may at a certain point not be longer feasible. I think we would be, working with the companies over the next couple of months to see, what data do we need to support a license application and what can be done. And it is not only, and then I'll stop, it's not only the clinical data, it's also the manufacturing information, the facilities information that will be very critical here and will make and will be a deciding factor as to when we would be able to move to accepting the biologics license application. Over.
Thank you, Dr. Gruber. And I would urge the committee members in their of the committee without a vote about some of these issues, unblinding, blinded crossover, or continuing whatever we can with a blinded placebo-controlled design. Dr. Fuller.
Thank you, Dr. Marshall. So yes, that's exactly what I wanted to comment on. It is a research dilemma. When you want to get the best data you can, you must have the controls there. But in this case, these are people who may decide that they don't want to stay in the study because it is such a severe issue. And so even if we kept the study as a blinded study and they're not there, then we wouldn't have the data that we want. So I think Moderna has done a great job of designing their study so far. And if that's what they recommend, and because we want the people who are in the study to remain available and accessible to get whatever data we can, I would probably go with the unblinding to keep them in the study to get what we can. And then the second point I want to make.
Okay, when would you unblind? Dr. Fuller, when would you unblind? Before or after they become eligible based on the priorities?
I think I would unblind when the study has gotten from them what they need in terms of the timing. So if I were in the study and they told me you are eligible in 3 weeks, but if you stay in 5 weeks or 6 weeks, we will be able to get this much information and we can make sure that you get this vaccine. So I guess it's communication to me. And then very quickly, I do want to re-emphasize, as we said earlier, the importance of having pregnant and lactating women studied here because that's a huge piece of our population. So how do we do it to make sure that those people
Thank you. Oh, there we go. Oh, it's actually working now. I think, yes. In terms of what we can get out of the ongoing studies, I think we need to take transmission off the table. That needs to be a separate study. The two issues I think that we can derive information about are the potential of asymptomatic infection. Because if we actually are inducing sterilizing immunity, that's good. But if all we're doing is converting mild infections to asymptomatic, that's good. But it's not as good because there may be still ongoing transmission. But the other more important thing to me is duration. That's the one issue that I'm most concerned about with very, very limited data. The blinded crossover would allow us to continue to collect duration data, which I think is very important, but it's not going to permit asymptomatic infection data to be accumulated. So we would lose that. And so I would, if there's going to be a quote pseudo unblinding, the blinded crossover, I think would be the way to go.
Thank you. Next is Dr. Moore.
There's one question I have, or it's not a question, it's a comment, and I don't really have an answer for it. But with 2 large vaccine trials that are now currently blinded, and they're ongoing, the vaccines are shortly going to be released publicly in some way, or at least Pfizer has been released. That suggests that there are going to be some people that are blinded in, for example, the Moderna study, who are in the study because they're personally tremendously afraid of getting COVID. And they may move over to get vaccinated. And if they've already been vaccinated, then we have a risk of over-vaccination and also adverse events occurring that we don't recognize are actually due to the fact that people are not being vaccinated according to the protocols that we have. I don't have an answer to that. I don't have an answer as to whether it's better to unblind or blind to address that question. But the other point is that I do disagree with Dr. Perla. I do think that transmission is actually perhaps the most central thing that we need to address as of right now in this epidemic, and to try and get our best handle on that probably is not the or the nucleocapsid antibody, but rather direct detection of nucleic acid. So that's one reason why I'm pushing for repeated NP swabs. More importantly than that, perhaps, is even if we don't have an answer as to whether these vaccines do limit transmission, is that I would hope that both Moderna and Pfizer would work with public health officials to try and establish with their well-defined cohorts. For example, are there protocols for ring of vaccination that we could use that will work or have the best chance of working? Because ultimately we anticipate that by next summer we will have a low rate of transmission, and then we will be putting out fires, and we need to know how to put out those fires with these vaccines if they do interrupt transmission.
Okay, Dr.
Arnold, can I just respond real quickly to Patrick's comment? I didn't mean that transmission isn't important. I simply meant that I don't think you can get it out of this trial design.
I understand. Having done a lot of observational studies on transmission, I tend to agree with you. It's a very difficult thing to study unless you're studying that subject. Dr. Cohen.
Sorry, apologies about that. I just wanted to add to Dr. Fuller and others' comments that I agree that Moderna's plan sounds reasonable, and especially given the logistical challenges that a study sponsor would potentially face in terms of when a particular individual in the study becomes eligible. I think given the variability that will happen at the state and local level in those criteria, it would be hard for them to implement that across the board. And I also believe that given the large number of observational studies that are being implemented in combination with multiple different groups, that some of these questions, while it's not perfect, while a clinical trial blinded would be ideal, I think that if you can look at some of these questions from a multitude of other observational studies, we will be able to understand, we'll be able to answer some of these questions to a similar degree of confidence.
Dr. Offit.
Right. Just to get to Dr. Krilla's point, there is an ongoing trial that is being planned for early next year on college campuses where people will be vaccinated or not, and then those that are vaccinated will be followed to see to what extent they're contagious by doing, extensive contact tracing, which is really the best way to do it, as Dr. Monto alluded to, and then look at these sort of, the nasopharyngeal secretions to see if you can eventually have a biomarker for what that contagiousness is. But that is being planned and apparently it's being funded. So, good news.
Dr. Moore. Again, let's try to get a sense of the committee about the unblinding rat crossover issues.
I didn't hang up I didn't have a question, sorry.
Oh, you didn't? You're still there. Okay. Dr. Kurgan.
Yeah, so I think I really like Dr. Cohen's comment. I definitely like the idea of continuing the blinding portion in the crossover design because of the advantages it gives you in terms of following placebo individuals. But I think the realistic piece of this and the challenges that will entail for the differential groups in terms of when they will get access to vaccine will make this really difficult to do. And I worry in terms of different states and their approaches to this that will be difficult. So I was leaning towards the side of who would be doing blinding because that would provide some real advantages. But I think in some ways the realistic aspect of this really makes this going to be difficult. So it may be impossible to approach that side. So I think in an ideal world, I think we'd like to keep the blinded portion of the crossover design, but I think the reality of what's happening may make that too difficult to do.
Thank you, Dr. McGinnis.
Thank you. I am in favor of the blinded crossover approach. I think it's powerful, and I think we may have a little bit more time than we actually think. I could imagine a scenario where you could articulate the priorities, and it could be even on a state level. I don't think there's going to be this much vaccine floating around for a few weeks. So even though people may want to walk and get in the queue to get an EU, I'm just not sure what the supply is going to look like, and you may have a little bit more time than we think. So I think in principle, I like the blinded crossover. I think it's powerful, maybe the best we can get in terms of being able to continue to assess safety. I think the crossover could be tailored to a particular geographic area. I'm not saying it's easy to do, but I would entertain it. And my third point is we've been talking about pregnancy registries, and I just want to iterate that what I think we're talking about is a pregnancy exposure. We're not actually proposing a pregnancy registry, but for exposure of FDA-regulated products. So those are my 3 points. Thank you.
Thank you. Dr. Taubman has a suggestion for us.
Thank you. Right, so I'd give a suggestion for a framework of discussing this, but I think that we've addressed the ethical issue. There's really no ethical issue with with having to unblind these folks. They don't have to be. So the issue is really, I think, boiling down to what's practical, what's workable. And I guess that when people say it's not feasible,
not in priority groups in the blinded study, you're saying that what Pfizer is doing is completely impractical. Because that's what they're doing. But what they've told all of their folks, at least in my state, I assume it's the same way everybody got, is that the vaccine transition option is a voluntary process that offers all participants 16 and older in the placebo group an option to transition to the vaccine group. Interested participants can transition at 2 time points. To determine the order in which participants can begin the vaccine transition option, Pfizer and BioNTech are following the guidance of the U.S. Center for Disease Control Advisory Committee for Immunization Practices, ACIP, which has prioritized healthcare workers for direct patient contact. Now, there's also commentary that we got. There's 148 of current trial participants who specifically recommend. They're fine with saying that as a vaccine developer achieves EUA, it should be permitted and indeed encouraged to unblind members of its placebo arms who would naturally qualify for vaccination under their state vaccine distribution plan. Dr. Cohen pointed out this variance, and I understand that, but all we need is a few more weeks. I mean, if we just can get a few more weeks of data by maintaining placebo control for those who are not in the priority groups, and that will be, in this case, Moderna, it's 25% will go out as healthcare workers, then we gain a lot. So it is feasible, or if you're saying it's infeasible, you're saying that what Pfizer is doing is not feasible. And I think the last point here, and Dr. Goodman explained this, there's a real reason to have uniformity here between the different sponsors. And since Pfizer's doing this, there's no reason, there's no ethical problem with having Moderna follow the exact same practice protocol. So my suggestion would be that we recommend that Moderna do what Pfizer's doing because it is feasible for a period of time, just a few weeks, which would be really helpful. And then the secondary thing would be support for the blinded crossover.
Okay, I've been asked by committee members if we are going to have a vote on this. My sense, Marion, from what you told us is that you would rather we did not and just give you the sense of the committee. Am I correct?
Yeah, you're correct. And I think, I really thank the committee for being very clear here over the last couple of minutes to really, speak out on their preference. It is complicated, and I was trying to sort of keep the tally a little bit here on what I was hearing. And
so was I, and having great difficulty. Yes, because there are nuances.
That's right. I feel that I'm speaking here for the Office of Vaccines, but at the same time, I have not had a chance to confirm it with my colleagues. So if you could continue the discussion for a bit longer, because I don't think that all the committee members really opined here, and I would like to take a minute to get some responses, because I asked the question of my committee members, of my people here, to weigh in with their opinions on this as well. So if you could spend maybe a couple of more minutes discussing this very important question.
Right, Marion, we'll talk among ourselves about this, but I just want you to think about, if we do have a voting question, what that would be, because I'm not clear. This is not a black and white issue.
Yeah, I know.
And I'm not clear what the vote would be about. So please, if this is going to be a voting question, let's have a clear question, because I'm not sure. We don't want a lot of abstentions and things like that. We defeat the purpose.
Well, this is why we tried to forward this.
Right, I understand. That's why you didn't want to vote in the first place, because it's so difficult. Could I ask all the hands to be lowered, and those people who have not spoken on this question, because that's what we're hearing, please try to tell us what they would think about it. I see Dr. Hildreth.
Dr. Montel, you invited me to comment?
Yes, please, Dr. Hildreth. If you've got a comment and an opinion, we're looking for opinions. Opinions are usually pretty cheap, so let's get them from everybody.
Sure. I want to express my strong support for the plan that is outlined by Dr. Beatty to have an open label crossover. We can still get a lot of information about safety. And as a matter of fact, I totally agree with him that the participants who got the placebo should not be disadvantaged because after all, we are still under a national health crisis, and the whole point of this was to get a vaccine that could be used to slow down COVID-19. So I have a strong opinion that it might even be unethical for us not to offer the vaccine to the placebo recipients. And I agree with him that
this would be right. This would be right now or when their priority group comes up, if that's feasible.
For me, it would be okay either way. When their group comes up, they should be given the opportunity to get the vaccine. I just really feel strongly if we don't do that, we're going to lose the placebo participants and maybe do harm for future recruitment of vaccine trials. So I just think that I agree with his plan for an open label crossover, and that's what I would recommend to the FDA. Thank you.
Dr. Sawyer.
So the point was brought up earlier that the blind is already going to be severely eroded by the local and systemic side effects of the vaccine. And I think now that that information is being widely publicized in the media, people are really going to figure out whether they got vaccine or placebo. If you got 2 injections and each time your arm hurt and you got malaise the next day, you're going to figure out that you got the vaccine. So I think behaviors are going to be modified based on that. And so my opinion is that blind is already eroded to the point where it probably won't matter. So I'm going to support the crossover approach. And I prefer the crossover approach to allow people to be vaccinated when their tier
That's
comes.
blinded. The crossover is blinded.
No, I'm supporting non-blinded
crossover. You're supporting it, an open label.
Open label crossover, but when their people come up in their tier.
Okay. Dr. Wharton.
So, since I didn't really specifically address this point when I spoke earlier, I wanted to say that although the blinded crossover seems really powerful and has a lot of and seems very valuable, right now healthcare workers being vaccinated in many different parts of the country, and to ask the 24% of healthcare workers in the placebo group to go unvaccinated while a blinded crossover change in the protocol is implemented really doesn't seem feasible to me. And it is preferable that people be kept in the study, and that can best be done by offering vaccination in the appropriate tiers as they come up, and additional data can be collected on those vaccinated persons as the study continues. So that would be my suggestion.
Thank you, Dr. Rubin.
I'm going to echo Dr. Wharton, but I wanted to go a little bit farther, saying that the open label study seems like the only choice, but it's not a terribly good choice. So I think it's better to keep them in a study, but for future sponsors and for future trials, you can derive a lot more information out of the crossover design, particularly around AEs. And that's what I think we would learn a lot more about. So I would favor that in the future, but I'm supportive of an open label trial now.
I agree, and the problem is we're dealing with an unprecedented situation, and there are a few things that people didn't think about going in. Dr. Sylvester.
Yeah, thank you, Dr. Monto. I agree with what Dr. Rubin just said. I think that it's not a perfect world. The open label makes sense at this point in time, and maybe in the future we ought to be thinking about the crossover that's blinded. I'm worried also that with a greater than 90% vaccine efficacy, will people enroll in future vaccine trials knowing that they're not going to be able to get it? So I think the inevitability towards the crossover makes sense, and let's work on this one at this point. So I'm in favor of open.
Dr. Meisner.
Thank you.
Any further comments? I know what you said before.
Yeah, and after listening to this fascinating discussion, it's very hard to reach a conclusion. I will just say that this will be, if we don't do the blinded crossover, this will be the last opportunity, because once the vaccine is licensed, no more placebo-controlled trials. So we will be throwing out that opportunity. Now,
if I could interrupt, I think that's one of the reasons we have question or discussion item number 2. What in the world do we do to collect in the future placebo-controlled data?
Yes, at least in the United States, that will be very
well, that brings up another question.
Yes, and also, it's going. What is this going to mean for the other vaccines when they start their or are already in their phase 3 trial? Will they follow the same regimen that Moderna and Pfizer follow, and there won't be the option of a blinded crossover? Because why would a subject participate in that trial if she or he could get an authorized vaccine? So, and I think that what Dr. Sawyer said is also true. And remember, anyone who wants can go out and get an antibody test and find out whether they got the vaccine or the placebo. So it's not
that true.
It's not a secret. And I think Dr. Cohn's comment about practicality is very important. So I would still prefer a blinded, randomized crossover, but it's also going to be very, very hard to do that. Over.
Thank you, Dr. Perlman.
Yeah, so the only thing I would agree with all the past discussion. I just wanted to give my opinion. I like the blinded crossover, but it sounds like it's not going to be feasible because of this ability for people to just walk into the vaccine, and particularly people who are in healthcare settings now. So if it could be instituted immediately, that would be one thing, but it doesn't sound like that's really going to happen. That's, I think, what Dr. Bateman was saying this morning, that it was logistically going to be very difficult to do that. So that's why the Moderna approach may be the best.
Dr. Kim.
I don't have any specific reason to add to all the discussions that have taken place already, but I just want to go on record in saying that I would support the open label.
When? Right away after the EUA or after the individual's priority group comes up? That's what Pfizer is doing. Okay, let's move on to Jim Neaton. Dr. Neaton.
Yeah, I prefer the priority-based unblinding. This morning, it was pointed out that there's nothing in the consent about that you get the vaccine once the study's over with if you're in the placebo group and it's effective. It's also, all consents have a requirement to explain the data to the people from the trial that you're in and its implications for them. That and the press that this trial and the Pfizer trial and the AstraZeneca trial have already received, I think makes it very difficult, plus the local circumstances of healthcare workers being vaccinated. So I think try to maintain the blind between the vaccine and placebo as long as possible. Try to keep the people in the cohort because you want to follow everybody for another 2 years. But in order to do that, the practicalities, I think, are such to do it in some type of a stage by priority kind of setting if people can structure it that way.
Dr. Schooley.
As much as I'd like to see things remain blinded as a scientist, I think from the practical perspective and from the perspective of the realities of vaccine availability and logistics, we need to realize that our trial participants are going to want to know what they were in. They're going to walk if they don't know, and I think it's really important to keep them in the trial. So I would support an unblinded crossover. I think we have to also, I think it's going to be complicated trying to understand when the vaccine is really going to be available in each location with the way our country works. So, and it will take some time to get the logistics of even the unblinded crossover set up in a synchronous way starting today. So I would favor going ahead and beginning to make those changes in the bureaucracy and then being ready to do it when it's in place in a synchronous way as best we can with the bureaucracy we have to deal with.
Dr. Cohen, and then the final word from Dr. Taubman before we look very briefly at the second point.
I'll just, just to clarify what I said earlier, I think you can very easily separate out the healthcare workers from the other groups, but there's not going to be some sort of this person is going to be eligible now. Health departments will be opening up vaccination for different groups more organically. So I think if you could vaccinate the healthcare workers now, and like Mr. Taubman said, and keep the blind for a majority of participants for several more weeks, I think if a participant believes they're in a group that is now being recommended for vaccination, the sponsor should not be policing that. Similarly to how I don't think health departments will necessarily be policing that. So that just clarifies my previous comment.
Dr. Lee?
So I would agree with the open label. Although normally I would suggest the prioritization, I would agree with Dr. Schooley that it's such a hodgepodge here, it's impractical. And the other consideration I think we need to keep in mind, and starting this as soon as possible, is they do have this drug supply that apparently they have available that they could use for this purpose, which has something of an expiration date. So, but I favor the open label crossover. Thank you.
And quickly, Dr. Chatterjee.
My comment, Dr. Mondo, was actually about the other study. It's not about this first.
Okay, why don't you wait for a minute while I recognize Dr. Taubman and you can kick off that discussion.
Thank you, Dr. Monta. By the way, it's not Dr. Taubman, it's just Mr.
It's Mr. Taubman. I keep trying, but we do it by a knee-jerk reaction.
Thank you. I just wanted to see if I have this right from just listening to folks. It sounds like there's some disagreement, but predominantly people are okay with open label, but I didn't hear anybody objecting to the prioritization, meaning that, yes, you unblind, but you do it when their group comes up. We just heard there's some various mistakes, and there's going to be some problems with it. And as Dr. Cohen pointed out, healthcare workers are a very clear group, and the other groups, when we get to them, are going to be not so clear. But Pfizer believes that that's doable. So we can at least try it. They can try it, and to the extent it doesn't work, it doesn't work. But in the meantime, if people aren't going to be able to access vaccine now anyway for a period of time, if they're not healthcare workers or nursing home residents, then we gain something by saying, yes, open label, but when their group comes up. And also, we avoid any ethical issues.
Dr. Gruber, do you have some comments?
Yeah, I just wanted to make a brief comment. I had conferred with some of my colleagues, and the consensus is, as I stated before, that we will keep this question as a discussion point, and it should not be voted on. Thank you.
Thank you. And Dr. Chatterjee, you had a comment to start us off on the what happens if we don't have clinical a placebo group. What other additional studies can be done?
Yes, thank you, Dr. Monto.
And we don't want any open discussion of all the observational studies to learn about how vaccines work, but focus on this current issue of the lack of a placebo group.
Okay, what I was going to talk about was actually additional studies, such as co-administration of other vaccines.
Okay, that's on the table. I didn't want to get into studies of transmission and things of that sort. Okay, please. But please go ahead.
If I should
continue. Please go ahead.
Yeah,
yes,
so
please.
other populations of these studies, I thought should include older adults, those who are 75 and above, because the numbers of participants in that group, I think, are relatively quite small. And then also residents of long-term care facilities, I'm not sure that those folks were included in these trials.
Thank you. Very helpful. Dr. Hildreth.
I'm here. I was going to agree. I agree with the previous comment that it would be nice to do some studies in people living in assisted living facilities, and that wasn't specifically part of this. And that's a crucial group that we need to have some data from. Thank you.
Dr. Pergam.
Thanks. I have to say I completely agree with Dr. Chatterjee that one of my concerns was the small number of elderly patients in the 75 and older. That's important. We need to expand on that. I also
let me just ask you, what would the design be? Because we can't do a placebo-controlled design.
Yeah, I think it's I would study it just as a potentially as immunogenicity alone. That might be sufficient. I mean, it's probably we can't do a placebo-controlled design, but I think you at least have the data from the primary trial to see what immunogenicity looks like between the two. And I think that's probably going to be the best you could do. I think that's also true in the immunosuppressed population, which I think is a really good, I know they are working on these trials, but I think that's going to be really important. There aren't going to be the ability to do placebo-controlled trials in that sense, and I think you'd have to look at immunogenicity as well. So you're looking at patients who necessarily can't produce a robust immune response and see, how much less of a response you get in those groups. I think those are going to be really important studies for the larger population, particularly since immunosuppressed patients will make up, 4 to 5 to even 6% of the entire U.S. population.
Thank you. Dr. Gans.
since we're talking about other studies, a couple of points that haven't been raised. I'm trying not to repeat things, but I know that there's a lot of overlap here. The other studies that we actually haven't talked a lot about are really looking at other conditions. We talked about Bell's palsy, but there's other neurologic outcomes that I really think have to be high on the list. And so we really need to consider, especially when we go down into the children studies. And so I want to urge those particular things to be looked at. The other part of it is the cardiac findings. I mean, we think most of this likely from the SARS-CoV-2 receptor there may be specific to the virus, but we haven't figured out if they're immunologic or not, and we're seeing a lot of different cardiac manifestations. So this needs to be studied not only just as potential outcomes of the disease versus the vaccine, but also in people with cardiac disease. So I think that's a really important piece to keep in the forefront as we're moving forward, one to think critically about. In terms of some of the studies that need to happen, again, we talked about some of the immunologic studies in children, I think are going to be particularly important because we can extrapolate, particularly studies that we've done in children where the T and B cells do not follow the same pattern as each other and as an adult because they have different maturation of particularly their T cell responses. And therefore, it's going to be really important as we understand this to really, I want to just reiterate, really doing those studies ongoing. Okay.
Thank you. And next, Dr. Sawyer.
One of the things we eventually need to learn is what happens if you get one dose of the Pfizer vaccine and then a second dose of the Moderna or vice versa. That mistake is going to happen a lot as we start to disseminate vaccine around the country. The interim guidance that's been issued so far to immunization registries is to not give a third dose. In other words, to assume that even though you're mixing products, you're going to get an adequate immune response. So at some point, it would be nice to know if that's really true.
Dr. Rubin.
I know that we're not supposed to propose other studies, but again, correlates of protection are going to become extremely important in interpreting a non-placebo. Okay, and correlates of protection are going to be really important in interpreting these trials because of the lack of placebo. We're going to have much more difficulty assessing safety, and there's no easy way to do that when we have no placebo control. But we can at least get at efficacy if we have some good idea about what protection looks like.
Dr. Lee.
I think one of the interesting things we might want to consider that does not require placebo control is a non-inferiority trial of 2 doses versus 1, because I think you're going to have a certain subset that doesn't get the second dose, and if you have reasonably good vaccine efficacy with 1 dose, then I think we really need to think about that. So just we would be looking at it for pretty much at the incidence of COVID-19 in the 2 groups, but I think the non-inferiority one would be really one to think about. Thank you.
And how about different doses? We heard some questions raised about the dose that was suggested or the timing of vaccination.
Especially for children, Donald.
Especially for children.
Yes.
I'm trying to get a discussion going. It's hard virtually. Dr. Kurilla, is that you?
Thank you, Arnold. Yeah, let me just echo Dr. Rubin's point about the correlates of protection. I think this is probably one of the most critical features, not just for this vaccine, but for future vaccine trials. It will really make other vaccines realistically approachable in terms of their clinical trials going forward. If we can move towards some sort of accelerated approval, it would the immunogenicity, I think, would be a very good endpoint in that work. The other thing in regards to the 1 versus 2 doses, I think that's an important trial, but I would also like to emphasize that in the follow-ups, that what's being done, the sort of surveillance for under the EUA, I think there needs to be some aggregated data to look at who only got 1 dose, because there are going to be people who are not going to come back for that second dose, and see whether there are any clues that may be quite informative without having to go through a formal trial to sort of get an assessment as to whether that's a feasible approach.
Dr. Schooley.
I just was going to reemphasize the immunocompromised patient population, not just because we need the data, but because they also are a place where we can have a wider splay of immune correlates to look at, and might get some correlates immunity data in a relatively short period of time if they are starting at a lower point in terms of their vaccine-induced immunity.
Right. Immune correlates are a recurring theme, and it looks like we may be blessed with an immune correlate here with this vaccine, which we haven't seen with other vaccines. And clearly that's a message that immune correlates are paramount. Dr. Gantz.
Thank you. I just wanted to follow up on a thought. I kind of mentioned it in my last comment. I think Dr. Offit had started out with this as we're investigating this disease further, and we know that hopefully populations get immunized. One important component will be to look at the adverse events as they follow natural disease versus the adverse events that follow vaccination, because as we all know, vaccination is highly protective, although often not 100%, but as long as they reduce the actual events of the severe adverse events, then actually that should be a issue of protection that is studied ongoing. So for instance, developed policy and other of the outcomes that we've seen. Thank you.
Dr. Sylvester.
Yeah, Dr. Monto, you raised an interesting question about the timing between the doses. And I think that there have been some interesting studies that we've seen in the vaccine world where the longer you wait for your second dose, the higher your antibody levels may be. I think the practicality of that in a pandemic may be difficult. I think people are going to want to line up, and Pfizer's got a 3-week window, and Moderna now has a 4-week window. I don't think many people will say, I'll just wait 8 weeks or 12 weeks before I get my second dose. So I like the question, and it's a great academic question. I'm not sure in a pandemic it's a practical one.
Dr. Gantz? No, Dr. Perga.
Thanks. Arnold, the dose issue that you brought up is a really important one that I want to come back to because, again, thinking about populations that tend to have less response to or less side effects, it looked like the older population had less complications from the second dose of the vaccine, which might suggest they could tolerate a higher dose. And we've seen in other vaccines that higher doses are more beneficial for, whether it's zoster, whether it's influenza, could be beneficial. And so it could be a real value in targeting those populations with maybe a slight difference in immunity, either the immunocompromised population or the older population as targets to do studies looking at higher dose. And I know Moderna had the 250 was the highest dose, or I think that's right, would be at least an option to try and see if there was better outcomes in immunity from the higher dose of the vaccine in those groups.
Thank you, Dr. Perlman, and then finally Dr. Fuller before we go on to the voting question.
Yeah, so I just wanted to reinforce the idea of doing a pediatric trial, and also pointing out the problems because children don't get particularly sick with this. So it'll be very important to think about whether we're going to measure serial serology, serial culturing, and for little children this will be very hard, but I think this is really important because this may be the major group that's unvaccinated in a short time.
Okay, and Dr. Fuller, final comment before we discuss the voting question.
Yes, thank you, Dr. Monto. So looking really far ahead, a couple of questions which have to do with duration of protection. What will happen if this vaccine isn't a lifelong vaccine, which we expect that it is not? So how will we know if somebody needs a boost, or how will we know if they're protected against new strains that may evolve from coronavirus? And I know that's not an easy study to design, but I just want to put it out there because if we really want to coexist with this virus or variations thereof, we need to be thinking about those sorts of things.
So thank you all for this very vigorous discussion. A crossover blinded design, but the realization that that may be impossible. We know what Pfizer has proposed, and FDA will be negotiating with Moderna about the way they will address this problem. So I think we've really had the time, fortunately, to go over this in the kind of detail that it really needs. Now, we're going to have a discussion of the voting question. We will then have an electronic vote, and then I will ask the committee members who wish to explain their vote, don't need it from everybody, to explain their votes. So the question is based on the totality of scientific evidence. It's very carefully phrased. Based on the totality of scientific evidence available, do the benefits of the Moderna COVID-19 vaccine outweigh its risks for use in individuals 18 years of age and older? So, hands up for commenting as you wish on this question. Dr. Gantz, you were the first.
Thank you. Thank you for allowing us to opine about this really important topic. I think that this is a really opportune time for us to move science forward, and I would say that the evidence that has been studied in great detail on this vaccine is highly outweighs any of the issues that we've seen. And I think it really supports us being able to, with the pandemic in our background, really move forward and finally provide a safe and effective way to get to herd immunity. Again, understanding that this is for 18 years and older, and that obviously we need to be able to provide this to all of our population to get there, but it's a first step. Thank you.
Thank you, Dr. Kurla.
Thank you, Dr. Monto. Yeah, I have some serious reservations about this question because we've been discussing this whole meeting has been focused on the emergency use authorization for this vaccine, not for full approval under a BLA. And the question really doesn't reflect that. It could easily be seen as full approval. There's quite a bit of confusion, I think, not only in the general public, but many in the media reports of last week and this week talk about this panel approving the vaccine or recommending this vaccine for approval. And we even heard today during the open public hearing session, several medical professionals who talked about approving. I think that the distinction between an EUA product, which is still an investigational product, and the full approval of a product under full approval with a BLA is a distinction we need to maintain, and I think we're losing that simply by looking at this as an age-related. Anyone over the 18 years of age and older, it doesn't strike me as really addressing the emergency, which is severe and serious, life-threatening COVID disease in specific populations. So I have a lot of problems because this could be interpreted as us actually recommending full approval of the vaccine, and in the minds of the general public, that may happen and may preclude adequate, not only adequate evaluation of this vaccine, but other future and ongoing COVID vaccines in development.
Thank you. I appreciate your concern. I wonder if Dr. Gruber could address amending because the emergency use authorization, but the question doesn't really state that.
So when we published the agenda for this Unpack committee meeting, the topic is the committee will meet in open session to discuss emergency use authorization of the Moderna COVID-19 vaccine for the prevention of COVID-19 in individuals 18 years of age and older. So that is the topic of today's Unpack discussion. It is to discuss emergency use authorization. That's what the agenda says. We phrased this question the way we phrased it because, as was stated, under a vaccine authorized under an EUA is a product that has not been approved. It's a non-approved product. And under the EUA, in order for us to grant or issue an EUA, we have to make a determination that the benefits of the product outweigh its risk.
Do we just add, Marion, what if we just add the words EUA under an EUA to this voting question? Would that be possible?
Based on the totality of scientific evidence available, do the benefits of the Moderna COVID outweigh its risk
for use under an EUA in individuals?
For use under an EUA in individuals 18 years of age and older. We can
you have to take this to your lawyers, or
no,
can you make a determination?
We can do this. We can say for use under an EUA individuals 18 years of age and older. If the committee needs that clarification, then I think we can safely do so.
Metro, may I make a
yes. Yes, please, Dr. Hill.
The question is very clear. Do we think that this vaccine benefits outweigh the risk? If we think that, then the FDA will make a decision as to whether or not to issue an EUA. That's not what we're voting on. We're voting on whether the benefits of this vaccine outweigh the risk. And then it's up to the FDA to make a decision as to whether or not to issue an EUA. So I think the question should be left exactly as it is.
I completely support Dr. Hildreth.
Ditto.
Dr.
Okay.
Monto?
Yes,
Dr.
please.
Pretty much. And Dr. Gruber, I have a little trouble with it the way it's written also, because it's going to be very hard to study other vaccines, experimental vaccines, when a person looks at the sentence. And what I would suggest is that we write through 2 months of follow-up, or put some qualification in there that defines the length of time that it's been evaluated, because this is a blanket statement that everybody over 18 years of age should get it.
No, this is the question that is phrased the way it's phrased, because we want to know if under EOA, where the vaccine, the product is still considered a non-approved product, but could be given during a public health emergency, if the benefits of this product outweigh the risk. It does not imply that it's under an EOA then, of course. If we determine that the benefits outweigh the risk, under the authorization, under an EOA authorization, it can be given then to individuals 18 years of age and older. But that does not equal that the product is approved.
But a lot of people won't understand that they could you say at least the experimental vaccine.
No. I mean, no. I think once we start qualifying in terms of the duration or anything like that, it's going to be so confusing because the duration may get longer as we go forward.
Arnold, let me offer an alternative. Marion, instead of an age, what about the people at risk for serious COVID disease?
No. And let me just say that we have a question now. We are advisory to FDA. They have put in a question that they feel comfortable with. Am I correct, Marion? And that is what we are voting on. If the vote is not in favor, then we can discuss this further. Marion, how should we proceed?
I would like to proceed with keeping the voting question as currently phrased.
Okay, and there it is.
Dr. Michael, may I ask a question about the phrasing?
Excuse me? Dr. Hildreth? Yes.
Are we going to go back and retrospectively change the question we voted on for Pfizer?
Well, that's another issue I was thinking of. How would I explain that we have a different question?
Yeah, how would we explain that?
Yes, I get it. I get that.
Dr. Monson, may I ask a question that's not related to the phrasing, but a very important one to the question?
Yes, please, Dr. Fuller. And then we'll try to go by in order. It's a lot easier to manage.
Thank you. Dr. Gruber, I definitely hope this does not happen. But what if there is some adverse event that appears that is very broad, that this is if we think the benefits outweigh the risks, but it turns out the risks are so high, how does this EUA get withdrawn? What would be the conditions to say that we can no longer do this?
So, as Dr. Fink had elaborated on in his introductory remarks, an EOA, and he did say this last week, and I believe he said it today too, can be revoked, and there can be several reasons. The one could certainly be if we see that the risks outweigh the benefits of that product, then we can revoke the EOA. So that is, but that's right now we're voting on the data, or we're looking at benefit and risk based on the data available to us, and as we have presented them today. And we, of course, as was stated, we will have continued follow-up, active safety follow-up of the recipients of this vaccine under an EOA. And if we determine that the risks are no longer acceptable, and that the risks outweigh the benefits, then we can revoke the EOA, Dr. Fuller.
And FDA would do that?
And the FDA would do that.
Yes. Okay, thank you. Thank you for the clarification.
Dr. Robbins, let's just go by recognized individuals I recognize.
Thank you. Yeah, I disagree with Dr. Meisner. I think the question that's being asked of is, do we have enough evidence in hand to say that the benefits of this vaccine outweigh what at the moment, as far as severe safety issues or theoretical risks? I think the answer to that question is clearly yes. I mean, the question is never when do you know everything, it's when do you know enough. You know, we have trials of 44,000 and 30,000. That's as big as any general pediatric vaccine trial. The difference is length of follow-up. So we don't know whether or not it's going to be effective 6 months from now or a year from now, but there are systems in place to know that. We don't know whether or not it's going to have a rare serious side effect, which is true of any medical product, but there are systems in place to know that. And frankly, given what we know so far about the height of the immune response, about what we have with, you know, with T helper cell and cytotoxic T cell response and so forth, we can feel pretty comfortable that this vaccine is going to have a benefit that lasts for more than the 3 months or so that we studied it. I think it's a pretty easy answer. You can't qualify things as being experimental because you could always say that about any medical product. I mean, when the HPV vaccine came out, we could say, well, we think that that it's okay for 7 years because that's all we have data for it. So I think the answer to this question, at least as far as I'm concerned, I completely agree with Dr. Gans, is clearly yes. Thank you.
Dr. Cohen, and we're going to be discussing this until 5 o'clock Eastern, and at that time we're going to put it to the vote because there is also another chance to explain your vote after this. That's Dr. Cohen. I see it. Go ahead.
I would just ditto to what Dr. Austin said. I completely agree that the question is the right question, and the data clearly suggests that the data clearly shows that the benefits outweigh the risk.
Okay. Dr. Bergam.
Thanks. I completely agree with Dr. Offit and Dr. Gans. I think the preponderance of data is totally in support of moving this forward. I don't see any value in changing the terminology of this particular voting question. I also think this idea that the EUA process is going to change future vaccine trials, et cetera, feels a little bit strange to me. We're talking about a pandemic, which is not very common, where we really need to move this forward, and there's really an effort to get this done quickly. I don't see as much of a risk in the long term that this process is going to be used on a regular basis for other vaccine trials. So I think we need to focus on what's at hand and focus on the question here. And I think there's no doubt in my mind that the data it looks like the benefits outweigh the risk from what I've seen.
Mr. Tubman.
Yes, my camera's not coming on, but can you hear me?
Yeah, we can hear you.
I'm fine with the question as is because it is based on the totality of scientific evidence available, meaning that's what we have today. And based on that, unbalanced, strong data, that is very unclear disease. I think the balance supports it. I did have a concern, though, I'm glad there's discussion about whether to change the question or not, because I was troubled by the fact that the FDA was weighing in again on what the question was. I was changing the question. And basically, we've been told it's an independent committee, and we want to be transparent. If the committee feels that a question should be changed, the committee should change it. There doesn't seem to be willingness to accept the committee's decision. And the answer Dr. Gruber gave us, well, first go to my question, and then after that, if it doesn't pass, we can do a different question. That, in reality, doesn't work because almost nobody's going to want to vote no to this question as written. You deprive people the opportunity. And this happened last time, not to bring up the whole story, but with Pfizer, there was strong feelings about including 16 and 17-year-olds. And because that was not presented as a separate question, which it should have been, people were sort of forced to have to make a choice. But I think the committee really should be independent and decide for themselves whether the question is acceptable or not. In this case, I think it's not.
Dr. Meisner.
I didn't really.
didn't realize my hand was still up. So
okay, well, thank you.
No,
I'll let you have some burning thing to say.
No, what I want you to say, Carl, is that I agree with what everyone has said, and I am in favor of yes on this question. My only point is I don't want people to interpret this the same way they would a licensed vaccine. It is, as has been stated, based on the available evidence, but that's limited. But if everyone else is comfortable with it, I'm fully comfortable.
And in reality, Cody, whatever we say, the media is going to say, interpret it in whatever way they want. So,
yes.
Dr. Wharton.
So the question is written seems to be aligned with how we think about EUA, and based on the totality of scientific evidence available, I strongly support that the benefits of the vaccine outweigh its risk for use in individuals 18 years of age.
Okay. Dr. Meaden?
Question. The answer is yes to the question. Thank you.
Okay, well, okay, then you don't have to explain your vote afterwards. So.
All right.
Dr. Chatterjee, and then finally Dr. Rubin, and then we will vote the question.
Thank you, Dr. Monto. I just wanted to follow up on several of the previous committee members that commented on this, and I understand the difficulty that some people are having with the wording. Perhaps, as scientists, we tend to be very precise in what we say, and we want it to be as to the point as possible. But I think what is not mentioned in the question, and of course what we are all talking about, is that we're making this decision during a pandemic. And so it is this really unique circumstance that is forcing us in some ways to word the question in this way and to answer the question in this way. So I would say I'm comfortable with the way the question is written and willing to vote on it.
Thank you. And finally, Dr. Rubin.
Thank you. You can hear me?
Yes, we can. And see you too.
Thank you. Thanks. I'm glad to be recognized. Just want to remember why we're here. We're here to provide, for two reasons that I can think of, to provide the FDA advice and to see advice that they want. So we wouldn't get so hung up on the question because they make decisions, we don't. And the second reason we're here is to inspire confidence in the public that we've looked carefully at the data. And I think when we worry about the details of the wording, I'm not sure that we're helping people understand that what I almost certainly will be a very strong vote in favor is just a strong vote in favor.
Thank you, Dr. Rubin. It's a delight to come to the end when I don't see any hands raised, which was not the case last week. So now let's call the question. So we are going to be voting, and then after the vote, those who wish to explain the vote will have a chance to do so by raising their hands.
Thank you, Dr. Monto. Can everybody hear me okay?
Yes, we can.
Okay. So our members and temporary voting members, as seen on the next slide, excluding the industry representative, will be voting in today's meeting. And In regards to the voting process, Dr. Monto will read the question for the record, and afterwards, all members and temporary voting members will cast their vote by selecting one of the voting options. These include yes, no, or abstain. You will have 2 minutes to cast your vote after the question has been read. Once all the votes have been placed, we will broadcast the results and read the individual votes aloud for the record. Please note that once you cast your vote, you can change your vote within the 2-minute time frame. However, once the vote has closed, all votes will be considered final. Does anybody have any questions related to the voting process before we begin? Okay, I don't see any questions. So Dr. Monto, if you'd like to go ahead and read the question.
The question that we are voting on is based on the totality of scientific evidence available. Do the benefits of the Moderna COVID-19 vaccine outweigh its risks for use in individuals 18 years of age and older?
Thank you. So members and temporary voting members, you have 2 minutes to go ahead and cast your vote.
Arnold, this is Mark Sawyer. I want to point out that my vote says Pfizer BioNTech, not Moderna.
Right. This is the wrong vote.
You are right. I was busy trying to find where the voting was. Can we read?
Yes, we will be taking a revote. Just 1 moment.
Can we just revote last week's vote?
Well, we might get a different conclusion. Got the right one up now.
Yes, I believe the question has been updated. So we will restart the timer and clear out the current results so we can take another revote. We have 30 seconds remaining. Okay, our 2 minutes is up. So if we could go ahead and close the vote and broadcast the results. And I will read the individual votes aloud for the record. So Dr. Cohn, we have a yes vote. Dr. Sawyer voted yes. Dr. Rubin, yes. Dr. Carrillo abstain. Dr. Perlman, yes. Dr. Schooley, yes. Dr. Gans, yes. Dr. Lee, yes. Dr. Moore, yes. Dr. Chatterjee, yes. Dr. Meisner, yes. Dr. Fuller, yes. Dr. Hildreth, yes. Dr. Neaton, yes. Dr. Offit, yes. Dr. Wharton, yes. Dr. Kim, yes. Dr. Pergam, yes. Dr. McInnes, yes. Dr. Monto, yes. Mr. Taubman, yes. And that concludes the vote. It looks like we have a favorable vote. So I will pass the floor back to Dr. Monto. Thank you, everybody, for putting in your votes today.
Thank you. Now, anybody who would like to explain their vote should raise their hand. Mr. Taubman is first.
Thank you. I voted yes because the balance is strong. Last time, can you hear me?
We can.
Balance is strong for approval, so that's the way I voted. Last time, I did recommend that we not grant EUA broadly, but rather limit it to priority groups to allow for further data to be collected. And since there is limited supply anyway, and that would be to preserve the data we would get going forward. That was not accepted by folks, but we were assured that when Pfizer moved forward, people who were not in priority groups would be maintained in the study, and that was really important to me. I'm very concerned about Moderna's proposal, and it does sound like from the discussion, I know FDA did not want to vote on that. I can see why, but it seemed like there was strong support for if they're going to unblind, they should do it on the basis of when a group comes up in its priority, and not unblind everyone right away, which is what Moderna has proposed. I think that would be really disturbing. Finally, I did want to say thank you to the FDA folks, because they put a tremendous amount of work into this. I think Dr. Meiser said this at the beginning of the meeting. There's very long hours reviewing the data, understanding this, working with the sponsors. They've been under tremendous pressure here, and even they've been under improper political pressure, even bullying and threats. And I think they've valiantly resisted that and showed that science is going to prevail here. So big set of gratitude to the hardworking FDA folks, Dr. Marks, on now.
Thank you, Mr. Taubman. Dr. Fuller, you'd like to explain your vote?
Yes, I would. Thank you. First of all, I want to thank the FDA for the incredible work they've done, and this committee as well for the transparency that went into today's schedule and having more time. We're in an unparalleled crisis. I did not think an EUA was the way to go, but since the train has left the station, I appreciate that Moderna has given us a very transparent and thorough study that even from the beginning seemed to be very well organized with getting people with underlying conditions, with monitoring activity throughout the study, with even including the serology and nasal swabs, which are not completely analyzed at the moment, but which have great potential to look at important aspects. And then lastly, the care for the study participants throughout, including a plan for monitoring adverse effects, as well as what to do with people who now may want to move in from the placebo. So I appreciate the way that they've conducted a much more transparent and clean study. And lastly, I know that now that we have vaccines available, that we still have to use the preventions that are available such that we can keep each other safe as we go through getting to the type of protection however long it lasts. So I want to thank FDA and all of you for helping with this discussion today. And that's why I said yes. I didn't feel that way last time. Thank you.
Thank you, Dr. Fuller, Dr. Kurilla, and then for the final word, Dr. Hildreth.
Yeah, thank you, Arnold. Camera not working again. I abstain because I'm very uncomfortable with the language. I think in the midst of a pandemic and with limited vaccine supply available, a blanket statement for individuals 18 years of older and older is just too broad. I'm not convinced that for all of those age groups, the benefits do actually outweigh the risks, and I would prefer to see it more targeted towards people at high risk of serious and life-threatening COVID disease. And we have that; they have that information, and we understand to a certain extent those high-risk groups. So it could be targeted. Lastly, I would have preferred to have seen, rather than an emergency use authorization route, an expanded access program. I think it would have given us a lot more opportunities to continue to collect the data. And my concern about future vaccines was not on non-COVID vaccines, but other COVID vaccine candidates that are in various stages of development. Thank you.
Dr. Hildreth.
Thank you, Dr. Monto. Sorry about the train. I just want to make the point with what the remarkable scientific achievement this is, and say thanks to all the scientists present and past who contributed to this. To go from having a sequence of a virus in January to having 2 vaccines available in December is a remarkable achievement. And I just want to say that. And congratulations all those who have. Thank you.
Thank you, Dr. Hildreth. You've echoed my feeling about what a remarkable achievement has been reached here, having the sequence less than a year ago. I just wanted to make 1 or 2 comments before closing. Our vote was even more overwhelming tonight than last week. I don't think that anyone should interpret the difference in the vote being one way or another comparing the 2 vaccines that we have considered. Academics have a way of getting involved in details, and what we have done for the last 8 or 9 hours was to go over the details. And some people took the issues last week, especially those involving different age groups, the 16 and 17-year-olds, to drive the decision that they made, which clearly was made based on that issue and not on the overwhelming evidence for risk being less than benefit, clear benefits with these vaccines. So I'd just like to close by thanking the committee members, thanking FDA for giving us an agenda which allowed much more open discussion, which I think benefits all of us, including trying to advise FDA on some of these very tough issues that we are facing. And congratulations to us all for achieving this emergency use authorization for a second vaccine, which, along with other events, will eventually and sooner, we hope, break the back of the pandemic. Now I'd like to hand the floor over to Dr. Atreya to formally close the meeting.
Dr. Atreya, your phone's muted.
I'm sorry. Thank you all, Dr. Manto. We described my sentiments, and you all did a great job, and thank you for all your service and work. We greatly appreciate it. And then, so, yeah, I would formally close this meeting. This meeting is adjourned now. Thank you very much. Have a good evening.
Thank you. Good night to everybody.
Good night, Dr. Manto. Thank you.
Thank you, Dr. Manto.
Thank you all. And with that, we will officially adjourn.
All right. So I'm going to cut that off. Stop that. And let's get over here to, it's getting ready to make a big jump here. 1 second, guys. Let me get the chart up. Come on. Okay, here we go. All right. All right. Yeah, so we got the news. We got what we wanted. And it's like we discussed as always. When there's a share the, what they call a sell the news event, well, the end result is what you get. You had people who rode the momentum today. Again, stock came from 137.03, as you can see here clearly, went up as high as 147. So you had a 10-point move on somewhat information that was already priced into the asset, right? And so if you put that in the perspective in terms of the daily move, they're going to be people who going to take that bag. You know, how often do you get 10 points, right? And regardless of what it may do for the rest of the night, of course, we can go look at the chart and figure out where's the areas of support. It may ping or bounce around. Of course, you're going to get ready to hit some congestion around this area here in a minute, maybe even back down towards that 137. But clearly, you see here, you have congestion here around this point here, a little bit lower than where it's at now, of course. But right around that 137 price is a lot of pinging, right? You got some pinging here. You got pinging here, a little pinging around there. I mean, pretty much that whole area there, it's like a straight line of just pinging. So it's going to definitely find its way back, find its way down there. It's going to test that level. Of course, breaking through that will be significant because now you got a good chance to eat up into this area here, right? Now, if it hits that area and bounce, of course, that would be superlative for anybody who will scalp it tonight because they'll dip by the scalp of the first test of that point. And if it doesn't, blow right through it, they'll play for any retrace for about like a couple points, right? 137, 138, 139 potentially, maybe even back up towards, close by 139. But I mean, it's a lot of mass outflow. You could tell for the fact that the way that the candles are. Of course, let me do it like this, right? The size of those candles, that's the institution, right? Those are institutions. Those are not retail traders, as they would say, right? It's retail traders baked in there too because their stop losses are getting hit, right? Stop loss rate. This is old school, right? That's why they call it a sell the news event. So algos are trained to dump a large mass values of volumes of shares. Everybody that has a stop loss in play, that has what they call after hour, good till covered or cancel, sell orders for after hours as well. Their stuff getting triggered. It causes a domino effect. It's what we call like a bull squeeze, right? That's why people make more money shorting stocks than they do inherently obviously going long sometimes because again the money comes fast, it comes furious, and the dumps are violent, right? When they are violent, they tend to kind of cause that precipitous value where it seems like the sky's falling, right? Because you see your money turn over so fast, it's like, oh man, and you panic, right? Most people panic, right? Even if they are watching it and they're doing a hard stop, right? I mean, if they're watching themselves, they're going to sell when they feel comfortable, as opposed to trusting the machine itself, then that's what it would end up being. It would be a scenario where they're just like, you know what, I'm out of here, right? and they'll sell, and then it'll do like what it's doing now, the small bounce. You know, you get that kind of recoil bounce, right? That elasticity bounce, right? The rubber band effect bounce, and it'll go back up, like I said, around about 138, possibly 139. We'll see if we make it that far, but we just tapped 138. So you got back maybe about a point and a half off that bounce. Actually, there are 2 points if you think about it, because you know we were down here. So yeah, about close about 2 points on that bounce from 136, right? So now you're back at the 138. So that's a 2-point bounce. Of course, that's good money, right? If you scalp trading that, you like, bam, okay, I caught, you know, at or near the bottom. I'm gonna play this little dead cat bounce. Of course, in this case, that's what this was here clearly up here. This is a classic dead cat bounce. You hit here, you got a massive point of support, obviously close to that of a $0.50 mark, 140.50. You bounced, came back, tapped in this case the VWAP here. Clearly, somebody waiting to short it there even more. Again, this thing is an art, right? Smart money knows this. They're like, oh, you feel, get up here close to this VWAP, we're gonna short you even harder, right? And then, again, from there, the follow through was obviously even more devastating, because again, what a dead cat bounce does equal a greater value of that, at least of the previous candle where it came from in terms of up here near the resistance, rolled back over, broke that support, and then it deluged down about the same length to that of, from here to up around that point there, give or take, right? So we'll say about 3 points. So let's say in this case, there was a little bit more. But from 140 down to around about 147 and change, and of course even went down to 146. But clearly you can see that kind of that that level, I should say, excuse me. Right, you can see the level of the bounce. Like I said, there we go, 139, just like I said. Right, yep, there we go, 139. So again, you played the bounce or the scalp. You come in to eat now. Of course, getting back over 140 is going to be monumental for this stock, right? To clear 140, they're gonna bring it. They're gonna have to bring it, because and I'm saying close above it. See right there? See, the first thing it did, what? It went to 140, and then what? It instantly got rejected. It hit like 140 on the dot. y'all saw it, right? And then you see how it's getting rejected a little bit. It's a lot of big money sitting in that's shorting it. Obviously, at those whole dollar marks, like 140, like we said, up there, top of the VWAP. Right, so they're gonna test the bulls. Right, if they want it, if the bulls want it, right? Right now they're like, man, it's sold off, man. Look, if you want it, you're gonna have to bring it. Just take back all those new barriers that you had for support. Now they're becoming your resistance again. Money that's sitting on those places that's heavily shortened. You're gonna have to break through all that congestion to get the stock to move again. Right, so that's why you see it struggling hard. I mean, it hit that 140. And just got rejected like Matumbo. I mean, clearly there. Look at that line right there. Right? So they're going to make another attempt at it, because you can see the shares down here. Money coming in. As you can see there clearly, the outflow was obviously, obvious here. Now you have somewhat of somewhat of a rising wedge. It has to really break above these levels here to kind of, get it outside of that theoretical pattern aspect of it being a fugazi, bump up before it rolls back over. News because of what the catalyst is, right? This is black swan event type of catalyst, right? Which in this case, it could be an inverse or bad. It could be good too, because again, it spiked up initially, and instantly when I saw a spike the way it did like that, I was like, sell it. For anybody that might have been wanting to think about selling it, sell it, right? Take that profit, right? You got that's over, over 10 points, on the day, right? $10 per share. That's good money. That's what makes what happen. So here we go. See 140 side sitting there, hitting right at 140. That's literally hitting right at 140 and just sitting there. Look at there. It's like you're waiting on the blue line, right? The metro, right? I mean, look at that. There we go. See, now it's trying to flirt, trying to get over that 140. Now, the interesting thing here, not just because it breached above it, it has to close above it. Like, these are 1 minute candles, of course. But in order for that 1 minute, or that 140 level to be confirmed, it has to close above it, right? At least 5 1 minute candles. At least a minimum of 5 minute candles, 1 minute candles, or 1 5 minute candle. right? And so, now you're having a little extension, right? We'll see if it holds. But as of now, it's still a rising wedge. No matter how you look at this pattern here, it's a rising wedge. And it's a good scalp, like we described. We described it like this is going to be a rising wedge, all right? Rising wedge pattern off the lows. We call around about that 137. It came down a little bit lower, actually about 136. But that's the market. You can't time the exact bottom or the top. All we can do is play around those areas. We can obviously play the momentum because this is momentum right at the moment. The price action is very volatile. Clearly, a lot of red to a lot of green, right? And now you got this kind of rounded pattern here that's starting to happen in the volume where it's kind of the volume is thinning out, but the stock is still kind of right at the point where it's starting to plateau a little bit. You're going to need more volume to come in because if not, you see what's already starting to happen. It's starting to what? Arch over, arch over, right? If the bulls don't bring it, get above that 140. Clearly, you see the line here, this 20-day moving average here, this blue, light blue line. When we get above that, you're going to roll back over. Right? You're going to roll right back on over. And those who wrote this little scalp trade right here, you're going to like, you know what, I'm out, right? We got much as we can because they're trying to play anything back towards the VWAP. They have their sights on this VWAP here, it's 143, right? That would be ideal. So 136 to 143, that's a nice little scalp bag. But they're going to get, antsy like anybody else. And then the first person that sells a greater amount of shares, if they watch on level 2, they may follow suit like the domino effect, right? And again, the overarching pattern here is clearly, as you can see here, roll over. Signified by the candle there, which is the what? Look at that doji. That reversal signal, right? Clearly right here. Clearly what's going on there. Will it go all the way back down, quite down, straight down? It could meet and around a little bit here before it takes that. But these kind of things are just instrumental. We know these patterns. We've seen them like a gazillion times, right? We're trained for this, right? We know how to operate in or around these type of moves when they happen, right? When they happen. Right? So clearly here you have a moment where it's using the 9-moving average at this point, which is obviously not that bad of a deal. It's pinging off the 9-moving average, which is the purple line, far as a pseudo support, because it's clearly a pseudo support. With the way that this thing is volatile at this point, and you having a pinching effect here between the 20 and the 9-moving average. Now, if it holds enough and that 9-moving average creeps up a little bit more, if it creeps up a little bit more, if Bull's gonna fight tonight, they better fight right now, right? If that 9 cross over that 20, yeah, we might get a little bit extra more extension. Then then we can start looking at this candle here, far as the next target, in terms of how they would want to come and try to take this candle here out. Closing above that around about 141 would be really good momentum to try to at least close above the VWAP. Probability of closing back above the VWAP would be great. Now that actually will still be positive for this stock. Why would I say that? Because the first thing people are like, well, why the stock ain't at 145, 150, 160, whatever. Well, again, the stock moved over 10 points today, right? Over that. That is a lot of money. I mean, think about where it came from. Actually, it was lower than more than 10 point on organically, because again, even if you count the after hours, right? And the pre-market had it down to 133, and we called it. Remember we said that 133, 132 area last night. We was talking about that on the live stream, and clearly there it was. It came down, tested that area, and then it bounced, and then it went from there 133 all the way up to like what 147. All right, that's a lot of money, right? That's about 14 points from trough to peak, right? Or peak to trough in this case, but same way, trough to peak here. That's a lot of money, right? People gonna take those bags, man. You don't get those kind of moves every day in the market. They're happening in and around the market, but surely so to be in a position where you can take advantage of it because you have such a well-known catalyst in that of the Moderna news with the vaccine, that veil that was hanging over it before they did the vote. Of course, that was the suspense, right? It's buy the rumor. Let's buy this rumor up, right? Let's buy this speculation, right? Markets love that speculation, speculative value, and they bought it, hook, line, and sinker, right? Doesn't negate where, again, you still look at the chart overall. I mean, look at the chart of Moderna here. as egregious as it may feel like it is right now, if a person does, trying to go long, he was trying to go long right at the bell somewhere, just kind of take advantage of the spot. The stock still actually is bullish, right? Look at the chart. Look where it's still. I mean, you talking about from 95, 95, 96, all the way up to where it's at now, and it's hovering and pinging around the same point of contention we were just talking about earlier. Around this 137, 136 area seems to be somewhat solid support. Obviously could be taken out at any given moment, right? We saw it came down to pierce that 133 into the 132s, right? From last night until this morning's pre-market session, around that point there. But nonetheless, it was able to kind of regroup a little bit, and you kind of offered up this somewhat like a a semi cup and handle pattern here. It's very, very limited here though. And it's affected because of the fact that the catalyst that it's dealing with. So the volatility is kind of overshadowing the natural progression of how the stock would move. If it had not had to worry about, it being such a monumental news feed scenario with the vote. All right. So again, the bounce, like I said, it has to close above 140 for at least 1 5-minute candle or 5 1-minute candles. And then we'll see if it's going to make some moves, right? Because right now, the downside scalp is solid. Because again, it held, remember, like I say, if it's 9 curl up a little bit, Bull's going to try to stay in it. Of course, and they did. It didn't drop and plummet below that. So you didn't have to have the rollover right away. Bull showed up like we discussed. The volume thinned out there. That was the moment of truth. That's why you see it here. This little thinning of the volume. And I said the volume has to come in. Well, here you go. You got some nice little volume coming in there around about, I would say roughly about 70, 80,000 shares here. If you say 30, 10, whatever, somewhere around there, 10,000 shares there. You're talking around about 70,000 shares, whatever. That's a good little amount of volume to keep turning it after you having such a liquidation, right? Or a stop loss rate, right? So clearly here, it's trying to make a good little scalp comeback. Someone is eating real nice. Obviously not your brother, because I'm trying to do both, right? Talk and monitor other stuff. So I can't be in trade mode, which I could, but I'm not going to touch it. Like I said, I'll wait tomorrow to short it. If it's going to fall, if it's going to rock it, I'll let that what's already existing keep rocking. Of course, I have this in a long-term portfolio. I scalped the calls twice today, right? So we planned this from last night anyway. Or I should say we discussed this trade last night. And it did its job, right? It did exactly what we said it was going to do in terms of crossover that 140. 145 would be obviously gravy, right? Biscuit Wheels Gravy Valley hit that area and exceeded it. And those calls were like $1. We looked at them clearly last night. They were like $1 and change around, at the price at that point was around about 137 or whatever. And 136, 137. And so we said the 140 calls, if you buy those, or the 140, I think with the 140 to 145, they were like 100 and something, maybe 200 at most, somewhere around there. But anyway, those calls printed monsterly. I feel very monster value today. All right, very monster value today. Right. So that's the the good, the bad, the ugly when it comes to investing and trading. Of course, this is all about trying to shake investors out of their trades, right? Market makers have a mandate to collect and harvest, shares of stock, especially those who are very volatile, because that's how they make their money. Also, they parlay in between different clients. We're gonna take some from these little retail traders. They'll know what they're doing with these stocks, and they'll turn around, harvest them, and then a big institutional fund manager come in and buy them off them, right, off the market maker, right? And they'll obviously dump it back into the market. That the low hedge funds come in and just buy those dips. Right. So, So again, as you can see here, clear, like I said, if that 9 cross over that 20 moving average, you're gonna get an extra little bump. Clearly, there you see at this cross here, this crux moment here, you see a cross on the 9 moving average over the 20. It did exactly what we discussed in terms of one of the scenarios. Right now, it's fighting to stay obviously in the trend. This is good in terms of the small little micro pullback. You don't necessarily want to lose the 9 altogether if you don't have to, but you're still good because you still have a 20 here, right? Just coming right back to that same area if it comes down that low. If it bounced in around that area, of course, if it still holds 9, it's still fighting there. Then you probably still can kind of see if anybody else going to show up to the party, right? Because right now, those who are exhausting their funds to try to get this thing off the lows, they revenge trading or they averaging down. Well, that it can only last so long if you don't have no institutional traders trying to come in behind you, right? Or a big fund manager, or even a single little, Robinhood trader with a ton of margin. He didn't went all in, I can't take it. I'm gonna go all in. He went all in with 100,000 on leverage and trying to turn the stock, right? Then the stock will catch a little bounce off that, whatever. But yeah, so that's it. So that's the story of Moderna. Now, interesting because with that also being said, last time we saw the S&P 500, especially after the Pfizer news, it kind of moved around, bumped around a little bit, then it went on its own little merry way. Right now, S&P 500 looks pretty solid still. The S&P 500 looks pretty solid still, right? It's not dealing with what we're seeing happening with Moderna here, right? And see, there we go. See, like I said, if we don't take out that candle over there to the far left, that big guy there, it's gonna bang around or even come back down. So that point, that candle end up being a recordable force. It slapped Moderna, right? It said, no, not today. And so y'all gonna have to work a little harder in terms of that. When I say that, those who are adamantly trying to trade it after hours here, right? In order for it to catch some traction there, it's gonna have to fight a little bit. So it's right back down. So we lost at 140. The 9 again proved to be just a nice little day trader scalp off the lows, which was great because again, you're talking about what close about 5 points on that little on that bounce, right? It's good money. It's good money. So you play that all the way back up to that 141. Actually about 6 points off the 135, but that's good money, right? Take that any day, right? Take that any time, B, as they be saying, right? So that's the story of Moderna tonight. Again, awesome day of information. Obviously, the narrative of the information was more imperative to that of the actual stock. Even though I'm an investor trader, I have to say that because this is a serious scenario. But it's funny because what did those doctors say? Y'all heard it out of their own mouths that they were trying not to send the wrong signal to the media. Because they say, of course, the media going to make their own assumption anyway because they did with the Pfizer vaccine. They had the news all wrong. They was acting like it was approval, like a full FDA approval. No, it was just a recommendation for EUA. That's why they kept emphasizing the EUA, emergency use authorization. So as to not allow these, pumping publication websites and seeking alphas and all of them to print the wrong, data on ambiguous news, right? On not being specific, right? Or detail orientated. And so they knew that from last time. They said, we're gonna cover our backs this time. They ain't gonna just be eating off, again, Wall Street just want to make money. They don't care who, when, where, and why. So they specified that, and I think that had a lot to do with also the momentum kind of staying, somewhat in check. Right, even when it rolled out in the very initial beginning and went up near 147, right, and then it's like, oh, this thing topped out. Look, the RSI here. I said, this thing crazy. It's going to get sold. Right, they're gonna sell that top there. I mean, look at that. It's like a peak, man. It's like a very a lemon 2 lead pencil peak. After you just sharpen that thing real sharp. Right, that was clearly that it's gonna sell that, and boy, did they. They sold all the way down. Let it bounce. Let it roll down a little bit more and probably cover their short, position down here. Made a nice little bag. Again, over 10 points to the downside. Took all day to make 10 points to the upside. And in just a matter of, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, about 13, 14 minutes, it gave all that back. So you picked up $10 per share short in it in about 13, 14 minutes with the work. That's why people short sell. That's why people short sell markets. That know what they're doing, of course, right? Because you don't want to get caught up in FOMO and get your count, slain out there trying to, play those more strategic strategies and stuff like that. But again, it's just a learning. It's a good learning mode to be able to watch these stocks, how they perform under certain variables of news or events or whatever. It's really good for building up your mental prowess. As far as how most stocks react when we say we buy the rumor, sell the news, something of this magnitude. Well, the markets did just that, right? It did just that. Yeah. But like I said, the SPY SP 500, it didn't phase it. Because we know why the SPY is up. We talked about that the other night too. The SPY is up because of what? Stimulus. They ain't worried about no vaccine. They don't care about no Moderna, no Pfizer, no BioNTech. They worried about one thing and one thing only. Stimulus, because that's what's going to move the market. The vaccine news alone is not enough. It's not enough. They had this moment in the very beginning when they began to announce these certain dates a month ago. Of course, the stocks would just break it out. Everything seemed like, right? But not the case now. Clearly not the case right now, right? So really good information here. Good little learning curve here. Really good little moment to, again, see these things happening in real time and then be able to ID exactly what was the end result, right? Or we would say the reasoning, right? It's not hard to figure out what's the reasoning, right? Clearly buy the news, buy the rumor, sell the news event, right? And if they decide to come back in tomorrow and buy this dip, well, of course, that's going to be on, that's going to be on them trying to play us as a short window, right? But it made its money today. You got to hand it to it. I cashed my little bag out earlier, bought a little laid off some lows, bounced and made some good little, headway on that one. Yeah. But again, that's the strength of the markets on both sides, the good and the bad. You make money going up, you make money going down. And when it settles, it'll pull like a Pfizer did, because Pfizer actually went up too. Then it stayed up until a little bit to the next day, and then it sold off during the intraday session. And then Pfizer didn't, look back until it got back down towards like 38, near 38, 30, somewhere around there on the lows. And it's just now starting to kind of bounce a little bit now after selling off for the last 2-3 days. So Moderna is that same boat. They're under that 3-day rule, so to speak. All right? They're gonna have to have something happen that really spark another interest. And of course, like if they get like an upgrade or something, right? Analysts will upgrade based on what? Even vaccine news, right? With this data here, just the approval. Right? Which a lot of people could say, they'll tell you, well, we already knew it's gonna get approved. So like what's where's the underlying speculative move in that? Besides the fact that everybody thought it was a sure end, they crowded the trade, and it didn't necessarily pan out for them if they bought not having enough time for the stock to kind of move. Like you buying next week or into January, then of course even when it's pulling back, you just like, okay, yes, all right, I got time. Right, so that's basically what we got out of this today. I hope y'all guys enjoyed this. This was awesome. Again, it's the data and the knowledge, right? That's what we come in for. We come here to get the knowledge. Some of you guys may have never sat through a FDA review panel. That's exactly how the panels be. And sometimes, it could be a stock that you may be invested in, right? Or thinking about taking a position on, and you'll get a chance to hear firsthand, how they fared, and then how these, so-called scientists are looking at data. Now, one thing we can say about that vote, this vote here was way better than Pfizer vote. They didn't waste no time. This one had no nays or no's. It was all yeses and one abstain. So like I've been saying, the Moderna vaccine is probably going to be the best one in America. Hands down. Hands down. All right. So the stock has already made a big monumental run. It's still holding on for dear life. And I say that because, again, people still feel like the stock should be a lot higher, and that's typical, right? That's always going to be typical. Everybody want their stock to win. Everybody. Right? Sometimes it's just not the case, and you just got to learn how to be patient, strategize around it, and wait. So again, so that's it. So I hope you guys found that informational, right? A lot of good information there to kind of parse, right? Like straws a little bit. But they gave you guys a cornucopia of different vantage points or viewpoints of how they were looking at the Moderna vaccine. And again, for what it's worth, you could tell they were way more bullish on Moderna vaccine than they was the Pfizer one. That's why it came into age, this whole idea of this young adult classification with any child at the age of 16. Now they up to the 18, I guess. Right? I mean, really good news for Moderna in that perspective, at least. All right, so you gotta take your losses alongside your wins, and realize that it's not gonna have it all a perfect scenario all the time. It's not gonna be 100% perfect. All right, so that's it, guys. I hope y'all enjoyed today's marathon sprint. This gauntlet of data that we sat through. Right. Of course, I'm gonna upload it over there on the other channel once it gets done rendering and all that, whatever. So that way, at least it'll be a copy of it over there too, right? So if a person want to come back and, of course, you can always go to the FDA site as well, but go back and listen to the channel, the broadcast again. You'll have it at your disposal. All right. So that's it. So Moderna here fighting for 139 support. All right. 140 seem to be again still playing. It's still playing the role of resistance, right? And so we'll see here. So clearly, it didn't close above that point of resistance for at least 5 minutes, right? Let me see what I got here. I got, or did it? Let's see. 40. Where are we? All right, so 1, 2, 3, 4, 5. So right at like 5 candles. Ha! Couldn't make this stuff up. Look at that there. That's the preciseness of the stuff. That is the preciseness of this stuff. Right. 1, 2, 3, 4, 5. This fifth candle came right back down to the point where it wasn't allowed the stock momentum to close above it, and that's why it broke down a little bit more. Of course, now it just consolidate. But I'm just showing you how to fine tooth, comb technical aspects of how some of this stuff just mind boggling. How other people or agos are seeing the market in some cases. Right. All right, so that's it. So that's our good old buddy Moderna. Going through it right now, a little bit after hours, and a lot of people, I'm pretty sure, on the message boards are cursing like they always do. And again, like I said, it didn't really affect the SPY. Look at the SPY, because the S&P 500 wants them stimulus checks. They want real money. They're like, vaccine is nice and all, but we want to cut some checks, either to themselves or the American people. Nonetheless, they're waiting. I mean, they're waiting on stimulus, right? Obviously, they're waiting on stimulus. So I think that's about it. Now tomorrow will be interesting because again, we closing out the week. We got quadruple witching tomorrow. If you don't know what that means or what that's all about, I would say definitely go to Google and type in quad, like a little 4-wheeler quad, druple, always in one word, quadruple, right? You know what that is at least. Witching, right? And that's what's gonna go down tomorrow. It's gonna be, the market's gonna be mad volatile. And they typically do it on the back end of the day too. So that way it don't disrupt the early morning ebb and flow. So they'll do it maybe like 30 minutes before the bell or sometime an hour before the bell or whatever the case. But when you see that's happening and the markets are all over the place, you'll know that it is quadruple witching tomorrow, which it takes effect tomorrow. All right. So yeah, I got SPY calls right now too, still long. So right now they're still printing money. Still happy about that trade. And the rest is what they say, history. Right, the rest is history. get excited. All right, so I think that's it. I don't think I missed anything, obviously, outside of the typical stuff we talk about. So yeah, I think that's it. I was over there checking the phone too. All right. So thank y'all for tuning in. Of course, I got to get ready for some other little duties here. Thank y'all for being stewards of information. Thank y'all for being warriors or aliens in your own right, because what we do is people ain't, if they just only knew, right? If they only knew, right? But shout out to you guys for taking a chance on yourselves to put yourself in a position where you can also, make your own money. Right? So it goes without saying, right? So that's it, family. Thank y'all for tuning in. Make sure you like on the way out the door. If you haven't already, subscribe to the channel if you're new. Of course, and go, if you haven't subscribed to the original channel, African Emporium, the regular African Emporium, not African Emporium 2, then go subscribe to that channel as well. We finally hit another threshold over there where we crossed over 2,000 subscribers. So like I was saying earlier, what we're going to do another t-shirt giveaway, right? The followers definitely picked up a good little amount, especially after, at a point where there was a couple of the books that I uploaded began to catch a lot of traction. So that grew to the subscriber base pretty much, very fast overnight. So as opposed to it being, something smaller, we're just going to make it a 2,000 subscriber event. And I'll give away, again, another t-shirt moment. So stay tuned for that, for the t-shirt giveaway. And we'll just keep plugging at this stuff, guys. Right? All right. So on the way out the door, let's see if we got anything we can ride to on the way out the door. All right. Of course, Joe Biden then won. Now he can. Well, I didn't say he hasn't officially won to January the 6th, right? That's what Trump banking on. But we'll just go ahead and say, yeah, he didn't defeat Trump, right? He put them hands on Trump. Biden put them hands or them paws on Trump, and Trump can't take that right now. So we'll let balloon hand Biden take us out with some of that punching bag energy. And we'll see you guys on the next live stream. Thank y'all for tuning in.
World full of busy bees working. I ain't never met a free person. I don't get along with these earthlings. I'm trying to seek purpose, fight for something I believe's worth it. Feel the curse, I'ma reverse it. I don't dream cause I sleep nervous. Yet I keep flirting, face death like a Steve Irwin. Whole heat, I'ma drink thermos. Tell the bartender keep serving. I'm trying to be perfect, using dirty shit to clean dirt with. Only happy when the weed burning. Traded oxygen to breathe herping. Till I see curtains, lungs fail and they cease working. Error something that I need urgent. Suffocating, trying to squeeze words in.
Thank y'all for tuning in. Peace, bye.